Good morning, everyone. Welcome to the European Medicines Agency's headquarters in Amsterdam for the joint Heads of Medicines Agency EMA workshop on real-world evidence methods that has been organised under the auspices of the Big Data Steering Group. It's a great pleasure to have you here. We have, and people are still arriving through the door, there must have been a bottleneck at reception, about 80 people attending in person, and we had 900 registrations for online. So interesting to see the huge level of interest and support and engagement with real-world evidence methods. Just to introduce myself, my name is Peter Arlott. I head the Data Analytics and Methods Task Force at the European Medicines Agency, and I'm co-chair of the Big Data Steering Group with Jeppe Larsen, who is also going to be co-chairing today's meeting with me. Jeppe is the Director of Medical Devices at the Danish Medicines Agency and a member of the DKMA Senior Management Team. Little bit of etiquette for those that are online. You'll be your microphones are muted. If you want to intervene, please raise your hand, and we'll try and include as many people as possible, and we'll also try and balance between stakeholders as much as possible. Feel free to use the chat box, but we can't guarantee to pick up everything in the chat. But often you get interesting side discussions, so feel creative. Just in terms of bandwidth and so on, if you're not speaking, if this is to the colleagues that are remote, then please turn your camera off. But when you do speak, it's quite nice if you do turn your camera on so that people can see you. And of course, do introduce yourself, because it's, I think, quite important to know the affiliations of the people making interventions. So on that note, and welcoming the colleagues that are still trying to find a seat, welcome, make yourself comfortable. It's a great pleasure then to open the workshop and to introduce EMA Cook, Executive Director of the European Medicines Agency, to give some opening remarks. Over to you, EMA.
Thank you very much, Peter, and warm welcome from me too. And I'm sorry I can't be with you today. I hope it's a nice morning there in Amsterdam, but it really is my great pleasure to open this joint HMA EMA Big Data Steering Group workshop on real-world evidence methods. So let me warmly welcome the large group of experts with us today. And I know we have representatives from regulatory agencies, from health technology assessment bodies, from industry, from academia, research organizations, patients associations, healthcare professionals, and of course the European Commission. Now, as you're all very keenly aware, clinical evidence is pivotal for the development and authorization of medicines, and clinical trials are recognized as the gold standard for assessing efficacy and safety. But real-world evidence is increasingly recognized for the potential to complement clinical trial data, particularly in post-marketing surveillance and in conditions where randomized controlled trials can be challenging or difficult to conduct. This is why the European Medicines Agency and the Heads of Medicines Agency have a commitment and ambition to integrate real-world evidence into the regulatory framework. And you see this in our initiatives such as Darwin EU, the recent adoption of a metadata list for real-world data, the launch of catalogues of real-world data sources and real-world data studies, the data quality framework for EU medicines regulation, and the vaccine monitoring platform. A comprehensive real-world evidence framework relies on high-quality data sources that are fit for purpose. And it's only high-quality data that will ensure that it can be reliably used for regulatory decision-making. Linking real-world data sources with valid methods is essential to inform decision-making. Our Big Data Steering Group and our Methodology Working Party are instrumental in shaping the use of real-world evidence within EMA's regulatory processes. The Methodology Working Party is the engine room for the Big Data Steering Group and is responsible for guidance development, for stakeholder engagement, and the facilitation of workshops such as the one you're at today. This workshop is a platform to discuss the practical application of real-world evidence methodologies, fostering dialogue with experts and stakeholders, and defining directions for real-world evidence and regulatory decision-making for the future. The insights and outcomes of this workshop will contribute to the development of further guidance on the use of real-world evidence. This is essential for informed regulatory decisions that ultimately impact patient care and public health, both in the EU and beyond. So let me conclude by encouraging you all to provide your views and to share your ideas on areas for improvement and collaboration, because this will help us shape recommendations for future work in the field of evidence generation based on real-world data. And this will ultimately lead to improved patient outcomes. So thank you very much for your engagement, and I look forward to seeing the results of this very fruitful day. Thank you, and I wish you a wonderful session.
Thanks very much, EMA, and we are indeed looking forward to a great day. You asked if it was a nice morning in Amsterdam. It's a sizzling 15 degrees and there's a light drizzle. So if you like light drizzle, it is indeed a nice morning. So thank you very much indeed. Have a great day. So great pleasure then to introduce again Jeppe Larsen from co-chair of Big Data Steering Group and from the DKMA to give a few additional opening remarks, perhaps more from an HMA perspective. Over to you, sir.
Thank you very much, Peter. And also, just on behalf of the Big Data Steering Group, I also like to say I'm glad to welcome all of the many participants who are both virtual and physical here in the room to participate in this workshop here on the subject of real-world evidence and methods. I think throughout the last couple of years, we've been working within the network of the Big Data Steering Group and also within the methodology working party on how to identify and also harvest a lot of those benefits that we are expecting the real-world evidence and also real-world data they can bring us. But I also think the expectations on the benefits from real-world evidence or also real-world data, they're great, both from the world of academia, from industry, regulators, and also from the healthcare sector. They are extensive, so therefore the question is also, how can we deliver on these expectations and how can we actually harvest these benefits? And I think within the Big Data Steering Group network, we have been working on how to establish both knowledge and the tools that are needed to harvest the benefits and also to build the competences across the network, both within the EMA and within the national competent authorities to ensure that we are actually able to deliver. As part of our work within the network, we have launched both the HMA, EMA catalogs on real-world data sources and studies. To main one, we have had several training activities on both data, methods, and also the conduct of data to ensure the validity of the data. And then I also just mention also, I think the medical device, traditionally it has not been part of the focus within the network, but it's also started to gain more focus since we are expecting to see more and more mergers of technologies, which also generates a need to ensure more uniform approach to the clinical evidence within the both worlds. So here, just as my final word also here, is we also need more extensive knowledge, and also just to echo EMA, then for the BDS Big Data Steering Group, we also hope for a good dialogue and also a good participation today, since that is also what we need in order to get further from where we are within the network, so we are all able to gain from the benefits we are seeing and expecting from real-world data and real-world evidence. So thank you, and I'll pass the word back to, I think it's you, Patrice.
So indeed, maybe I can, thank you, Jeppe, thank you very much indeed. Maybe I just give a couple of words of introduction. Patrice Verpiat joined EMA about 15 months ago as our head of real-world evidence, so big important responsibilities, and he's going to give us a scene-setting presentation, particularly what we're hoping to achieve on the workshop today. So over to you, Patrice. And if I can encourage you to go to the podium. Where are you? I can't see. Oh, it's there. Go to the podium, please, Patrice.
Okay, thank you. So maybe, indeed, thanks for the introduction, Peter. And next slide, please. Yes, I've been asked indeed to do a scene setting and to describe a little bit how the day will be organized. So if you move to the next slide. Yes, I think you've seen this already for several times, so maybe hundreds of times already, but that's critical when it comes to our strategy. And when I say our, it's indeed European Medicine Regulatory Network for 2025. We would like to indeed enable the use of real-world evidence and also establish its value across the full spectrum of regulatory use cases, as highlighted indeed in this paper that has been published in 22. And if you move to the next, it shows that, in fact, real-world data and real-world evidence, which are the topic of today, play a critical role, in fact, in bridging the gap between what we call clinical research and practice. And how do we do this? It will be indeed by ensuring that evidence generation is planned and guided by data, knowledge, but also expertise, that research question drives evidence choice and not the opposite, and embrace spectrum of data and methods. For sure, clinical trials remain the core of this evidence, specifically clinical evidence, but are also bigger, better, and faster. And you know already about real-world evidence, but what's also critical is that the patient voice guides every step of the way. The different healthcare systems are supported in their choices, and there is also a high level of transparency, because that's important to underpin societal trust. Yes, I can do it myself. Perfect. So in fact, when it comes to real-world data and real-world evidence, there are different methods, and we're all very familiar with this, and some of them will be the topic of today. So for sure, at the top, non-interventional studies, but also causal inference methods to estimate causal effects from observational data while accounting for confounding factors at best. Then we have the target trial emulation, which will be one of the topic of today, which is a method that aims to emulate a hypothetical randomized clinical trial using observational data. We have also PCTs, pragmatic clinical trials, which are designed to test the effectiveness of intervention in real-world clinical setting. And we have also now more and more machine learnings and artificial intelligence, which are techniques that can analyze large volumes of real-world data to identify patterns, predict outcomes, and generate evidence, as well as natural language processing. But there are also challenges and opportunities like data quality and completeness. We have also ethical consideration that needs to ensure the right balance between privacy and data sharing. We have methodological rigor, which is mandatory, more or less. Generalizability, which is assessing also how findings apply to broader population. And collaboration by engaging different stakeholders, clinicians, researchers, policymakers, to maximize real-world evidence research. So you will see that most, if not all of them, will be addressed today again. So in fact, we have also to take into account the current landscape, specifically when it comes to the EU guidance. And there is a series of guidances that have been released over the last years at the EU level. So starting by this guideline on research registry-based studies, and then we had also additional guidances linked to data or metadata, which was linked to the research catalog. Then we have the data quality framework, released at the end of last year, with a specific dedicated real-world data chapter that will be also coming by the end of this year. We have the catalogs. We will talk a little bit more about this, and also ongoing revision of the GVP8. So that's the current EU landscape, and more to come. What's also another important thing is that the methodology working party has been established a few years ago, established by the CHMP to leverage the expertise in some areas which are embedded into real-world evidence, for sure, biostats, modeling simulation, PK, and pharmacogenomics. So the MWP, as we say, is really here to support the different EMA committees and the SWOP, but also to engage with the different stakeholders, but also to prepare, review, update guidelines, concept papers, training and workshops for assessor. And you will see that the first part of this workshop will be dedicated to the reflection paper published recently, which is currently under public consultation. But these expert groups are nominated by the CHMP and have also drafted a 3-year work plan, and this work plan will be one of the topics addressed at the end of the day. So I was talking about indeed the EU context, but we are now operating in a global world, so that's why it's important also to consider the international aspect. For this, we have on the left side the ICH M14 guideline, which has been indeed published and for public consultation, released very recently up to the end of August. And this first dedicated ICH guidelines on real-world data, real-world evidence is on non-interventional pharmacoepidemiological studies for safety assessment of medicine. In addition, following the ICMA restatement published in July 22, we have also worked with Health Canada and US FDA on an ICH reflection paper, which is entitled now Pursuing Opportunities for Harmonization in Using Real-World Data to Generate Real-World Evidence with a focus on effectiveness of medicines. And the proposal is to have two initial guidelines, one on real-world data, real-world evidence terminology, metadata, and assessment principle, and the second one on the protocol and report format, as well as study transparency. And this will be the two initial steps before additional potential guidelines. And I can share with you that this reflection paper has been adopted by the ICH General Assembly just 10 days ago. So we have a go now to move to the next steps. So this was about the environment, the scene setting. When it comes to the workshop today, more specifically, so our objective is definitely to hear the views of the different stakeholders, interested parties, and experts on, as I said, the draft reflection paper on real-world evidence, which is currently open for public consultation, released by the MWP and CHMP, but also on priorities for further regulatory guidance, development, and collaboration beyond this reflection paper. And it's a way for us to definitely engage with all of you on the next methods to come and to consider in regulatory decision-making. The way the day will be structured is about the first session, which we'll discuss after having presented the reflection paper. Then we'll move to a section where we will have more targeted, I will say, topics on methods, so the target trial elimination and estimate frameworks, but also external controls used based on real-world data used for clinical trials. And at the end of the day, we will move to this next 3 years, which is linked to the roadmap developed by the MWP work plan. So I hope with this that we will have indeed, as said by Emmer, a nice workshop and great discussions. So looking forward for having this. Thanks a lot.
Thanks very much, Patrice. Very clear, and I think we've got a really interesting workshop in store. So our next introductory remarks come from Thomas Delog from the European Commission's Directorate General for Research and Innovation, where he's head of sector for health industry. So over to you, Thomas.
Thank you very much, Peter. Good morning, everyone. And thanks a lot for the permission to speak from this place where I have a bit better control of all the equipment. So I will present a few developments happening at the EU level, which are relevant to real-world evidence from the technical and from the regulatory point of view. Let me start from the legislation. The main legislative initiative is the European health data space. It's a big piece of legislation which sets the ground for the rational use of health data. The main objective of EHDS is to facilitate the exchange and use of health data at the EU level, but also to empower the citizens to take control of their data. The EHDS introduces two types of data use. The first one, primary use, is for the delivery of healthcare, and the secondary use is for research and innovation, policy making, and regulatory activities. The EHDS is now reaching the final steps of the adoption process, and it is expected to finish in autumn 2024. So over the years to come, this is the legislation which will guide the data aspects of health research, and I believe it will affect all of us as patients or prospective patients, but also those who work with health data on the daily basis. I will now present several research initiatives that we funded across the years under the main program of Horizon Europe and under the Innovative Health Initiative. To start with, 2 years ago, we published a call under Horizon Europe, whose objective was to develop evidentiary standards for the analysis of real-world data or synthetic data, and to develop methodological standards and machine learning methods to help analyze real-world data for decision-making in regulatory and in HTA processes. The call resulted in 6 projects that now form the METRIL cluster. Most of these projects started in early 2023, so we are now very much looking forward to start receiving the first deliverables and reports. These projects cover, for instance, the use of registries and the use of AI in regulatory and in HTA decision-making in several health conditions. All these projects deserve a lot of attention, but because of the limited time we have here, I will mention just one, which is called the REAL for Reg. The objective of this project is to develop new methods based on AI and machine learning to analyze real-world evidence in regulatory and HTA decision-making. The consortium will conduct a retrospective observational cohort study based on national registries and on medical claims data. The group will run several use cases on breast cancer and ALS, which are aligned with the regulatory decision-making cycles of pre-authorization, evaluation, and post-authorization. In fact, which is not in the slide is that the project is coordinated by BfArM, the German regulatory agency for pharmaceuticals and medical devices. The consortium also includes other partners with regulatory and HTA expertise from Portugal and the Netherlands, and also a patient organization. And we very much welcome this sort of cross-border and cross-institutional collaboration in all areas of health research, but especially in the area of data, which would affect the regulatory decision-making. Apart from the main program of Horizon Europe, we have also funded several data-intensive projects under the public-private partnerships with industry, the Innovative Medicines Initiative and the Innovative Health Initiative. And this latter one is the one that we are running now. It is a partnership with several health industry sectors, covering pharmaceuticals, vaccines, medical devices, diagnostics, and biotech. The projects which I will now mention are co-funded by the EU and by the industry, which shows also the high interest of private partners in data-intensive projects. The first one is AIDEN, actually a flagship in that area, funded under the Innovative Medicines Initiative, so in partnership with the pharma industry. AIDEN standardizes the health data to enhance interoperability and to accelerate evidence generation. This network now includes almost 200 partners in 29 countries with over 850 million records, which I believe is an impressive figure. The main goal of AIDEN is to bring various health data to a common standard, the OMOG model. This process is ongoing very, very successfully. And this network is being used to answer several real research questions, also under the EU Darwin Initiative, which I'm sure will be mentioned later today. The use case of AIDEN covered medical devices, concretely the knee replacement, pharmacovigilance, HTA, COVID, so a pretty broad area of application. Sadly, the project has now formally finished. This happened a few weeks ago, but more optimistically, we hope that the work will continue under the AIDEN Foundation, which is also a token of the fact that these kind of initiatives can become sustainable in the long run. The next project is called IDERA. It's a very recent one, started only a year ago under the IHI. The aim is to process, to access and integrate health data from various sources, including digital technologies, PROMs, clinical trials, and routine care. This project will use wearables and digital applications for patient monitoring and will provide real-time data for decision-making using lung cancer as the use case. And this project will be compliant with the HDS, which is a very forward-looking approach to build the true pan-European health data ecosystem. The next project, which started recently, is called IMPROVE, also funded under IHI. It will integrate patient-related data for value-based healthcare using also patient-reported outcomes and preferences, as well as real-world data from the wearables and apps. And this approach here will be tested in several use cases in ophthalmology, oncology, cardiovascular conditions, inflammation, and neurology. So I could continue like that for quite some time, but here is just the summary of the IHI data-intensive projects. It shows the volume of data handled by the project co-funded with industry under EMA and IHI. And actually, I should already update this slide because yesterday I received a very fresh news that the Conception project, which is here in the slide, working on pregnancy and breastfeeding, has access to real-world data comprising around 150 million records. So you can imagine the scale. These volumes of data are impressive, and this is actually a demonstration of the fact that the data revolution in health research is there already. And we are very much looking forward to learn about the outcomes of all these projects in the years to come. Now, going to a bit different aspect, regulatory and policy. In February, the IHI held the Regulatory Science Summit. It was held in Brussels as a neutral platform for regulators to discuss various topics related to research and innovation. Here, the participants included stakeholders from industry. We had several regulators, including many colleagues here from the EMA, from national competent authorities for both pharmaceuticals and medical devices, and also colleagues from the FDA online. We also had a representation of national bodies, HTA stakeholders, a few departments of the Commission, and of course the IHI colleagues as the organizers. There were 5 sessions in the summit: rare diseases, pediatrics, real-world data, AI in healthcare, and regulatory sandboxes for enabling disruptive innovation. We had very good participation with over 50 participants on site in Brussels and over 50 online. And for the sake of time, I will not go through all the content, of course, but will focus on just one session on the real-world data. We discussed the generation of evidence that meets diverse information needs and requirements, including the alignment on common methodologies. We also highlighted the importance of integrating different sources of evidence, including from registries. And as expected for this kind of session, we also discussed the use of non-randomized controlled trial designs. There were exchanges on regulatory acceptance of data from such trials, in addition to acceptance from HTA stakeholders. And in the context of future initiatives, the group emphasized that all stakeholders should combine the efforts towards greater predictability and understanding of the use of real-world evidence for decision-making. Interestingly, we also consider the proliferation of real-world evidence frameworks. This trend can improve the quality and relevance of evidence using clinical and regulatory decision-making, but it also presents challenges in data standardization, privacy, and biases. And now, going still to the future and the funding opportunities. To conclude, I'd like to say that we will continue to invest in the development of methods using real-world evidence. The next such opportunity will come with the next IHI topic on clinical study endpoints using digital health technologies. This scope will cover new methods for analyzing patient preference information, clinical outcome assessment, measures derived from digital health technologies, all that in order to better quantify the benefits of therapies to patients. So you're all very welcome to apply to this topic. The first deadline will be in October. And now, an information which is maybe not so actionable to you, but probably also of interest, is that we had another topic under IHI on new methods for analyzing patient preference information. The call is now closed, and we hope to see the project outcomes quite soon in the future. So there is more to come. With that, many thanks for attention, wishing us all a great day and very insightful discussions. Thank you.
Thanks very much, Thomas. So we have finished our welcome addresses, and hopefully they have orientated you. And Thomas, I understood that all of that research funding and the collaborative efforts should, in time, give us better access to data, better methods, and through those methods, better evidence for decision-making in public health. So great to see that work. Let's then move on to session 1, and it's a pleasure to introduce 2 co-chairs, Kit Roos, who's Professor of Biostatistics at Radboud University Medical Center in Nijmegen, and also, importantly, the chair of the Methodology Working Party, and Mencia de Lemus, who is a member of the Committee on Advanced Therapies at EMA, where she represents patients. She was formerly president of Spinal Muscular Atrophy Europe, an umbrella association representing 25 national patient organizations across Europe. Mencia, Kit, over to the 2 of you. Thank you.
Yeah, thanks, Peter, for the introduction, and also thanks for setting the scene both by to also sketch the broader and then I think inspiring environment. We will have the discussions today, but we are diving into details first. We will our first session will be dedicated to the draft reflection paper on non-observational or non-interventional studies, followed by a panel discussion. And it's my pleasure to introduce Xavier Kurtz, who will be giving the presentation on the reflection paper, a medical doctor by origin and a PhD in epidemiology and biostatistics, and importantly has a long track record at the European Medicines Agency as head of the data analytics and work stream until quite recently, last year. He's officially retired, but I think we all noticed that's not really the case. And I think it's essentially also his personal drive which helps putting these topics forward. So I'm happy to give you the floor to introduce the reflection paper.
Thank you, Kit, and it's a great pleasure to present this draft reflection paper on behalf of the drafting group of the Methodological Working Party, and the people who have developed this reflection paper are listed here: Olaf, Carla, Stine, Juan Roa, and myself. I have to say that my voice has not fully recovered from a cold, so my presentation will not be too long, and if needed, Juan Roa will take over. So this reflection paper has been developed for some time now, since November 2022, and there has been several iterations with 5 drafts, which have been presented to and reviewed by the main committees and working parties who are mainly involved in the evaluation of data using real-world data. So the draft reflection paper has been approved for public consultation by CHMP in April and published last month. So the public consultation is for 3 months duration until end of August. Let's start with some definitions. So we have defined real-world data as data that describe patient characteristics, including treatment, utilization, and outcomes in routine clinical practice. Real-world evidence as evidence derived from the analysis of real-world data, and the non-interventional study. For that, we used the definition from the EU legislation, from the regulation 536/14 on clinical trials on medicinal product for human use. So it is a clinical study that does not fulfill any of the conditions defining a clinical trial. These conditions are listed here, but they are published, of course, in the regulation. I'm not going to cover them. We spent some time on the definition of real-world data. The definition that we propose here diverges slightly from other definitions, as it focuses only on the nature of the data, the patient characteristics in routine clinical practice. And the main reason is that for us, the nature of the data is what really makes the specificity of this data, of the real-world data, in order to fill knowledge gaps in medicines regulation. For many questions, what is relevant is how a drug is used, how safe and effective it is in routine clinical practice, and therefore this is why we focused on this in the definition. The environment has changed over the last decade. This was already mentioned by Emma. So, of course, clinical trials are the main source of evidence to evaluate the benefit-risk profile of medicines in marketing authorization procedures, and non-interventional studies are often used to support safety assessment, perhaps less frequently to support efficacy due to methodological limitations. However, electronic healthcare data and registries now provide new opportunities to use real-world data, generate real-world evidence derived from the clinical practice. And I give you here some examples. We have a large number of examples of use of real-world data in regulatory assessment. For example, to understand the clinical context, provide data on disease epidemiology, standards of care, clinical practice, to support the feasibility assessment and the planning of non-interventional post-authorization safety, efficacy, and drug utilization studies, and of course to investigate associations and impact, for example, an association between an exposure and efficacy or safety outcomes, and also importantly to monitor the implementation and to measure the effectiveness of risk minimization measures. So the objectives of the reflection paper is to discuss the methodological aspects of NIS using real-world data to generate real-world evidence, to highlight potential limitations, and to reflect on how some of these limitations could be overcome or mitigated to increase the reliability of the evidence. I have to say that during the development phase of this reflection paper, we were sometimes asked, why do we need a reflection paper on real-world data? As real-world data are frequently used in traditional observational studies for which the methods are already known and well described. So our answer is that real-world data are only part of the different types of data that may be used in observational studies. And we wanted to focus on these data because these are probably the ones that are very important to provide new information in regulatory assessment for regulatory purposes by providing data on current medical practice. And the quality of this data is, of course, important because of the high public health impact of decisions that need to be made on this data. So this is the table of content of the reflection paper. I'm going to review them, the main aspect of these topics, but please read the full paper and provide comments. What I'm going to say here are just highlights from these points. So what is the scope of the paper? The design, conduct, and analysis of NIS using real-world data to generate real-world evidence for regulatory purpose. Each of these terms is actually important because they have an impact on what is included in the reflection paper. It is important to highlight that the use of real-world data in the context of clinical trials is out of scope. For example, we are not addressing the use of real-world data as external control groups for a clinical trial or as data source for patient recruitment for clinical trials. Of course, general methodological guidelines already exist for NIS, and that's why we focus really on principles considered critical for the conduct and assessment of NIS using real-world data for regulatory purposes. A question was debated of what do we include, but we think that real-world data can be collected through primary data collection as well as secondary use of data collected for another purpose. But in both cases, attention should be given to the influence of the primary purpose of data collections. An important difference that we make in the paper is the difference between studies with descriptive objectives, so which provide description without a causal hypothesis, and the studies with causal objective, which seek to measure associations in comparison with a non-exposure or an alternative exposure. This distinction is important because we think it has, I will show it, it has implications for the study design. A few words about the legal and regulatory requirements. As I already said, so a clinical study is defined in the regulation and it is classified as either a clinical trial or a non-interventional study. And in principle, real-world data may be used in both. There is no overall regulatory requirement to use real-world data. However, the regulatory assessment requires that the evidence provided by applicants is valid and reliable to support a specific regulatory objective. Therefore, on a case-by-case basis, the use of real-world data may be desirable or may be required to fill knowledge gaps for the regulatory objective. There is no rule that real-world data should be used. And this is the objective of the study, is not to use real-world data. It's really to provide evidence supporting the regulatory assessment. So this means that the relevance and the suitability of real-world data for a specific application is to be discussed with regulators at an early stage of product or protocol development. We were frequently asked during the development of the reflection paper if we could provide guidance on when real-world data would be accepted, in which conditions, in which application they can be submitted or they should be submitted. We cannot provide this information because this is really on a case-by-case basis based on the research question and the regulatory application. I would like also to reiterate that there are already some guidance existing, the GVP that they need to be followed, the GVP Module 8, the scientific guidance on post-authorization efficacy studies, and there is, of course, the guidance on registry-based studies applicable to real-world data and real-world evidence. There are some overlap between this reflection paper and the guidance on registry-based studies, but normally there are no contradictions. Let's go to the study design. Again, we are repeating it, but it's because it is important. The choice of the study design is to be primarily driven by the need to obtain reliable evidence for the research question. So the use of real-world data is not the objective per se of a study. And the applicable methodological standards should be applied. We introduce we extended what is in the guidance on registry-based studies about the feasibility analysis, but we extended it to the feasibility assessment that is recommended to be performed as a basis of early discussions with regulators. So it doesn't replace the protocol, but it is a preliminary steps. And it may include a number of elements that would be useful to assess or address whether the data source and the design would be adequate for the research questions. The evaluation of the fitness for purpose of the candidate data source, the feasibility to implement the proposed study design. There may be a reference to the feasibility of the target trial emulation approach if it is used and applicable. And then the feasibility assessment should lead to a conclusion discussing the relevance of the real-world data source and the study design that is applied to generate the required evidence. And if there are different options possible, then they should be proposed and discussed with regulators. Now let's go to the studies with descriptive objectives. They may have very different goals, so we cannot provide a general guidance or recommendation or reflections about this, the design. They may vary and they should follow the best practice for the research questions. And again, we make reference to the NSEP guide. The results may be influenced by the setting in which they are observed, because there are many descriptive. And in this case, and if this is relevant for the research questions, the conditions that may influence the result of the study should be addressed. And this may include the setting and the type of available data, the healthcare system of the country where the data are collected, that it may influence the availability and accessibility of the data, the environment in which the data are collected, what are the prescribing practices in the country, the prescribing formularies, and so on, and the specificities of the data source in terms of terminologies, use of a common data model, and data quality. All these aspects may influence the result in terms of description, and I will address the heterogeneity aspect later on. Studies with causal objective. Again, the challenge here is to provide valid evidence in comparison with no exposure or alternative exposure, taking into account the risk of distortion of this result coming from the presence of selection bias, information bias, and confounding. And we state that the target trial emulation framework should be considered as a strategy. And there would be several advantages for it. First of all, the framework has been developed for non-interventional studies, but it has similarities with clinical trials. Therefore, it facilitates the overall contribution of the study to in the application dossier by complementing the evidence from clinical trials with some more coherence between the clinical trials and the NIS. It also helps the investigators to consider potential bias and adequate methods to address them. It provides a high level of transparency on the study design, the assumptions needed to emulate the trial, and the definition of the causal effects, and facilitate, therefore, the evaluation of the replicability of the study by others. It has been shown to mitigate some bias, such as the prevalent user bias and the immortal time bias. In the reflection paper, there are the reference for these publications. And also the estimate framework should be considered in order to increase, again, the coherence between what is being done for clinical trials and for NIS. Just a comment is that actually the target trial emulation framework could be considered as doing explicitly what regulators often do implicitly. When a clinical trial is not feasible, is not possible, and that a non-interventional study is submitted, often what is being done is to say, when in the evaluation is to say, yes, but what would have been in the ideal situation, what would have been the ideal clinical trial? And how does this study compares with the ideal clinical trial? So there are often some implicit trial emulation when there is a submission of non-interventional studies. And this target trial emulation framework then do it actually explicitly in the by comparing the design of boards. This is an example of a study using a target trial emulation approach. I will not go through it, but I just want to say that this type of table is very useful actually, because it identifies the protocol, what are the different protocol components, what would have been the specificities of these components in a hypothetical trial, and then what the emulation is doing, whether some of these criteria are the same, or if there are some change, or some are not possible, so there are some variation and so on. Now let's go to the important chapter of bias and confounding, and I will start with the selection bias, which is selection of patients based on a number of criteria. And it is important that there is an adequate definition and justification of inclusion and exclusion criteria, including the specification of the codes and the algorithms that are being used, taking into account a possible misclassification. And investigators could even consider wide inclusion criteria, allowing then at the analysis stage to make restriction, to make stratification and sensitivity analysis. We would like to highlight also that, and this is an experience from the COVID-19 and the studies with vaccines, that there are sometimes implicit selection criteria that are not fully considered, and that result from the method used to identify the study population and define exposure categories. For example, the health-seeking behaviors leading to comparison of different population when for people who have been vaccinated or not. If there are concerns about the generalizability of the association, there should be a comparison made. There could be a comparison made between the cohort's key characteristics and those of the target population using published research on national statistics. And then there is recommendations to use a new user design because, well, it is well known that using prevalent drug users may select drug users who are survivors of previous drug use. Some studies have a single cohort and then use an external or historical cohort used for comparison. This is liable to a lot of different types of source of selection. And therefore, in this case, the decision should be justified. And the likelihood of bias and confounding should be discussed with special attention to potential differences in selection criteria. Information bias, so resulting from misclassification or inaccurate measurements. This may happen at all stages of the data collection and extraction pathway. So these steps should be identified and they should be verified in order to see if the data source contains enough details on exposure and outcomes to correctly classify the patients. So it would be important to know whether there is a likelihood of misclassification. If there are any previous validation study made by a previous study, this should be identified and evaluated, and it's possible that in some case, a new validation study may be proposed. We would like to recommend to avoid assumptions about the direction of misclassification, at least in the design stage. So often there is a discussion section of study result that the misclassification is non-differential and therefore is not important and so on. These are based on assumptions that are rarely verified. So we would like to avoid these assumptions and also highlighting that their impact depends on the study objectives and that it is more important to identify, minimize, and measure potential misclassification of the key variables at the design stage. Misclassification may occur in defining the date and the timing of different milestones for the study, and this should be taken into account and attention to be given to them. Time-related bias is an important aspect. This is a type of misclassification, but the relevant time period should be identified at the design stage in order to plan the data collection and extraction of important dates and the change of the patient status over time. And there are some tools, a graphical representation that may help identifying what are the different milestones, the different time periods, and help understand what are the classification of these time periods according to the exposure categories. This is an example which is based on the cohort entry date, but there are other different examples. Confounding, I'm not going to be very long on it because it is now that, well, methods to take into account to confounding in the analysis stage, but it is important to identify then the potential confounders at the design stage in order to plan the data collection and analysis and to evaluate whether the data source has the necessary information in order to be able to control for confounding. It is recommended to use active comparators because there may people using an active comparator and the comparator of interest may be more similar than if there are no non-active comparator. And there is a discussion that I'm not going to explain here, but on the use of negative or positive control exposure or outcome, which may reveal residual confounding in one sense, in one direction or the other, and may help in the interpretation appraisal of results. So this is important. We consider it important in order to help to interpret the results. And the choice of the different in the design of negative or positive control exposure or outcome control may depend on the research question and the available data. For example, if the question is on safety, efficacy or effectiveness, and so on. Effect modification, we have a short section on it, is because it's again a matter of having identified what are potential effect modification in order to collect and extract the relevant data from the data source and to plan the data analysis. It's too late at the analysis stage when there are no data to say that it's possible that there are differences because there are effect modifiers. Governance and transparency, it is important, or even it is a rather short chapter of the reflection paper. And we stress that the governance of the real-world data source should be made available because it may help understand any restrictions related to the conditions of access, to the availability and to the publication of data, and understand the limitations of the data source and if there are some limitations that could have been solved or not. For studies that are contracted, the principles of the NCEP Code of Conduct should be applied in order to maintain scientific independence. So this is a general principle. And it is encouraged that, as in other guidelines, as in the GVP, to register the NIS in the HMA, EMA catalogs of studies together with a study protocol and study report. And if possible, to have the data source used, registered also in the catalog, and have updated information. Of course, the investigators of a study do not own the data, and they are not the one responsible for doing it. But if there are contracts between a company and the data source owner, then this may be part of the contract to have the data source registered in the catalog. And then having publicly available the codes used for the creation analytical data set is important for the replicability of the study. Yeah, this is, I have a slide here on the catalog of real data source and study. It shows you the link, just to highlight you that it has been launched, and I think that some of colleagues are outside the room with a poster in order, and if you have questions, they are available to answer your questions. Data quality. We have adopted here the framework that has been published by the FDA. So we identify two main components of data quality: reliability and relevance. Reliability being a property of the data irrespective of the use in any study. It had a question of, do the data represent the intended underlying medical concept, and are they trustworthy and credible? This can be evaluated and documented by using a data quality framework, and also the standard data quality management applied to the data source should be documented. Relevance is study specific, and it had the questions of whether key data elements are available. Is the size of the population adequate in the data source? Is the population representative of the target population for the study objective, and so on? So this is, it identified the fitness of purpose of the data source for the specific study. Multi-database studies, we have a section on it, and the main message here is to say that several databases may be used in order to increase sample size, but this should not reduce the quality requirements, and the information on the reliability and relevance of each of the databases should be presented. For data linkage, the protocol should describe the data elements used in the data, but also the linkage methodology, and also an evaluation of the impact of imperfect matching, especially if probabilistic matching is being used. Data quality frameworks, you know that there's already several data quality frameworks existing. Then they will stay, and there will be the HMA EMA data quality framework for EU medicines has been published for consultation. So we are not expanding a lot on this section here currently in the reflection paper, because it is important to know what will be the final document for the data quality framework. But expertise should be developed, and use of this data quality framework is not necessary evident or easy. So expertise should be developed using at least one data quality framework. Now, the last section is on statistical analysis. And first of all, regarding estimation and hypothesis testing, this is a general principle that analysis of NIS should focus on estimation and confidence intervals. However, when using real-world data, we use often very large database. And in this case, the confidence interval are not always clinically relevant. If you have an estimate of 1.2 with a confidence interval of 1.1 to 1.3, this does not have a lot of meaning. So that's why we say in this case, the evaluation should be based on the clinical relevance, on the estimate, the clinical relevance, but also then an appraisal really of the study design, the analysis, and all the possible bias that could have occurred. Time-dependent analysis should be planned when appropriate to the research questions in the cohort studies where events occurred at different time points. This time-related bias, which I mentioned earlier, have been frequent problems when following patients, and especially we have had many examples in the COVID-19 period with vaccines. Stratified analysis, sensitivity analysis, and missing data. So we have short section on it. They should be pre-specified and discussed with regulators, and there are recommendations in the ICH E9 addendum on estimates, which Juanjo will present later on. Heterogeneity. So this is the last slide, but certainly not the least important one. There is an increased availability of electronic health care database from different countries, and multi-database are now frequently done to increase the sample size, provide data from different databases and countries to verify the generalizability of the results, and learn about clinical difference between databases or countries that may influence the results. However, and this is the experience that we have now, is that heterogeneity is observed. And this is an example, a simple example of heterogeneity in the incidence of venous thromboembolism in different European databases, with heterogeneity increasing with age. There may be different sources of heterogeneity, and they may be related to some database characteristics, such as the recording or the coding system, as opposed to true differences in populations. Now, it is useful and it is perhaps necessary to understand the impact of these artifacts, the differences in coding and recording. But we have to keep in mind also that understanding these artifacts, the coding system, what are the differences, is not generally an objective of the study. So that's why and if it is not an objective of the study, it is not a main aspect of the result. So that's why we really insist that this should be anticipated as far as possible in the study protocol, and the management of heterogeneity should be presented. And this comes back to the feasibility assessment, actually, that having a good information and evaluating what is the impact of differences in coding system is important, and selecting the data source that may be as similar as possible as in relation to the use of a common data model, the use of the setting, and so on. What has been said before is important. So heterogeneity is not an aspect that should be dealt with at the analysis stage, at the discussions section of the report. This is really something to be tackled at the design stage, and this is something that I wanted to stress, and that the reflection paper also stress about. And this was the last slide. Thank you for your attention.
Thank you very much, Javier, for an excellent summary of this important reflection paper and for highlighting the most important features of it. So we are now going to be moving to our panel. There are going to be representatives of the different stakeholders, that is industry, patients, academia, and healthcare practitioners, that will first provide a brief position and then will lead to a discussion. So it is my pleasure to introduce the representative of the industry, Dr. Almath Spooner. Dr. Spooner is head of the Europe Regulatory Policy and Intelligence at RWE. Dr. Spooner is a dual qualified pharmacist and barrister at law and earned a PhD at Trinity College Dublin. She has a mix of public and private sector experience acquired through working across institutions, organizations, and sectors over the course of 2 decades. Dr. Spooner held leadership roles at the Irish Health Authority and European Medicines Agency. She was the first elected vice chair of the PRAC committee. She has also represented the European Union at ICH and contributed to various international initiatives. Dr. Spooner has a specialist interest in data, clinical evidence generation, and digital innovation, and is the current chair of FPS Integrated Evidence Generation and Use Working Group. So the floor is yours. Thank you.
Thank you very much. On behalf of FPI, I'd really like to thank the agency and the EU Medicines Regulatory Network for the opportunity to contribute to today's dialogue. I'd like to start, and maybe moving to the next slide, please, by saying that FPI welcomes this reflection paper. We appreciate that there's been a lot of progress on establishing capacity to generate real-world evidence with work on data sources and data quality. And it's now timely to shift the focus to consideration of methodological principles and good science principles more generally to establish the value of and confidence in real-world evidence. FP considers that the reflection paper is consistent with current thinking and good research practices for the design and conduct of non-interventional studies, and is well aligned with the draft ICH M14 guidance that Patrice mentioned earlier. In general, we see a growing consensus that there are a spectrum of approaches to evidence generation and that the fitness for purpose of the approach will largely depend on the research question. And in that context, it seems important to ensure that the reflection paper provides a balanced assessment of clinical trials and non-interventional studies. Amongst peer medicines developers, questions around scope and terminology were the most frequent points raised, and appreciate the focus that Xavier Kurtz gave in his presentation to addressing some of these questions. Recognizing that there are ambiguities around definitions and terminologies, this reflection paper provides a really good opportunity to establish greater clarity and to encourage dialogue among stakeholders on terminology and definitions. Currently, the paper defines non-interventional studies by what they are not rather than what they are. And in many ways, the paper seems to address a subset rather than a totality of non-interventional studies. However, I think it was clear from Xavier's presentation that there has been explicit decisions made, for example, around excluding the use of RWD in the context of clinical trials, that a conscious decision was made to exclude those from scope. And we do hope that that signals an intent to develop further guidance to address the use of RWD in clinical trials. But we were curious about that decision. Moving then to the next slide. For medicines developers, I think the real test of any guidance is how it's applied in practice. And this workshop is an important opportunity for dialogue. We're delighted to have representatives of the committees and assessors here today. And Xavier emphasised that decisions on the relevance of a non-interventional study using RWD to generate RWE, those decisions will continue to be case-by-case decisions. And we can understand absolutely why regulators would say that. However, we would suggest that consistency in implementation would be greatly facilitated by the development of a fitness for purpose framework that allows at some level the identification of criteria that may indicate that RWE will be suitable for use in a particular scenario. And there are links there with hopefully future work at ICH. We encourage consideration of mechanisms for further enhancing shared learnings and transparency around how real-world evidence and non-interventional studies are used in regulatory decision-making, because scientific advice is clearly very important, but the learnings from that come down the road. So if we can have real-time sharing of learnings, I think that will help the community of researchers to make progress together. In relation to transparency, FP agrees that study results registration is one component of building trust and confidence in research, and we have made proposals, and we would refer workshop participants to a paper by Aka and al. That was published last year. A smaller point, and it was addressed to a degree in Xavier's presentation, we would suggest that the structure of the reflection paper could further clarify the applicability of relevant text to primary and secondary research, respectively. We read the paper as being applicable to both, but certain parts of the paper focus more on the latter, on secondary research. There doesn't appear to be mention of quality control during conduct. Primarily the focus is on during feasibility, but some points are more relevant to secondary data collection, and making this more explicit in the paper might be valuable. So to conclude, we welcome this reflection paper, and we look forward to contributing to the public consultation and with the conversations with all stakeholders today.
Yes, thank you very much. And for my brief summary is very relevant and important comments on scope, external use as external control data, and how that in the fit for purpose framework going beyond a case by case direction. I think that's what I understood from your feedback, and the primary and secondary data collection. In terms of how we run the discussion, we will give the panelists first the brief summary, and then the discussion will start. As a reminder for those online, beyond the 80 that are in the room, it's possible to also put your questions, suggestions, and any discussion points through the chat. It will not be verbal, but we will follow the chat, and we can't promise to answer everything, but we will monitor to see what we can enter into the discussion. With that said, I'm happy to give the floor to Helga Gardas-Dottir. She's a professor in the use of real-world data for decision-making on medicines at the Division of Pharmacoepidemiology and Clinical Pharmacology at Utrecht University, and adjunct professor of pharmacoepidemiology at the University of Iceland in Reykjavik in Iceland. And her research interests are very much in the area that we are tackling today, application and development of innovative approaches to generate and analyze real-world data on safety and effectiveness of medicines to inform regulatory and clinical decision-making. And she's also co-chair of the NSEP steering group, so very close to setting methodology standards. And I'm happy to give the floor to Helga.
Thank you, Keith, for this nice introduction. I, as other stakeholders here, welcome very much the reflection paper and the publication on how best to use real-world data for non-interventional studies. I think it provides a very well-structured guidance on important aspects related to designing such studies. And while acknowledging that regular decisions are made on the totality of the evidence package, and in the evidence package, the contribution of an individual non-interventional study may vary, we do need to apply higher standards when conducting these. And the reflection paper presents information on this key ingredient and what is expected with regards to data quality, ensuring reliability and relevance of the data, application of high methodological standards, and transparency in conduct and reporting. There's again all key ingredients needed to attain to build trust when using results originating from such studies. And I specifically welcome the explicit focus and attention to feasibility assessments that should be performed prior to conducting these studies. We've seen in our work, for example, for past impact studies that assess the impact of risk minimization measures and their effectiveness, that a considerable number of such studies do not allow for a conclusion And the underlying reasons that we have assessed for those are related to aspects of study design and conduct, which could potentially have been envisioned if a thorough feasibility study could have been conducted in advance. And this is just an example from a real-world data use context where we feel confident and comfortable with using real-world data or non-interventional studies for decision-making. So I think insights into or critical thinking about these potential limitations for studies in advance might help in designing strategies to overcome those, as well as in making decisions on if such studies should be conducted at all, as conducting studies that are unable to answer the research question is a waste of resources, both for those that conduct those as well as regulators that need to assess such studies. Further, I welcome the advice on using target trial emulation approaches for studies with causal objectives, as it forces those designing these studies to explicitly define the research question, population intervention, and outcomes in attempting to mimic the clinical trial, and helps us in approximating the causal effects more accurately. But more importantly, I think the structured approach that accompanies the target trial emulation setup forces a more explicit documentation of each step and assumptions. And as a result, it enhances transparency and facilitates better replicability of such studies. So again, going to building trust in the results originating from such studies. And as the academic perspective also includes educational aspects, I think the guide also plays an important role for us as educators. It sets standards to how we should educate future professionals, those that will design these studies and use them, such as healthcare professionals or future regulators. So for us, we need to critically look at our curricula when it concerns on teaching and how we can actually implement educating on target trial emulation and, for example, the estimate framework within our academic programs. And lastly, I just want to use this opportunity to put on my NSEP hat and say that as the current co-chair of NSEP, I'm very proud and pleased to see the reference to the different NSEP tools and guidances, such as the NSEP methodological guide, the checklist for study protocols, and the code of conduct, explicitly included in the reflection paper. These guidances and tools are results of continuous work of the European research community towards attaining higher standards in conduct of such studies. So I think it stimulates us as a community to keep on the good work. And so from an academic perspective and with an ounce of NSEP view, I think I highly welcome the publication of this paper and look forward to see these recommendations develop in the future as we start implementing them in practice. Thank you.
Thank you, Helga. You quite right highlighted how the components of the reflection paper are intended to build trust on the real world data, and also how practice shows how challenging it can be to move from this theory to effectively develop a study that will be able to be used for regulatory decision making. Also, you highlighted the importance of or the interest on the studies that emulate the clinical trial setting for their practicity, and the definitively important role of education. So thank you very much for this. And now I'm going to move to our next panelist, who is the patient representative, Bettina Reel, who is the founder of the Melanoma Patient Network Europe and who was member of the first EU Cancer Mission Board. She is a physician by training and has a PhD in biomedical sciences, and she became a patient advocate after losing her husband to cancer. Bettina's work focuses on patient-centric innovation and covers a full spectrum from basic research over successful translational and clinical trials to timely and equitable implementation. Most recently, her particular interests have now been how to best leverage the potential of personalized medicines for patients and society through novel forms of collaboration and the support of health policy and governance. So Bettina, welcome, and the floor is yours.
Thank you so much for that. So as a patient advocate, I'm mainly looking at frameworks like this with under three headlines. The first one is the fastest possible way to generate knowledge and insight and find solutions for patients. As you have heard, my husband died at the age of 37, actually, of melanoma in a very short amount of time. So we as a family have experienced why we need novel therapies and access to novel therapies. I'm particularly interested then as a second point, how the way we conduct research directly affects the people subjected to the research. Usually we are mostly interested about safety concerns and whether patients understand the informed consent, but there's way more to it. We have been advocating that clinical trials are part of treatment, especially in advanced cancer, and as that have been considered under this perspective as well. So as a patient, if you end up on a clinical trial or randomized to standard of care, that defeats the purpose of participating in a trial. And I've been active in that space for a long time. And then as a third point is, whatever we choose as methodology, it directly affects patients. So I've been interested in how the choice of methodology affects our community and how we can come up with methodology that is better for the people we subject it to. Before I go through the framework and some points, I would like to highlight that at the beginning of this year, my network has formulated a patient consensus on data, AI, and data-dependent business models. And actually, the first consensus statement was that data use is an ethical imperative as people are dying and we have data that could save them. So with that, I'm very grateful for the opportunity to be here. And I was involved already with Get Real. So when we started with IMI, like real-world evidence a long time ago, and I'm delighted to see how much the discussion has actually moved forward and how we move from problems, and this is complicated, to something much more concrete. So I thank you for the opportunity. So I have 5 points that I would like to quickly go over when it comes to the framework. I would have expected to see a bit of sharper formulation on what the purpose of the exercise is. I think the potential in all this work is that it helps us address the issue of limited external validity of RCTs. We like to believe that RCTs are the end of it all, but we just had ASCO, the clinical community is discussing for years how we address the underrepresentation of women, of the underrepresentation of ethical minorities, about sociodemographics in our trials. We miss the elderly, we miss the multimorbids. That has been known for a long time. So now we have the opportunity to address some of these shortcomings, and I think it would be nice to see a bit more on the positive side of that. There is something about bias. Bias is, of course, always a problem. I would have liked to see a highlight that also RCT have bias, and now we have the issues that we have different types of biases that then create another issue. So that bias there will always be bias, so how we deal with it and how we compare different types of biases and relate them to each other. So I would like to have seen a bit more there. Something that as patient community we've been concerned about is, and that is missing in here, what is called analytical bias. I'm sure you have all seen the latest paper on red meat consumption, where depending on how you choose the parameters, red meat consumption, unprocessed red meat, is either good or bad for you. And this space is quite large. And that is something that concerns us as patient community, because dependent on the person who conducts the analysis, you might go one or the other way. So methodology, how to cope with this space, is, I think, much needed. My PhD is not in epidemiology, so this is not my background, but I do believe that we should focus on this space to ensure that what we analyze is actually trustworthy, and that then hopefully, it should be reported into so under the methods, there's a section in the framework, so I think that the parameters and the choice of the parameters has to be justified and needs to be reported, and that then will contribute to trustworthiness and trust at societal level into our methods. Then generally, I believe that our methods its form follows function. Our methods should follow a purpose. We've heard very early on that this is a framework that is providing principles for regulatory decision-making. Actually, Patrice said earlier on, evidence generation is planned and guided by data, knowledge, and expertise. I would like to have a guided by purpose added to that. We should inform our methods should always be under a heading of what we're trying to achieve and how this is supposed to benefit patients and societies at large. We should not be, and we've heard this also from Helga, we should not be conducting experiments because we can. There has to be a purpose and a motivation between them. So some of the issues I always find, it always makes me smile. So I have a background as a physician. When we read this, for example, treatment switching is a problem. So who has the problem? I mean, treatment switching is clinical reality. If our studies are unable to capture it, it is the methodology that has the problem. Someone said earlier on, we all will be patients one day. We want treatment switching. So if our methods cannot capture reality, we have to focus on fixing our methods and not complain about reality. Heterogeneity is another one. I mean, we heard about the artifacts. Sometimes for us, from a clinician perspective, heterogeneity is informative because it can give us a hint, a clue to improve something. And don't want to miss the opportunities to learn to improve our systems. And that is actually shown the linking to the last point, data quality. It's a recurrent topic. We've been discussing this for years, incomplete data, errors. It comes over and over again. And I fully understand why this framework is limited in excluding certain parameters. But I believe these topics, and actually this is something too that I believe is very, very important. We have to consider how we get the data and who provides us the data. I have conducted a workshop earlier this year for a European project funded under the EU Cancer Mission on a choice of a quality of life tool for a clinical trial. And it turned out that everyone wants something different from this poor tool. A clinician wants something different than a patient, than a regulator, than an HTA, than a payer. And we all try to squeeze it into one. If we are not able to deliver value to those who generate the data, the patients who collect it, the clinicians, we should not be surprised. We cannot formulate or come to a solution that our data quality improves. And this is larger than this framework, but I would urge the community to formulate then policy recommendations, for example. If you find certain artifacts always trip you up, if this is not visible, formulated as a policy recommendation, how are we as society supposed to address them if we don't know? So these issues come in discussions, but become not visible. I would invite you to make them visible, that someone can take them up and run with it. And last but not least, and this is more a technical issue, in our patient consensus, something that concerned people was their safety. Data linkage is managed here. Data linkage always comes with the issue that there are unintended consequences. And I'm sure no one who conducts these studies has nefarious intent, but we have seen this happening. So what was very strong in our discussion is that what we have to do is that we have, when it comes, for example, to data linkage, we have to have a risk assessment in place. We should monitor what's happening, and we need mitigation strategies in place that if unintended consequences occur, we can protect the people. And that is not just the data subject concerned. It can also, for example, be an ethical minority. It can be a subgroup. It can have large detrimental effects. If you think about it, if we do a genetic analysis and you all of a sudden find that there are certain associations, and can then think what happens for us as societies. So I think that's something we have to be careful about how we conduct it and how we communicate that. But overall, I'm very happy. I'm seeing how this is moving forward, and I personally have high expectations for the EHDS that I think we in Europe can be proud of, and something to look forward to and exploit for decision-making at regulatory level, but also in other places. Thank you.
Yeah. Thanks very much for this really valuable input. As we did with the previous speakers, I'm kind of refraining from totally summarizing, because I think you've made them quite adequately. And I think what I certainly take away is the focus on the purpose and the actual value it adds for patients in the end. And I think you've highlighted quite important aspects that are part of that general direction that you're giving to reflect upon this paper. Then we move to online. And it's my pleasure to introduce Holger Schoenemann, who is currently, I think, particularly leading quite an extensive initiatives around the GRADE network that I think most on this meeting are quite aware of, but has a long-standing career which ranges from starting in medicine, but that's actually adding to that a strong methodological component in epidemiology and biostatistics, and has been leading, amongst others, the Department of Epidemiology at the National Cancer Center in Rome, and has been chair of the Epidemiology and Statistics group at McMaster's as a full-time professor, And if I very briefly summarize, I think you have a very impressive academic and research contribution to the entire field of evidence generation over your career. So it's a pleasure to hear your reflections on this paper.
Yeah, thank you so much. Thank you also for the introduction, and thank you for accommodating me online. Unfortunately, I'm not privileged enough to be there in person. And I wanted to start by saying that I really welcome this paper and the clarification that it does provide. I appreciate, and I think this is very much in line with what the great working group's philosophy is. I really appreciate the role of causal inference in the methodology paper, and that really, if I do understand everything correctly, we are really interested in considering all data. I also want to follow up on what Dr. Will just said, but it must be done in a structured way. I think the section on statistical analysis and hypothesis testing is extremely important. I appreciated the comments on the magnitude of the data sets that we are dealing with, and that small relative risks can become statistically significant, and that the focus should be on clinical relevance. And I want to also address to some degree the issue that was just raised, which I also came across, and that is that the comment by Dr. Will that if we cannot, for instance, treatment switching, if the methods are currently not sufficient to address what people want, then we need to address that in our methods research. And of course, this goes into study design, but I want to make a case for addressing these issues in evidence synthesis. So, for instance, treatment switching is very hard to address in studies that have single brief interventions. Loss to follow-up is very difficult to address, and of course we can do that. But if that is not possible, then we need to do that through assessing the certainty that we have in evidence where things cannot be, or methods cannot be adjusted appropriately. I want to make one very particular point, and that relates to the ways forward. So this paper is great, but what are the ways forward? I think, it is very clear that from GRADE's point of view, most decisions are based on a combination of randomized trials and non-randomized studies of intervention. So it's very clear that we need to find frameworks for how to integrate them. And I'm only talking about interventions here, not anything else. Most decisions are based on bodies of evidence from both. What I think the methods development in the future should be looking at is frameworks and structured approaches to thinking about how non-randomized studies that may come from the real-world evidence space can be used as complementary, sequential, or replacement evidence. That relates to when non-randomized studies from real-world evidence generation, for instance, simply are of higher certainty or leave us with higher certainty in effect estimates than others. The other big issue that we have not addressed, I think, sufficiently is the issue of congruency between non-randomized and randomized studies. Congruency in the effect estimates is what I am referring to. Current congruency frameworks typically rely on testing for relative effects of interventions, and that is probably not the best approach for doing this. So the work that we are leading in the GRADE context, and I think this would be very complementary, is to look at what defines congruency between randomized trials or causal studies and real-world evidence. And in particular, this would focus on defining thresholds. And the reason why this is so relevant is, it was just mentioned whether or not studies are applicable to women. Well, in order to say that we can use non-randomized studies or real-world evidence to infer that the results from randomized trials that provide maybe higher certainty at a given point in time are can be extrapolated, we need to define what similarity in effect estimates is, and what we are suggesting that this is being done on predefined thresholds, where we bring in patient values, people's values, to agree on what, similar to sample size calculations, what small, moderate, and large effects are, and define these congruency thresholds in order to then make confident extrapolation from groups that are, for instance, investigated in randomized or narrower clinical trials to those that are outside of the trials, with predefined thresholds that would say results may not be incongruent. For instance, if results from randomized trials suggest a small effect, and all of a sudden we see very large effects in non-randomized studies. So those type of additions to frameworks, such as the one that is presented here, I think will help us in the future to address, in particular, regulatory extrapolation questions, that is, generalizability issues from one population to another. I stop here, and I just wanted to congratulate the authors on this paper, and I'm looking forward to next steps.
Yes, thank you very much for your perspective, which as far as I see it, actually adds the component of going beyond an individual non-interventional study or an individual randomized trial, but have a proper considerations which are relevant to assess the totality of evidence. And I think there is definitely an important challenge for us ahead to capture that part of the problem, because I'm quite aware that substantial number of guidances or reflection papers we provide are actually at an individual study level. And I think this is a component I think we definitely need to tackle.
Yes. Yeah. Thank you.
Yeah, thanks for the perspectives of all panelists. Then we move to the discussion, and I'm very happy that actually we have a knowledgeable moderator for that discussion. Professor Olaf Klungel will moderate our discussion, and he is a professor of pharmacoepidemiologic methods and head of the division of pharmacoepidemiology and clinical pharmacology at the University of Utrecht, and adjunct professor of pharmacoepidemiology at the Southern Denmark, University of Southern Denmark in Odense, and he's both pharmacist as well as epidemiologist, and he has a very strong record, I think, in research in particularly the area of real-world data and efficacy, and particularly also safety for drugs. And quite a few of the references that were here, like NSAP and previous European projects that have contributed to the field, actually Olaf has been quite important to make that move in the direction we are now. So I'm happy to give the floor to Olaf to moderate the discussion and pick up from the panelists and the floor.
All right, thank you, Kit, for this introduction. And well, first let me say that I'm very happy with the responses and feedback from the panelists, and of course also let's say the positive note on welcoming this reflection paper, but also identifying some areas for further clarification, potentially some gaps, and I think important to reiterate that if you can also provide this in the public consultation in writing, then we can also document actually these feedbacks and appropriately respond to that. But for this session, I think it's important that we also directly address some of your feedbacks and questions. And yeah, let's see how to do this in a structured way. I picked up some items that I think would be nice to that most of you have had addressed. And one of the topics was, I think, on fitness for purpose and feasibility studies. So in the reflection paper, there is some description on that it is important, but we do not go into much detail. We also refer to the guidance on registry-based studies, where there's more detailed guidance on this, but there's also different frameworks available. But maybe let's, if Xavier's voice is recovered a bit, would you mind addressing this particular topic of fitness for purpose, and why we have, let's say, briefly talked about feasibility, but what about additional guidance development in this area? Should it be part of the reflection paper, or should we build on previous frameworks?
Yes, thank you. Well, there are two aspects here in the questions, and first of all is the evaluation of the data quality as a generic name, which I said would, and in the reflection paper, is as two components, the reliability and relevance. Now, in terms of, these are important aspects to consider, and relevant could be also, and both could be actually related to fitness for purpose. In relation to the feasibility, I would say that the feasibility assessment is a more formal way of presenting the assessment or the evaluation that is made about these two aspects, reliability and relevance. I'm not sure about, what is the difference in terms of, the name of the terms and the semantics of fitness for purpose or relevance. So there is some, I don't think that there is any strict terminology about it. That's why we are using the terms that FDA is using, reliability and relevance. And the way they may be presented, the result may be presented is in the feasibility assessment in order to be able to evaluate whether the proposal of the data source and the real-world data is fit for purpose for the research question.
Right. Thanks. Yes, and maybe to add to that, there's what was mentioned also is that there's the data quality framework, but currently also a deep dive chapter into data quality framework for real-world data is in development and will also be at some point published for consultation. So there is also the opportunity to let's say further detail and so not in this particular reflection paper, but that will be another piece that we can refer to. Maybe also on scope, I think Dr. Spooner had a question about that, or raised, let's say, what the scope is, and also about non-interventional studies, that it is a definition by exclusion, and whether we in this reflection paper would let's say apply the guidance is applicable to a subset of non-interventional studies. I don't think it is, but maybe Xavier, you want to also address that particular question?
Yeah. The first point to say is that we are in a regulatory world and regulatory context, so we have a legal definition of non-interventional studies, and this has to apply and to be used. Having an alternate definition of a non-interventional study would not be appropriate in this regulatory context. Now, whether the guidance applies to a subset of them, yes, of course, because it applies to non-interventional study using real-world data for real-world evidence for regulatory purposes. As I said, all the terms are important here. So, now, in terms of specific aspects, so this may be in terms of objectives or methods, but we do not address the methods. We say descriptive studies with descriptive objective and studies with causal objectives. I think they cover all types of those studies in terms of objectives. So I don't think that aside, apart from what we have said on the scope, it has limitations for the methods applied.
Right. So no, I agree. So it's not very much a restriction in that sense, and also the example given on let's say epidemiology of disease studies, that would fall under the heading of descriptive studies. So that would be definitely applicable. Okay. So there were just one thing I picked up also was, let's say, the situation of the real world and where anything can happen, like switching, stopping drugs, etc., and that we may describe it a bit as a nuisance or challenge for the methodology, but in fact it can be very informative, and we should, of course, try to find and develop methodologies and apply methodologies to study that, because that can be very important for patients as well. So that's well taken, I think. But given that it is challenging, it's also difficult at this stage probably to have clear guidance on that. So it's also more of an area for further development of methodology probably. But methodology that is available and that is established, I think, should be cited there. I think the last panelist also, I think, very nicely indicated the importance of ways to integrate evidence from different sources, so non-interventional studies and trials, and for which questions the evidence is being generated. And in terms of trust, I think a last point, maybe also, I don't know if panelists want to respond to that. Do you believe that the reflection paper and the guidance that's in there will help actually to gain trust, build trust, improve trust in non-interventional studies, and maybe not in general, but for specific types of questions, of course? Is there any of the panelists that would like to give some comments to that? Yeah.
I do believe that any effort to lay open the way we proceed and how we take decisions increases trust. It's not just the method, but also how the decision was then taken based on the information that was available, because there is always some type of preference or some value judgment in there. And actually linking back to, and I thought there was a bit missing what real-world evidence could give us, is also acceptability in our societies, not just by patients. I don't know whether it's me now. Sorry. Whether it is, for example, if we see utilization long-term. So first, for a medicine or a treatment to work, it has to reach patients. For that, it has to be used and it has to be taken. And we have seen in our own community that preferences change over time. And I would to see something that is found also in the data, that patients think about medicines differently, physicians think about them differently, and that then affects utilization. So I think that type of preference is valuable. And that should be find reflection somewhere in the decision making. And I do believe that then increases trust, because people can just track what others have done. The worst is a decision without any explanation. So that's why this is so valuable.
Any other comments from panelists or?
I think it also links to the concept of, shared learnings across the research ecosystem. So, being able to understand the role that real-world evidence has contributed to decision-making, I think that really links in with what you're saying.
All right, thank you. I'm looking at the chairs because being conscious of time. Is there an opportunity to ask the audience also to comment or ask questions to either the panelist or the presenter?
Two quick reflections. If we are strict on the schedule, we are kind of on top, but I would open the opportunity for also the floor to come up with questions, responses, and reflections.
Okay.
And the floor is verbal here. We have a bit of difficulties of seeing the chat, but the chat is also the floor.
Okay. Right, so from the audience. Yes, please.
Okay, Denis Lacombe. Just a quick question for data quality. I was wondering, maybe I missed it, if you made a dichotomy, a difference between data that would be collected retrospectively versus data that would be acquired prospectively, and if it would make sense to address that.
So we haven't made any distinction about that. Real-world data can be data collected for primary purposes or data reuse because they were collected for a different purpose. So data quality frameworks are applicable to both. There is no distinction.
Thank you. First there, yeah. Okay.
Good morning, Rima Isam. I wanted just to go back to the fitness for purpose comment. I think there is variation in how people interpret that concept, as you rightly said. Some definitions, for example, in the US in the Duke Margolis paper, doesn't focus on just data fitness for purpose, but also of methods fitness for purpose. I was wondering whether that's a consideration that's something that you considered.
Well, we have not, as I said, we have not used the term fitness for purpose, but we have discussed in terms of, for the feasibility assessment, indeed the feasibility of the proposed study design in the data source that is considered. So this is a matter of methods, and are the methods usable in the data source, and what is the feasibility of this? And there may be different options in terms of study design.
All right, I saw a question over here.
Yeah. So, hello, I'm Andrei Borgrescu from the EMA. First of all, thank you, Xavier, for presenting the reflection paper and all for sharing your views. I've taken a lot of notes. And I have one question which regards one of the points that you don't spend a lot of text in the reflection paper, and you say that it's usually treated a lot in the literature and discussed a lot, and that is the confounding. And you say that is one of the few things in the reflection paper about it is that it is important to identify the set of adjustment variables beforehand, I guess, at the study design. And I was wondering if this is compatible, or what is the opinion around using more data-driven selection for confounding confounding adjustment variables, like high dimensional propensity scores, for example? Or is this rather discouraged, or should it be part of our tools of methods?
Good question, Andrei. So I think the reflection paper is, if you want, method agnostic with respect to how you control for confounding. The encouragement, of course, is to do that or think about that at the study design stage, because that will obviously help you a lot to mitigate the risk of confounding at the end. But again, if someone wants to propose to do that through propensity score, it's a method that is available, and I think it will be, if proposed, it will be assessed in due course by regulators, and whether it's admissible or not, or acceptable or not, it will be part of the assessment.
Thank you. All right, so I understand that basically anything is acceptable in principle, and it's a matter of discussion and case-by-case approach rather than...
Anything is susceptible of being submitted. Whether it's acceptable or not is another thing. And as I said, that's part of the outcome of the assessment.
Thanks. Yeah.
Jesper Kjær, Novo Nordisk. Xavier, thank you for a wonderful presentation. What a lovely work you've done with the reflection paper. It's very clear. I think some of the things you said earlier in terms of making sure that there needs to be a reach out by industry to the regulators in the early consultation about when this is relevant to use real-world evidence versus an RCT. I think I'd just like to, based on Bettina's comments on the purpose, suggest that there could also be an opportunity to give further clarity of the principles that colleagues in EMA and HMA, obviously, that are rational, that are driven by science and evidence, obviously getting to the point that the reasons behind why something is acceptable and something is not, based out of a purpose, maybe this really could lend itself into a public-private partnership activity to define what that looks like. So really bring regulators, industry, and patients together and define some of those principles that I think otherwise could end up being a guessing game by the industry, or when this is actually within frame or not frame. I think the paper you have developed lays out some very, very nice dimensions of where to think it's acceptable or not acceptable. So thank you for that clarity. But there's room for more work, I would argue.
Thank you. That's an important comment.
can we finish with this
Yes,
nice comment?
of course. So thanks everyone for contributing to this discussion, and back to the chairs.
Well, so thank you very much. Hopefully, in the next sessions, there will be a more in-depth diving on some of the issues that have been discussed now. So there will be more opportunities to continue with this interesting discussion. We're 7 minutes late, but I would stick to the starting of the next session. So we have a coffee break now. Thank you, everybody, for your contributions, for your work, for your questions, and we will be back here in 17 minutes. Thank you very much.
So, welcome back. I hope you've all had the opportunity to get a good cup of coffee. I hope also all of you in the virtual world also have had the possibility to come back to their seat. Thank you to the former presenters and also moderators and the panel from the previous session. We'll move on in the program now and go to session 2A, where we'll be looking into the real-world methods to support the EU regulatory decision-making. I'm happy to give the floor to Harald Ensmann, who's the chair of the CHMP, and also to Daniel Morales of the EMA.
Yeah, thank you, and now from my side as well. Welcome back, everyone. So, session 2, regulatory decision making. So, that's where the rubber hits the road. Let's be frank. We do have quite various different perspectives here. And yes, the most important is that of the patients. But we will have somewhat different perspective possibly from physicians, from payers and the HTA bodies who support them. And since we are talking here not only about all kind of treatment, but we are talking here about medicines, there may be somewhat different perspective from industry, from marketing authorization holders as well. And of course, regulators might have, again, a somewhat different perspective here. And let's be honest, this different perspective may in some way modify a bit how we see the purpose of what we're doing. I think we should be open to that and say that is not necessarily always exactly identical. But let me face on that what we have in common. I think all of us, no matter where we come from, will agree there's much data, there's much material out there, and we want to use that. But from the regulator side, and again, that is something why the second session is probably the important one, if I'm afraid. From the regulatory side, there still is a frequent, there's much out there. Yeah, we want to use it. Yes, but. So, and that means what we need to achieve here, building on the common purpose we have, is to find the role, to find the position of those real-world evidence, to find acceptance for this real-world evidence and to see where it fits in. Yeah. With that, I guess we can already jump right into it. And it is my pleasure to announce first speaker, Xavier Garcia de Albeniz is certainly perfect to open here and to create the groundwork for what we go. As a director of epidemiology as RTI Health Solutions, and if you look at his bio sketch, causal is the key word here. Without further ado from my side, over to the first presentation.
Thank you very much, Harald, and thank you for the invitation. It's a pleasure and an honor to be here to give an introduction to regulatory grade causal inference. I want to start with some terminology because I think the word estimand is going to be used in later sessions. In causal inference, we call a causal is estimand to our research question, as simple as that. Then we have the statistical estimand, which is the representation
of that causal estimate as a function of the variables that we have in our database. And the causal estimate and the statistical estimate are joined by the identifying assumptions. And then we have the process of estimation, which is where we crunch the data. We use our estimators to obtain a number that represents our effect of interest. And in that process of estimation, we need to be explicit about the modeling assumptions that we want to use. So the first step, which is defining the causal estimate, i.e. our research question, is the most important one, and that's the purpose of target trial emulation. Target trial emulation is a tool to articulate our causal question. It's not new. And the foundation is that for every research question, we should be able to come up with a hypothetical target trial that would answer that research question. The target trial emulation framework has two steps. The first step is specifying the target trial, our research question. The second step is emulating each component of that target trial. The motto of target trial emulation is don't do with observational data anything you would not do in a trial. Xavier Kurtz already presented an example of this table, which is the main tool of target trial emulation. In the column on the middle, we have to specify each of the components of our hypothetical trial, the eligibility criteria, treatment strategies, etc. And then on the right, we have to explain how we are going to emulate each of those components. In this table, the causal estimate is defined by those components, from eligibility criteria to the causal contrast. That defines the causal estimate. I'm going to divide my presentation in two sections. The first section will be about how to use target trial emulation to address a problem present in real-world data analysis, but not in randomized clinical trials, which is the alignment of time zero, exposure assignment, and the start of the follow-up. And the second part of my presentation will be how to use target trial emulation to address a problem that is present both in real-world data analysis and in randomized clinical trial, which is estimating the effect under complete adherence to treatment strategies. For the first part, the first thing we need to do is defining time zero. Time zero is the point in time when we start counting the outcomes. And it's very important to define time zero and align it with the application of eligibility criteria and exposure assignment to avoid different types of selection biases. This is never a problem in randomized clinical trials, because in randomized clinical trials, we have randomization, and that's naturally the start of the follow-up, and that was where we apply the eligibility criteria. But in real-world data analysis, this might be more complicated. And this complexity is illustrated by these embarrassing examples for us epidemiologists. There are some examples where we had observational studies in conflict with randomized clinical trials that then they were harmonized when a target trial emulation aligned time zero eligibility and exposure assignment. These are examples that are nowadays being taught in every 101 in every EPI 101 course, and highlights the importance of aligning these three components. In some situations, identifying time zero is very easy. For example, when we compare two active treatments, time zero is very easy to identify. If we compare vaccine A versus vaccine B, time zero is the time of vaccination. If we compare the initiation of radium-223 versus the standard of care in patients with prostate cancer, the time zero is the time of therapy initiation. That's very clear here. You might be wondering why I use this specific example, and it's because this was a real, an actual post-authorization study required by this institution that we perform in RTI, and that I'm going to use as an example for other concepts. But there are other situations where defining time zero is more complex. For example, when we compare initiating a drug versus not taking the drug, those who don't take the drug might meet the eligibility criteria for the whole study period. So they can have a time zero for every day of the study period when they are eligible. So when should I assign them the time zero? Here I have two options. I have the option of choosing a single eligible time, the first time they become eligible, or a random time when they are eligible, or I can use all eligible times. As you can imagine, choosing all eligible times is methodologically more involved, but statistically more efficient. I will extract more information from my data. And target trial emulation framework gives you the tools to do this sequential emulation of target trials. The other very important point is classifying individuals into treatment strategies at time zero. Sometimes this is very easy. As I mentioned before, if we compare vaccine A versus vaccine B, time zero is the day of vaccination, and I will know that day in which group they are. But there are 3 situations where assigning people into treatment strategies is not that easy. For example, when we use a grace period, sometimes in the analysis of our observational data, we need to use a grace period, which means that we give patients a few weeks or a couple of months to start therapy. Because in real life, there can be some time from the time when we take the decision to start treatment to the actual time when people start treatment. When I used to practice, I remember prescribing chemotherapy to patients, and from the time you decide to prescribe the treatment until they actually get the treatment, they might need to obtain extra diagnostics or do some paperwork because the chemotherapy is very expensive. They need to find a slot in the hospital to receive treatment. During those 2 weeks, they can have complications and not being eligible for treatment anymore. You account for that situation of the real world by using grace period in your data, in your real world data analysis. And the other situation where assigning treatment at baseline is complicated is when you study duration of treatment. For example, if I want to compare receiving 2 years of aspirin versus receiving 5 years of aspirin, at baseline everybody started aspirin. So I don't know in which treatment group they fall. Because I don't know for how long they're going to take it. So at baseline, my treatment strategy is not well defined here. And the third situation where classifying patients into treatment strategies might not be well defined is when we study dynamic strategies of treatment. A dynamic strategy of treatment is when we need to wait for something to happen to prescribe treatment. For example, this was common in HIV research when we didn't know when to start antiretroviral therapy according to the CD4 count. At baseline, patients might not meet the criteria to start therapy. We have to wait until the CD4 count drops below a threshold to start therapy. So at baseline, I don't know in which group they will fall. Target trial emulation gives us the tools to address this uncertainty, and these tools are cloning or random assignment. So as a summary, when we analyze real-world data, we must align time zero, eligibility, treatment assignment, and we can be in the situation when both of them are well-defined, where one of them is not well-defined, or when both of them are hard to define. Target trial emulation gives us the tools to specify both time zero and treatment assignment for all these possible situations. So we avoid self-inflicted design biases in these studies. Okay, the second part of my talk is about a problem that is present both in real-world data analysis and randomized clinical trials, which is estimating the effect under complete adherence to treatment strategies. Why is this important? Because we have 2 options to define our causal contrast. We can estimate the effect under initiation of the treatment strategies or the effect under complete adherence to the treatment strategies. If we are doing a real-world data analysis where the outcome is a safety outcome, we don't really care about the effect of initiating the drug, because we might conclude that the drug is safe because there are not adverse events because people just didn't take the drug. We don't care about that. What we care is about what is the safety profile of receiving the drug under complete adherence. That would be the observational analog of the per protocol effect in a clinical trial. It's important to define the causal contrast because that will drive our statistical analysis. If we estimate the effect under initiation of the treatment strategies, we will only need to adjust for baseline confounding, assuming everybody is under follow-up. But if we want to estimate the effect of complete adherence to the drug or to the treatment strategy, we will need to adjust for baseline and for time-varying variables that can affect adherence. And you might wonder, adherence to what? Well, adherence to a treatment strategy. A treatment strategy, and I really welcome the point that Bettina did before. A treatment strategy is a set of rules to assign treatment at each time of the follow-up. In other words, we don't evaluate the effect of receiving a drug or not. We evaluate the effect of receiving a treatment strategy that is composed of receiving this drug now, but you can stop that drug when there is a clinical condition to stop that drug, and you can continue to other treatments, because that's what happens in real life. So treatment stop, treatment switch, treatment combination is not a problem here. Here we define what is the clinically relevant treatment strategy that we want to evaluate. So let me give you a couple of examples of treatment strategies. The first one is the most simple example that you can find. And this is an article we published in 2017, where we compare baseline screening colonoscopy versus no screening. As simple as that, point exposure. Nothing else needed here. The second example are the treatment strategies that we use for a post-authorization safety study required by this institution in 2018. Here we compare in the initiation of radium-223, that's the beginning of the strategy, and then we explain in which situations patients could stop the drug. And those situations were after receiving 6 cycles, or if they had toxicity, cancer progression, or a worsening of the overall health status. Patients could stop the drug in those situations, which are clinical indications to stop the drug. And then we explain what to do after they stop the drug. We said that they could continue receiving other systemic drugs for prostate cancer as clinically indicated. And we also said that they could never combine radium-223 with another drugs. We did a similar specification for the control group, and we also specified which were the concomitant treatments that were allowed in these treatment strategies. That in this case was androgen deprivation therapy with first-generation anti-androgens could be received at any time in both groups. So this is an example of a comprehensive treatment strategy that mapped what happened in real life. The next step is how we handle post-baseline events. Well, it depends. It depends on which type of post-baseline events. If this is a treatment change, the first thing that you have to ask yourself is, well, what is the causal contrast? If your causal contrast is the effect of initiating the drug, you don't care about treatment changes. But we said that we are interested in the effect under complete adherence. So if that is our causal contrast, the next thing that we have to ask ourselves is, what is the treatment strategy? Because you can stop the drug and still be adherent to your treatment strategy if you stop your drug, for example, for toxicity or because it's not working anymore. The second post-baseline event that we might need to handle are competing events. Competing events are events that preclude us to observe our outcome of interest. That is part of your research question, of your causal estimate. If you foresee competing events, you have to specify which is your effect of interest. You can choose to estimate the effect on the composite outcome, or the effect on the outcome through all causal pathways, that's called the total effect, or the effect on the outcome in the absence of competing events, that's the direct effect, or the effect on the outcome not mediated by its effect on the competing event, that's the separable direct effect, or the effect on the outcome only through its effect on the competing event, that's the separable indirect effect. You tell me in your causal estimate what is your effect of interest, and I will tell you which statistical method you need to use. And finally, you might have losses to follow up. That pertains to the statistical analysis to estimate the effect had everyone been under complete follow-up and the corresponding assumptions. So to finish, to inform patients, clinicians, policymakers, and regulators, we need to evaluate populations who are identifiable at the time of decision-making. We need to compare treatment strategies, not treatments. We need to estimate effects under complete adherence, especially for safety studies. We need to choose relevant causal estimates, research questions. We need to avoid self-inflicted design biases by aligning time zero, exposure assignment, and eligibility, and we have to outline the identifying assumptions. Target trial emulation framework does exactly that. Thank you.
Yeah, thank you. I do think we want to have enough time later for the discussion. Therefore, I will immediately push on. And after Xavier now here being a medical doctor, it is my pleasure to announce Juan José Avellan, a mathematician. Broad background, public offices, academia, pharmaceutical industry. And in his biosketch, I read specific interest in utilization, effectiveness, and safety of medicine. And of course, my pet peeve is, of course, effectiveness versus efficacy. So anyway, without further ado, Juan, over to you.
Thank you very much, Harald. Yeah, I'm going to talk about the use of estimates in target trial emulation. Both Xavier Kurtz and so the two Xaviers essentially have already alluded to this morning. Xavier Kurtz said that we have included, or is included in the reflection paper, and I will touch upon that. And Xavier Garcia de Liniz has made a very eloquent introduction to the framework. So this is the disclaimer as usual. So the specific text used in the real world, in the reflection paper of use of real world data and interventional studies to generate real world evidence says that it's recommended to use the target trial emulation framework to formalize the design and analysis of non-interventional studies using real world data with causal objectives. With studies with causal objectives, we have in mind the studies for comparative or safety purposes. And the reflection paper goes on to say that the estimate framework described in the ICH E9 addendum on estimates and sensitivity analysis in clinical trials should be considered in the design of the hypothetical trial. Now, people in the real world evidence realm may have wondered why, if we are used to use this PICOT framework and we have used it for 30 years, why do we now need to change to the estimates framework? And it's a fair question. And I'm going to try to explain why this recommendation was included. To start with, you need to think that the beauty or the appeal of the target trial emulation framework is that it makes explicit the target trial that you would like to run in order to address your research question. And if you need to specify the target trial, then you can consider, or you should consider the framework used to define research questions in clinical trials. And that is the estimate framework as of 5 years ago. And now the estimate is defined in that addendum as a precise description of the treatment effect posed by a given question attached to a clinical objective. And this is very important, precise description of the treatment effect. So it's all about adding clarity. Now, if you read the guideline, you realize that it's driven by causal inference principles. And I'm saying principles, although it specifically avoids terminology related to causal inference methods to prevent people to think that the only way to implement the principles outlined in the guideline is to use those methods. And I'm going to illustrate the fact that the guideline is driven by causal inference principles with the definition of treatment effect. Treatment effect is defined of how the outcome of treatment compares to what would have happened to the same subjects under alternative treatment, either if untreated or treated with a different medication. Now, the first time I read this, same subjects, what are we talking about here? Are we going to run crossover studies for all our confirmatory studies? That's not possible. But if you look at the book of Hernan and Robbins of causal inference, you see in the first figure of the book, this chart on the right, that essentially says, well, you have a population of interest and you're interested in how the outcome, certain health outcome would compare in those treated versus untreated. Then if you just select two subgroups, treat one group and not the other, and you compare health outcomes, you can establish association, but not causation. In order to use causation or to establish causation, you need to compare exactly the same subjects. Now, the little thing in blue at the bottom is mine, it's not in the book. And essentially, in randomized clinical trials, you do the association thing, but the randomization allows you to interpret the results in a causal way. So the addendum not only provided a framework to guide sponsors on how to define the estimate in a precise and unambiguous way, but also cover other topics like lack of logical connectivity between trial objectives, design, conduct, analysis, and interpretation of results, the definition of missing data, data that would be meaningful for the analysis of a given estimate, but were not collected. Sometimes they hear in the real-world evidence realm, well, you cannot reasonably expect that someone stays in a data source or whatever. So why should we consider missing data? It's not about expectation of data collection. It's about whether the data of those subjects will be meaningful for what you're trying to estimate or not. And to that end, it distinguishes between, for example, things like treatment discontinuation, which is an element to discuss at the estimate level, as Xavier Garcia-Albeniz said, versus the study withdrawal or loss to follow-up, which is an estimation issue because it generates missing data. It also clarifies the definition of sensitivity analysis as an analysis to address robustness of the results to departures from assumptions made in the primary analysis. And this is important, and I will touch upon that in a minute. So in a nutshell, these are the key elements of the addendum on estimates. So you need to consider 4 elements. First of all, the objective of your study. For each objective, you need to consider at least one estimate, and you need to define the estimates through its attributes, the population, the variable endpoint, the treatment conditions. I think that's the terminology used in the addendum. You could refer to it as treatment strategies, as Xavier said, the summary measure, and intercurrent events, plus the strategies to deal with those. Then, once you have defined your estimate, you need to think of analysis method. And you need to specify the method and make clear what are the assumptions behind that method. And finally, you need to choose sensitivity analysis to address the robustness of the results to variations in the assumptions in the primary analysis. Now, if you look at this, for example, in the real-world evidence setting, often people say, I'm going to change the definition of case for a sensitivity analysis. So according to this framework, that is not a sensitivity analysis. That is a different estimate. Now, as I said, estimates are driven by causal inference, and one can argue that PICO were also driven by causal inferences. And in fact, you can map probably the PICO elements to the estimate attributes, more or less. We can debate whether this mapping or some other mapping, but there is an element that is new in the estimates and was not present, or at least not explicitly present in the PICO framework, which are the current events and the strategies to handle them. And I'm going to try to illustrate why this element is important. But the main point to me is that the way I see this, the estimates framework published 5 years ago, I see it as an evolution of the PICO framework published 25 years ago. So, as I said, I'm going to illustrate why the intercurrent events and strategies are important with an example that I extracted from the training materials of the ICH E9 addendum. So in the past, one could find something like this in a study protocol of a randomized clinical trials. The objective of this trial is to investigate the efficacy and safety of dapagliflozin for the treatment of type 2 diabetes mellitus compared to some control. The primary endpoint is the change in glycated hemoglobin, so this HbA1c biomarker from baseline to week 24. The analysis method will be an ANCOVA. Now, you can identify the PICO elements in what is written here. The population, patients with type 2 diabetes, the intervention, dapagliflozin, control, the control treatment, outcome is changed from baseline in the biomarker of interest, and the time frame, week 24, after initiation of treatment. Now, during the assessment of the evidence submitted by the applicant wrote data collected. So some subjects started rescue medication at some point during the follow-up, and data collected after initiation was excluded or were excluded from the analysis. The regulator disagreed with this and essentially said, well, I have included a sensitivity analysis in which, although I acknowledge that after taking rescue medication, the data will be affected, I still consider this data relevant for the treatment effect of interest, and I'm going to include it in the analysis without any adjustment for that. So you look at the two treatment effects that they were pursuing with their strategies, with their analysis strategies. They are different. one was after the comparison of the two arms in the absence of rescue medication. The other, the regulator, was after the treatment effect, regardless of the use of rescue medication. Now, the PICOT elements were agreed by applicant and regulator, as stated in the objectives, analysis methods, etc. However, the two understood different things. And these two different things are two different treatment effects, which are appropriately thought of as different questions or different estimates, and not two different answers to the same question. In other words, presenting one analysis as a sensitivity analysis of the other would not be correct in this framework. So what was missing? The identification of an intercurrent event, in this case use of rescue medication, and a strategy to handle it. The applicant used what we call a hypothetical strategy if you want. The regulator wanted a treatment policy. Now this discussion should have happened upfront at the design stage and not at the analysis stage. Now this is the definition of intercurrent events, events that occur after initiation of treatment and affect either the interpretation or the existence of the measurements associated with the outcome. And we've mentioned rescue medication, but there are other examples of intercurrent events: treatment discontinuation, either due to lack of tolerability or lack of efficacy, treatment switches, death, and Bettina, treatment switch is not a problem. It just poses challenges with respect to the interpretation of the data. And the thing is that these elements have an observational nature, and because we cannot identify upfront which participants in a clinical trial are going to experience intercurrent events. The guideline actually proposes 5 different strategies to handle intercurrent events. Treatment policy means that we're interested in the treatment effect regardless of the occurrence of the intercurrent event. Hypothetical, where one is interested in the treatment effect in a hypothetical world where the intercurrent event would not occur, like, for example, in the absence of rescue medication. And in this strategy, if you use this strategy, the data afterwards may not be meaningful. A composite strategy where actually the occurrence of the intercurrent event informs already about the efficacy of the medicine. And you may say, well, if someone discontinues treatment, then they may be a non-responder already. While on treatment, a strategy where you're interested in the treatment effect while the subject has not experienced the intercurrent event, and the principal stratum will be the more challenging one because you're interested in the treatment effect in the stratum of subjects who would or would not experience the intercurrent event. And this is important, maybe you're interested in that subset of subjects who would not experience the intercurrent event in either of the assigned treatments. Now, I want to revisit the elements that were included in the original paper of the target trial emulation in the paper by Adnan Robbins, particularly the causal contrast of interest, ITT and PP, so intent to treat, and per protocol, and Xavier Garcia-Belniz alluded to this. So the estimate framework essentially diminished the role of the per protocol analysis and actually expanded the strategies for to implement the intent to treat principle. So it's not just these two options, in my opinion, that are relevant. It's not treatment after initiation versus treatment after full adherence. You have a gradient here in between. And as I said, you know, the important thing is to identify the intercurrent events and choose a strategy to handle those. And again, the per protocol, the ICH E9 states that it's very challenging to identify an estimate to which the per protocol analysis would align, because it essentially is a mixed bag of intercurrent events, all mixed together, all put together. And the idea is to isolate those, identify those, and choose a strategy for each of one. Here's an example, and I think in the interest of time, because Daniel Prieto will come after me, I will show an example. And I'm going just to conclude. So the PICO framework and the estimates framework were developed in the real world evidence and clinical trials worlds without essentially talking to each other. But actually there is a lot of common ground, and that common ground is causality. Both are in the pursuit of causal interpretations of the results. And that offers a bridge between the two worlds that we need to exploit. What we think is that the use of the estimates framework within the target trial emulation will make that bridge more robust. And we think also that considerations to other elements which are in the addendum on estimates and were included also in the reflection paper using real-world data in non-interventional studies would be beneficial, particularly that estimates and estimations are separate discussions, that changes in estimate attributes, like, for example, the definition of case, lead to different estimates and they are not sensitivity analysis. And the logic about set first your estimate, then choose your estimation method, and then consider sensitivity analysis to assess the robustness of the results to assumptions made in the estimation method. And I will conclude here. Thank you.
Thanks, Juan. Very clear, and thanks for keeping to the time. Again, we want to have time later for the discussion. That's why we immediately will move on, and our next speaker will be online, Dani Bretio Alhambra. And we are back now from the mathematician, back to a clinician trained in family and general practice, what is a nice contrast. And from his impressive list of academic titles, I just highlight, he is a professor of real-world evidence at the Erasmus Medical Center. And with that, no more words from my side. Dani, over to you. Welcome.
Thank you so much. Yeah, I'm just going to give you an example of a target trial emulation study we are running in the Darwin EU initiative. It's a vaccine effectiveness study, as you will see. And the research question we want to answer in this study, which is ongoing as we speak, is the effectiveness of HPV vaccination to prevent severe disease outcomes in women, including invasive cervical cancer and precancerous lesions like SIN2, SIN3, and the related surgery or intervention for those, which is conization. And we're looking at data from 3 European countries, Spain, the UK, and Norway. There's 2 sets of objectives. The main objective is to look at the vaccine effectiveness, as I said, against invasive cancer, SIN2, 3, and conization. And the secondary objective is to compare between brands of valencies and to compare also dose schedules, if data allows. The first pitfall in this design, of course, is that there is the potential that there is differential uptake of vaccination according to outcome risk. So women more likely to have or perceived to be more likely to have these outcomes could be more likely to take the vaccine, which would affect the baseline exchangeability assumption and cause confounding. And I will explain later how we deal with that. I'm going to go through this quickly, but basically we have the different components of the estimates framework and target trial emulation in our protocol in different sections. As Juan Xavier elegantly presented, there's a lot of common ground between both, and I will try and cover the most important elements of this in the next slides. So in terms of population, we were interested on women eligible for HPV vaccination. Things have changed over time a lot, but for this particular study, we focused solely on women eligible at any time after the launch of the HPV vaccine in the contributing data partners. They had to be 9 years older as per the approval of the vaccine. And the vaccination schedules in the 3 countries is very different and has changed over time, as you can see here. I'm not going to spend too much talking about this because this is all in the public domain, but just to let you know that both the UK and Catalonia or Spain have changed their guidelines over time, whilst Norway has stuck to the initial vaccination schedule. Okay, so continuing with the population, one initial decision we made to minimize confounding or maximize this baseline exchangeability was to restrict the population to those eligible for universal vaccination campaigns. So that means women eligible for vaccination program, universal program in each of the countries. So they had to be born on or after 1995-96, and in observation and alive in the database between the ages of 9 and 15 years old, which is when the vaccination happens. to further maximize exchangeability, in this case, conditional exchangeability based on the baseline characteristics of these participants, we matched on database, on GP practice or care site or region, and on year of birth. And then we further, in a second step, matched on propensity scores, which are, as you mostly know, conditional probability of vaccination based on the baseline characteristics of these participants. The treatments studied here were 3 brands of vaccines, as you saw earlier, and of course the unvaccinated group, which were only relevant for the primary aim. Start of follow-up or time 0 for the primary analysis, and I'm going to stick mostly to the primary analysis from now on. With the vaccine in the vaccination group is the time when they received the first dose of the vaccine before the age 15, and then in the unvaccinated, that would be the time when they're matched vaccinated pair received the vaccine, so they are aligned on the same date. We end follow-up at the time of death, lost to follow-up typically due to migration or end of study or outcome. In terms of endpoints or outcomes, we measured at 5 years, 10 years, and 15 years invasive cervical cancer, SIN2 or 3, or conization procedures. And maybe I'll highlight very quickly here that there's a potential, at least for some of these outcomes, that a differential uptake of screening in the vaccinated versus unvaccinated could cause also confounding over time or loss of exchangeability over time due to survival bias. The analysis is primarily per protocol, although we did an intention to treat as well. And basically, the unvaccinated population was censored if they received the vaccine. That is extremely rare that people would receive the vaccine if they haven't had it at the age of 15 in this particular cohort because of the vaccination programs. But if that happens, we would censor them. And the vaccinated are only censored if they receive another vaccine for the dose analysis, not in the primary aim. So intercurrent events in the unvaccinated, as I said, we censor when they are vaccinated, therefore dealing with with this intercurrent event with a hypothetical strategy. In the vaccinated, we basically don't do anything about dosing, incomplete dosing. So if people have had only 2 doses, that's what they get. In the analysis plan, we said that we would match, as I explained already, on year of birth, year of first dose, and geographic region or GP practice, primary care practice. And then we further match by nearest neighbors using propensity scores. In terms of analytical plan, we estimate incidence rate ratios at 5, 10, and 15 years, and cumulative rates and risk ratios. And we also run a time to event analysis using where we will report hazard ratios. We have 2 key diagnostics that we are going to use 3 maybe, to measure the potential impact of compounding. One is, and this is a figure from a previous paper, not from this study, which is still ongoing. This is to see whether there is any impact of propensity matching and population matching in terms of compounding. So we are expecting that the people unvaccinated and vaccinated will look more dissimilar before matching than after matching. We also have negative control outcomes that we will measure to see whether the matching has had an impact on reducing the risk of unrelated or not vaccine-related, not causally related to vaccination outcomes. And we have a few of those in the analysis, and we will hope to see that they are similarly balanced across vaccination status. And then we also have, and again, this is a plot from a previous paper on COVID vaccine effectiveness. We also hope to see that the testing over time, so smear testing in this case, is balanced across vaccination arms, as we do see in this study, where at the top you have unadjusted rates that are higher in one group than the other, but then after matching, we see very similar testing rates across the match groups. In conclusion, both the estimates framework and the target trial elimination frameworks are useful and complementary to specify causal analysis like this. The estimates framework, in our experience, is especially useful to focus on this and how we handle intercurrent events and how they can affect our estimates. The target trial elimination is particularly useful to define timing, time zero, as Xavier explained, and how we design the study. Our study design and analysis should improve conditional exchangeability at baseline and over time. And I believe that the use of diagnostics should be used to detect departures from causal inference assumptions. And I will finish there, hopefully on time for the next speaker. Thank you so much.
Thank you so much. Indeed, perfectly on time. Much appreciated. And therefore, we will immediately then continue. Rima Isam from Novartis will be our next speaker. And I highlight she has abundant experience on the other side of the Atlantic as well. So might broaden our perspective with that as well. Rima, over to you.
Thank you for the kind introduction. I'm Rima Isam, and I will be discussing the target trial and estimate framework in post-market safety study. I'll be illustrating the application with a hypothetical case study so it will be more like a sponsor experience, but I'm happy to bring in other experiences during the discussion. Next slide, please. Those are the typical disclaimers. So these are my take-home messages. First, the case study will illustrate how specifying the target causal estimate is critical in post-authorization safety study. Now, all the frameworks that we've heard before, the target trial emulation and the estimate framework, are tools to try to clarify our causal estimate. Why is this important? Once you have the target causal estimate that's stated clearly, it clarifies the purpose for all stakeholders. What was the knowledge gap? What is the causal discovery objective of the study? And then also, once that is clear, it clarifies the path forward for the sponsor for planning and interpretation. Because then the choice of data sources, the choice of study design, the choice of analysis, all of this have to align with this target causal estimate. So that's my main point. So we're a big fan of the reflection paper. I think it's setting the right rigor for planning those studies. Now, I will switch in the second part of my talk to illustrating some practical consideration that occurred in the hypothetical case study. That is that one has to consider the pace of information relative to market authorization of a new drug. That is, in the time frame that the study is conducted, the knowledge gap or the estimate of interest may evolve. That also some of the relevant information that can inform this knowledge gap may accrue beyond the post-market study. And then finally, that one has to consider very carefully when to lock in or when to pre-specify that estimate and also the estimation methods relative to that time frame. Next slide, please. So this is my hypothetical, thank you, the hypothetical case study. This is a drug, and it will be a biologic drug that is first approved to treat patients with a chronic indication, and then patients are expected to have repeated exposure. The pass request is stated here. And I will walk through some of the attributes that were discussed by the previous speaker of either the target trial emulation or the estimate framework to illustrate how this language already hits a lot of those attributes. So the request is asking for a comparison. Therefore, the objective is a causal objective. Population of interest is those that are eligible to receive NOVD, this new therapy. The treatment is NOVD, but the comparator is less clear, and I'll go back to that in the next few slides. The outcome is malignancy, and the time frame is 8 years and more. So what remains a little bit uncertain with this language is what is the comparator, when is the start of follow-up for both the treatment and also the comparator, and also what happens with events after the start of follow-up. How do we handle these intercurrent events in the analysis? There is also the summary measure is not described, but I will not go over that in the next few slides. I think that will deserve 15 minutes just by itself. Next, okay, yes. So just to continue with this hypothetical case study, I wanted to illustrate some milestones and timelines from when the marketing authorization happened to when the final analysis and reporting happened. So if I start at the marketing authorization of the drug, what happens immediately afterwards is that there is a selection of external partners. So it could be data holders or contract research organization that would start collecting data. With those partners and also obviously with the regulatory authorities, we will develop a protocol and a statistical analysis plan. After or while the statistical analysis plan is finalized, the first person is recruited until the last person is recruited. And then finally, 8 years beyond the last patient is recruited, we have the final analyses and reporting. Of course, the protocol and SAP development, when they're set, they're not set in stone when they're finished. There are maybe a few amendments that happened over that time frame. But typically, the amendments are very few because you always need to justify why you're changing your plan. Of course, you have to have a good reason for all the partners involved. So typically what happens is the objectives don't change, but maybe there may be secondary or tertiary objectives that are added. Similarly, the primary analysis rarely changes once it's decided upon, but rather there may be additional sensitivity analysis that are added. So yes, there could be amendments, but once it's decided, it's rare that you go back on some of those decisions. Now I put a timeline in the bottom, not this is all fictitious, but it's just to say that if you see a report today, most likely the request was made at least 14 years before in 2010, just to sort of give you an idea of how long the process can take. Because sometimes when you talk about real-world data, people imagine that the data is already there. But I wanted to illustrate that that's not always the case when you have to set the estimate. So let's get to my first point of why I think that using the target trial emulation or using the estimate framework can be very helpful in adding clarity to what is the question of interest. So here, and we can focus for now on target trial 1 because it's a little bit easier. What I'm sharing here is, a diagram that shows maybe the hypothetical randomization that would happen at index date when people either receive NOV-D or another therapy, and then the 8-year follow-up that has to happen afterwards. So in target trial 1, people didn't have any prior therapy, and then they initiated NOV-D. But the question is, what is the comparator? Is it all other therapies for the same indication for people who also would be eligible to take NOV-D? Or are we specifically interested in a comparison to other drugs of the same class, for example, as NOV-D? So there is a choice there, right, of what is the comparator group. The next question is also what happens before people initiate NOV-D. Now, in the target trial 1, it's naive users. They weren't using therapies of the same class as NOV-D, and they just initiated. That was their first therapy in that class, in that biologic class. Whereas in the target trial 2, they initiated NOV-D after they've had other drugs of the same class. So then they're coming in already, maybe potentially with different characteristics. So in this case, again, are we interested in only the naive users, or are we interested in also the experienced users? And again, the comparison both times, even if the comparator group is the same, are leading to different estimates and different evaluations of risk. So so the target trial framework can help us already assess these two questions. When is time zero? What is the comparator? And also, what are we comparing to what? So now I will go to the intercurrent event. But before that, I want you to notice here in my diagram that in my simplified diagram, once you've initiated one drug, you continued on the same drug for 8 years. That is very rare that it happens in practice. Most likely what happens is something like this. There is a very dynamic treatment landscape. Some of these indications are for symptomatic diseases. So people could maybe stop taking the drug and take holidays, while their symptoms are done, and then maybe restart if their symptoms come back. So there is a very dynamic treatment landscape. And then the question is, okay, so what is the comparison of interest? Are we really interested in what would have happened if people had continued on the drug that they have started? Or are we interested in characterizing the risk of malignancy from eligibility to 8 years, regardless of what the person would have received, and trying to attribute it maybe to the mixture? Or maybe we're interested in a dose response. Is the more you take a particular drug, the more likely you are to maybe experience a malignancy. So as illustrated here, whether you use the target trial framework or the estimate framework, you don't have an answer to these questions, but at least it helps you have a dialogue on which question is really of interest for regulators, for patients, generally for public health. So the second point is, of course, once you did all this hard work of defining or clarifying what the target causal estimate is, then everything else follows because it has to align with this very big decisions that you've made. This will guide which data source you're going to need is going to be relevant and reliable for answering these questions. How can you design a study, whether you're doing primary data collection or whether you're doing evaluation of algorithms in real-world databases? How can you design a study to better emulate this target trial? How do you assess power and feasibility of a particular study design? What are the sources of bias that can come? Again, bias is relative to the target causal estimate. And then also, how can you design analytical methods to eliminate, minimize, and mitigate sources of bias? So I'll switch gears now and talk about the practical considerations, and I'll go back to my timeline here. So in addition to whatever is happening in the past study, there is external information that becomes available. So that includes information from extension study for the same indication for people who are exposed to NAFD. There could be also randomized clinical trials for NAFD in other related indications. So maybe there is an indication expansion. There is also more information available about real-world utilization. Who uses the drug? How long do they use the drug? And if they're switching, what are they switching to? There could be some changes in clinical practice or especially competitive landscape of the drug. So how does that impact this beautiful application of this beautiful framework? Well, I think one has to be realistic. There is sometimes a high knowledge gap that motivated the market authorization. But that knowledge gap in 2010 may look different than the knowledge gap in 2024, regardless of what happened with the post-market authorization study. So then, would it make sense to sort of, at the outset, not only specify these causal estimate, that is the target, but also maybe other estimates that could inform the knowledge gap as well. The other, and those are related comments, obviously. It all has to do with the sort of the type of information that that's available. So even if the estimate doesn't change, if the estimate is still the same, there may be more information than the post-market authorization study to answer that question, to fill that knowledge gap. So then how can we integrate more formally that information as the time goes on? And then finally, one has to think about pre-specification of the analysis plan, right? Of course, we can all agree that if we wait until, you have to write the final pass report to finalize your statistical analysis plan, that's too late. There is potential investigator bias, and everybody wants to build trust in the findings, so that is too late for everyone. So, but I think also that trying to pre-specify, especially the analysis plan and sometimes even the estimate too early, right after market authorization when the knowledge gap is very large, it is really also counterproductive. What may be just right is to wait until there is a little bit more descriptive estimates available, especially about drug utilization practice, before you lock in a causal estimate. So, of course, this is what I would say for this hypothetical NAVD, but what is just right for other applications and other questions will have to depend on a case-by-case basis. But this is maybe to be thought-provoking. So here are again my take-home messages. I hope I conveyed that I'm a big fan of the target trial emulation and also of the estimate framework, because I think they are tools that are very helpful in clarifying the knowledge gap and the causal discovery objective, and they do help sponsors and everyone in planning and interpretation. There are some practical consideration when using these framework, and that is the pace, that has to do all with the pace of information. So thank you very much for your attention, and I'm happy to take any questions.
Thanks, Rima. And that concludes the presentations. And I'm very happy that obviously we are on the right track to make things happen here. We are clearly getting beyond the new theoretical where we want to go. We are on the way. And we have now indeed enough time for the discussions. And with that, I'm happy to hand over to Daniel, who will structure and guide us through this. Daniel.
Great, thank you very much. And again, thank you very much for the presenters that came before us. So we have 20 minutes for a discussion. We have three panelists, which I'll introduce just now, and they'll briefly pass on their initial perspectives and reflections, and obviously discussion section's an excellent place for that. And then we'll have an opportunity to ask questions, and we'll also allow obviously the presenters from earlier on to participate in that, so please do jump in as well from that perspective. So I think for the first panelist, I'd like to introduce Helene Nordahl, who's a Senior Director at Novo Nordisk, leading a global regulatory affairs team into integrating emerging technologies and methodologies to link clinical trial and real-world evidence. So it'd be great to hear your views on this as well.
Thank you so much, Daniel. And thank you so much for excellent presentations, for wonderful presentations that actually demonstrate and summarizes the universe of target trial emulation. And I really appreciate the deep dives into the particular considerations and challenges. When I was reflecting on these presentations and also preparing for today, I have thought of three things on my mind. It's the why, it's what, and it's who. And in particular, I think with the presentations today, it's clear that the target trial emulation framework is definitely shown potential to minimize bias and also to make us better in designing the observational studies. It certainly has a potential to bridge the real-world data into our clinical development programs within the industry. But it's not there to replace the randomized controlled trials, rather to complement. And I think the most important thing has also been touched upon today is really to focus on what are the purpose, what are the questions that are suitable to be answered, what are the type of trials we would like to articulate. And I have seen kind of at least two different two-folded approaches. One of them being you want to create or emulate a hypothetical trial that you're not really able to do in the real world or in practice. The other one is this about benchmarking an existing trial. And one of the examples could be that you have a large-scale point of care trial running, randomized trial running in one country, where you're looking at the treatment effectiveness, maybe even on a long-term outcome like a cardiovascular event. And then, the question is, of course, it still remains unclear whether the differences in health systems could have an impact on the generalizability of this trial running in this particular country, or even the transportability into other countries. So I think that's really an area where this question, what are the research questionnaire, that's really an area where there's an interest in really trying to understand whether we can create a target trial emulation to benchmark such a point of care trial. The other point I want to go into is the what. I think it's quite clear that when you plan, execute, and report a randomized controlled trial, we have very, very clear guidelines on that. We also have standard operational procedures that we follow very, very closely. However, there are still some uncertainty in the implications of target trial emulations for regulatory acceptance and, of course, the decision-making as such, where I think even looking at this reflection paper and now this particular tool being called out really speaks for, okay, how can we start the dialogue at an early stage, even be part of maturing and be part of the transparency that we have discussed also today. But I still see that there's still a lack of guidance. We know that there's some really nice templates for how you design, how you set up the target trial, but this about, for example, how do you report? What are the requirements for the reporting? That's maybe still an open question. The last part is the who, because I think there are also opportunities for collaboration here. Could it be that we still lack a lot of experience and lack also the expertise? And suddenly we have seen a lot of the results coming out in scientific aspects, but this about bringing it into the industry, I still think there's a lot of need for continuous collaboration between academia and also agencies and sponsors, not just between, but also across, definitely. And I could also maybe come with an example from the US, where FDA has now opened their Advancing Real-World Evidence program via the PDUFA. The industry can submit several proposals every year, and then there's actually a track with several opportunities for interacting with the agencies. So something like that, a pathway where we can have more deep dive conversations, could be one way, one option among others as well. So I think with that is kind of my conclusion for setting the scene from my side. Thank you so much.
Thank you very much for those perspectives, and I think you've highlighted some of the challenges, but also might be needed to overcome some of those challenges as well, which we may expand upon in a few minutes. So move on to Anthony Matthews now, who's an associate professor in epidemiology and causal inference at Karolinska Institute. And his work focuses on developing, refining, and implementing different causal inference approaches like the target trial emulation. So Anthony, over to you.
Thank you very much. Yeah, and thank you again for the speakers and that talks. They were very insightful. So I'm going to talk about the target trial framework mainly because that's where my expertise lies. So generally, we believe it's a powerful tool that provides an organizing principle for the design of observational studies that use real-world data. And I think the most powerful aspect is that it really helps us articulate a causal question, which lead to clinically interpretable results that can then be used to inform a decision. And I want to make 4 main points. I want to build on Zabi's remarks about the effect under complete adherence to treatment strategies. I want to talk about the fact that I actually think target trial emulation is a 3-step process, not a 2-step process. I want to talk about how it's created a common language between trial investigators and researchers that use real-world data. And then finally, I'll just touch on one misinterpretation. So building on Zabi's remarks, the 2 main points he made were around the alignment of time zero and the effect under complete adherence to treatment strategies. Now, a lot has been said in the literature and here today about time zero, so I don't want to dwell on that, but I'm going to echo Zabi's point about how we need to clearly specify the treatment strategies we're interested in, and I want to reiterate their importance. So it's been said by several people today. One place I think we can continue to make improvements in observational studies is clearly specifying the treatment strategies we're interested in, which could include, for example, treatment switching. Then we estimate the effect under full adherence to them. So now, traditionally in observational studies, we've been very vague about what our exposure is. For example, many papers might have said they wanted to estimate the effect of, for instance, beta blockers versus no beta blockers on the risk of death. Now, this is non-specific because there's no detail on duration, dose, how often treatment should be taken, stopping rules. And the result of that are observational studies that estimate an effect that incorporates the mix of heterogeneous use patterns that are difficult to clinically interpret, different adherence, different doses, things like that. But if we predefine specific treatment strategies we're interested in, then we can estimate the effect under full adherence to them, as we talked about. And of course, we need to use the correct methods to account for potential time-varying predictors of non-adherence to those treatment strategies. And I actually believe a lot of this confusion is stemmed from the Strobe guidelines for observational studies, which just states that your exposures must be clearly defined, which is ambiguous at best. Whereas the consult guidelines for RCTs state that investigators must define the treatment strategies of interest, which clearly define how and when the treatment under study is to be used, that is dose, duration, and reasons anyone's allowed to stop. And hopefully this can be addressed in the target guidelines that are being produced now, and I believe Zabi is a part of that too. So secondly, I want to talk about how I believe target trial emulation is actually a 3-step process, not a 2-step process, which we've seen today. So in all target trial papers, we see a table that outlines the target trial and then how it was emulated. The target trial column outlines the target trial that can be emulated in the available observational data. But in practice, there is, or at least there should be, a step before that where we specify the target trial that we'd ideally like to conduct. So what we do is we first specify this ideal target trial, and then we engage in an iterative process until we are satisfied that the target trial protocol reflects one that can be emulated in the available real-world data. The question then becomes, does our updated target trial answer the same question as our ideal target trial? And in many scenarios, the answer is probably no, because the observational data aren't collected to make causal inference. So we won't have the data required to answer that question. We'll then need to either collect more data or look for additional data sources. However, to really articulate precise causal questions, the specification of the ideal target trial is important and should actually be independent of the data source that one has available. Thirdly, I'm going to talk about how I believe the target trial framework has created a common language between trial investigators and researchers that use real-world data. I think one touched on a little bit. So I work a lot with trial investigators. And when we first started working together, I had a bit of a joke in our group, and we talked about how they had separate trial glasses and separate observational glasses when they were interpreting studies. Their way of thinking about and interpreting these studies were completely different, even though the general aims of what they wanted to do was exactly the same. They wanted to, the cardiologists, they wanted to estimate the effectiveness and safety of some cardiovascular treatment. Now, teaching them the target trial framework has provided a common language between us both. So what we can do now is we can work with the trial investigators and the people who use observational data to work towards the same aim. And I believe this can create a critical mass of effort that is greater than the sum of its parts. And finally, I want to talk about one misinterpretation of the target trial framework, which perhaps is not present in this room, but generally. So we receive it regularly from reviewers and in commentaries and high impact journals, is that the target trial framework is an attempt to replace trials with observational studies. This is as far from the truth as you can probably get. All we're trying to do is to make observational studies better. And the main limitations of these studies still persist, like confounding, for instance, but they aren't compounded with additional problems like misspecification of time zero or unclear treatment strategy protocols. And if anything, the increased use of the target trial framework is an opportunity to find places where observational data can complement trials to support regulatory decisions, not to replace them. So overall, the increased use of the target trial framework in studies that use real-world data can provide a base for in-depth discussions between experts to decide what is and is not acceptable in relation to study design. And I think it also provides a link between observational studies and randomized trials, so the design quality of all studies that ask questions about the effectiveness and safety of treatments can be judged symmetrically in regulatory decision makings. Thank you.
Thank you so much, Anthony. I think it's really interesting to see some commonalities in the importance of the type of trial that this might be applied to for what Helen was saying as well. So we'll just move on to Ria Fitzgerald now, who is a medical doctor who works at the Health Products Regulatory Authority in Ireland. But also she sits as a prac rapporteur, but also interestingly has a master's in both epidemiology and statistics. So I look forward to hearing what she has to say. Over to you, Ria.
So many thanks for the excellent presentations today and the views from the previous speakers, and for the opportunity to provide my personal reflections. So during my master's, I actually explored propensity score methods in an observational study comparing treatment with active comparator for COPD. And although the aim in that was to replicate the intention to treat analysis of a trial, some observations struck me as quite relevant to today's discussion. So quite a number of exclusion criteria had to be applied to emulate the trial, and propensity score matching with backward selection was used to balance baseline characteristics. Notably, there were some indications that the observational study cohort had somewhat milder COPD, and a prior treatment covariate, which was not captured in the propensity score model, remained quite strikingly imbalanced. And there were a number of other covariates that appeared important in the causal relationship that were not available, for instance, information on smoking patterns. Despite this, the study did appear to replicate the findings of the trial, although the trial also included a placebo-controlled comparison, which the study could not replicate. So the question, I think, for me today, given my background, is whether TTE, which has largely been related to effectiveness to date, it appears, could also be relevant to post-authorization safety monitoring, where it is often neither ethical nor feasible to conduct a clinical trial, and so there is a focus on non-interventional studies. And selection bias due to the presence of prevalent users where mortal time can be very problematic. There may also be a lack of clarity in relation to the interpretation of the estimand, and also the impact of issues such as competing events. And methods used to adjust for baseline covariates can lead to bias when trying to deal with time-varying confounding. So having a structured approach at the outset with explicit consideration of such bias and the relevant estimand does have the potential to optimize post-authorization safety studies. And in a broader sense, the principles are applicable to the assessment of real-world evidence, in particular non-interventional studies, for instance, on safety signals. However, there are a number of aspects to consider. So firstly, the application of eligibility criteria may be conducive to causal inference, particularly for quantifying an effect, but it may limit generalizability and potentially also sample sizes. And pharmacovigilance is by its nature the study of heterogeneous populations. And also situations where TTE potentially would not be feasible do tend to arise. For instance, there may not be a suitable data set or a suitable comparator. And even if a suitable data set appears to be available, if there's insufficient information on time zero or substantial missing data on a key covariate, that could hinder the TTE approach. And if not all elements of the trial can be emulated, the results of the TTE may not approach those. And even if all the elements of the trial might be emulated, it is important to acknowledge that the dataset ultimately remains observational. Yet a recent systematic review highlighted that among studies implementing TTE, only 37% reported on any unmeasured confounders, and only 39% described the method for selecting variables. And another review highlighted that among studies implementing TTE for intention to treat effects, a proportion used conditioning on baseline characteristics rather than methods which would allow a separation of the design and an assessment of balance before the analysis, such as propensity scores. And furthermore, more advanced statistical methods are required, particularly for per-protocol effects, which may be of more interest for evaluating safety. There is also a need to consider other sources of bias, such as the lack of blinding. And I think these considerations may be important where in the safety setting, there may not be a target trial available against which to validate the approach. So overall, I do think TT is a promising approach. I think particularly if accompanied by a proactive assessment of the risk of bias, and more generally speaking, its core principles should be considered in the assessment of non-interventional studies. But it does require a sufficiently detailed data set and specific techniques. And there are some specific considerations in the setting of pharmacovigilance, such as generalizability. And therefore, I think it may still need to be considered in conjunction with other available data, though it could certainly support and improve the evidence considered in the regulatory decision-making process. Potentially further guidance on the approach and when it might be considered would be important for its implementation in the regulatory context. Thank you.
Great, thank you very much, Ria. And it's really interesting to also hear that you make a point that target trial emulation may not be feasible at certain times, and we hear a lot about how we should use it in depth. But the detail of also what Anthony was saying about and other colleagues about design the trial that you would like to do and see if it can be applied in the real world data, and if it can't, but then what the basis of that decision is, the devil in the detail, when it can be used and when specifically it can't, maybe is an area for further discussion at a later stage. So we're running a little bit behind time. So I think very briefly, if there's no questions in the chat, but maybe we could go around if there's one key point or something any of the presenters or even the panelists would want to make a takeaway message, please, now is your time. Anthony, please.
Yeah, I think it's actually just one challenge that I think we have with pushing this forward a little bit is that we need to really convince people that observational studies need to be put under the same scrutiny as randomized trials. Because one of the big parts of the target trial framework is that you need to create this incredibly detailed protocol to clarify the estimate you want to estimate the effect of. And it's commonplace for trials to do this, and investigators do feel a responsibility because they're essentially experiment on people. But researchers with real world data generally don't feel the need to go into so much detail because there's no immediate risk, because they're just using data that's already been collected, right? There may be a delayed risk if they get the incorrect results. But this sort of real in-depth development of the protocol is required to generate clear, actionable evidence. So if we can find a way to hold observational studies to a similar standard to trials, then I think we would see that the quality of the evidence that we generate would increase.
Thank you. Juanjo?
So one point, I've heard for many years now, people in the real world data saying clinical trials are experiments that are kind of in a bubble and they do not reflect real world, so they're not extrapolable or generalizable. On the other hand, I've heard people in the clinical trials world saying, oh, evidence from real world evidence cannot be trusted for a bunch of reasons. Study design, lack of data quality, etc. I think this type of messages are not helpful. We all pursue the same objectives, which is generate evidence that we can trust and rely upon to make decisions, data-driven decisions ideally. So I think the target trial emulation and all the tools we have discussed in the estimate framework can help to, as I said, to establish this bridge and make it strong, and maybe stop that type of kind of negative messages thrown at each other, which, as I said, do not help.
Well, thank you. On that note, we're going to bring the session to a close. It's been there is a question in the chat apparently. Yes, Danny Preter-Lambert, would you like to come in?
Yes, can I ask a question? So my name is Jan Cornelissen. I'm a seconded national expert at EMA, but a physician, professor of hematology at Erasmus University, and I headed the prospective hemato-oncology study group HOVON, who performed many randomized studies the past 30 to 40 years. So as we witnessed until now is that the theoretical framework for generating real-world evidence has been developed to a high degree. I'm impressed. But the studies, the concrete studies being done are lacking a little bit behind, to put it even mildly. So for instance, large groups as EORTC or HOVON have hardly performed these kind of studies, and if they do, they can learn a lot about the quality we heard this morning. So this is more or less the background of my question, Dani. I would like to ask you, in your HPV vaccination studies, did you monitor toxicities? Did you monitor comorbidities? So in order to come up with a benefit-risk assessment, you also would need to identify the risk, the toxicities, and so on, and especially in individuals with pre-existing comorbidities.
Thank you for the question. Yeah, so this particular study is not about safety, it's about the effectiveness of the vaccine. So the study, by definition, by design, doesn't include safety outcomes. We do adjust for comorbidities and risk factors and so on at baseline, as I explained, using a number of strategies. But the study is not about safety. Vaccine safety studies usually benefit from different alternative study designs, and sometimes we prefer to use other designs for safety compared, typically because effectiveness, especially in this particular vaccine, is expected to be more longer-term outcome, while safety typically happens in the shorter term. I'm not saying it cannot be done, but it wasn't requested as part of this study. Thank you.
Well, thank you very much. And thank you again to all the speakers and the panelists. And on behalf of Harold and I, we're going to close the session just now and hand over to Peter before lunch.
So thanks very much. Great session. Thanks, Harold. Thanks, Daniel, for chairing, coordinating it. For those people that are online, we're going to break now until 1:30, but we will start pretty prompt at 1:30. So there's a little technical advice. Leave your WebEx links open, just it prevents then problems when you try and rejoin. For those that are in the room, it's photo time, okay? I was inspired by Eleanor, who's LinkedIn mad, and she's sending photos from the back, but we're going to have a better photo, Eleanor, with everybody together. Now, can I please ask, Very tall Danish people, Jeppe, stand at the back, okay? And little petite people from Portugal, for example, stand at the front, okay? But we get really close, don't breathe, and then there can't be a virus risk. And then once we've had the photo, which will be taken very efficiently, there's a sandwich lunch outside. Okay, so we're going to close now the meeting. Stay online remotely and photo then lunch. Thank you. Can I just ask, Gosha, Juliana, are we broadcasting again? We're ready to start? Okay, so the control center says we're ready to go. Hopefully everyone's had a good break. And we're going to go into session 2B, because 2A was not enough. So we're going to talk about real-world data-derived external controls in clinical trials. And it's a great pleasure to introduce Carla Torre, who's come all the way from Lisbon and is a member of the CHMP with a special interest in real-world evidence, and Marcia Ruckbell, who's a biostatistician here in the Data Analytics and Methods Task Force, to navigate us through this session. Over to you.
Thank you, Peter. Good afternoon, everyone. So in this session, we will cover how real-world data derived external controls in clinical trials can somehow support regulatory decision-making. So what are the role of these external controls in medicine's development? What are the key challenges? What are the key uncertainties, and how somehow that influence the final regulatory decision-making? What are the main considerations in what concerns to design, in what concerns to analysis, in what concerns to the methods? So some of these topics will be covered in this session. Overall, this session has 3 parts. So the first part will have some introductory remarks in order to set the scene about the external controls. In the second part, we will have the presentation of the 3 use cases from the industry, from the FDA, and from the EMA. And in the last part, we have a panel discussion with different stakeholders across our regulatory system, namely pharma, regulators, academia, healthcare professionals. And I believe that we'll have some time in the end for a Q&A. So we will start with Elina. Elina will kick us off to this session. Elina is a statistical assessor at the Finnish Medicines Agency. She's a member of the Methodology Working Party, and she's also a member from the Scientific Advice Working Party and a very brilliant statistician in this field. Many thanks. Elina, the word is yours.
Apologies that I cannot be there in person, and I'm also just under a thunderstorm here in Finland, so I hope it doesn't create too much background noise. It's hailing and I can hear the thunder, at least myself as well. Okay, so this is the usual disclaimer. And yeah, as Carla already discussed, this is the scope of this session, or at least the scope of my presentation. So already in ICH E10, we have externally controls mentioned, an external control trial being something that compares a group of subjects receiving the test treatment with a group of patients external to that study. And here in this presentation, we also focus mainly on external controls in the context of pivotal clinical trials. And external controls where the data source is real-world data. Data source for an external control could also be a non-interventional study, or it could be a clinical trial, or maybe something else even. But here we really focus on the setting where the one part is a clinical trial, and then that is compared against an external control coming from real-world data. So we are also not discussing hybrid designs where the control arm would be a combination of internal and external control. And I'm also kind of course, in the remit of this workshop, discussing a situation where the external control study has a role in a regulatory submission in terms of being some sort of pivotal evidence which is relevant for the decision making, right? So it doesn't have to be the only piece of evidence, but it is something that is intended to be taken into account in the regulatory decision. And not only supportive evidence or no evidence. And what could that no evidence be? It is, of course, the sponsors also do a lot of drug development in a way not intended for regulatory purposes. For example, when you do your own proof of concept studies or your own analysis which molecules to take forward in development, this kind of things, there you may, of course, for your own internal decision making, use different standards than we use here for regulatory decision making. So here we focus on the studies which have some sort of a pivotal role in regulatory decision making, which is then not the only use of external controls. So then, when we do benefit risk assessment, when we assess marketing authorization applications, we end up with a binary decision, right? Something is either approved or it's not, or it's given CMA or it's not given CMA. But that decision is, of course, not based only on the p-value on the primary endpoint, but it's based on a big part of evidence and usually several studies and pharmacokinetics and all kinds of things. And we factor a lot of uncertainty during the review of those marketing authorization applications. And that can be reflected in the EPAR, in the European Public Assessment Report. Sometimes that is reflected by exclusion of data from SMPC section 5.1. So the sponsor may propose something to be included there, an endpoint, and then we conclude that though it may be clinically relevant, but because it's not somehow reliable enough, it's not there for inclusion. And also it is important to take into account that when that final decision is made, maybe to grant marketing authorization, that there sometimes are post-marketing commitments, right? For example, post-approval safety studies. So we end up with a binary decision. There is some, can you hear me well? I hear some background noise.
We can hear you well, but probably someone has the computer on. And
Yeah.
we'll see some feedback. Try again, please. Thank you.
Yeah. So, where was I? So we evaluate a lot of data which ends up in a binary decision. And the benefit risk assessment importantly considers also multiple endpoints for one study, right? And the important role of RCTs, randomized controlled trials, is that they provide comparative data for many endpoints, right? From safety to quality of life, and from safety also things like tolerability and whether the patient were able to receive in an oncology study the background chemotherapy as planned in both arms, things like this. So we get a really granular data for a lot of data points, pharmacodynamic markers, clinical secondary endpoints. So it's a big data package that we evaluate. And something that I also want to mention here, that though it has been said already many times in this workshop that an RCT is kind of the gold standard, they are still not easy and straightforward. Yes, they are the gold standard to make the causal claim, right? That is the most powerful tool if the intention is to make a causal claim, right? Distinguish treatment A from treatment B or treatment A from placebo. Yes, they are, but they are still not easy and straightforward. We have a lot of methodological considerations there, and a lot of things can happen in RCTs as well, but we know how to assess these uncertainties, and we have that toolkit for that, and that's basically, in a way, my day job, right? Either in giving scientific advice or assessing marketing authorization applications. So it's important to keep in mind as well that RCTs are not easy to assess. So then, when we now try to what we just discussed and think about, okay, how does it impact that benefit risk assessment if we actually have an external control from real-world data as part of that benefit risk assessment? So what does it mean for us? And I think the most easy answer is it adds uncertainty, right? There's always uncertainty, but it adds uncertainty. And for example, not an exhaustive list, something that puzzles my mind a lot as a numbers person is that the data source and treatment are two identical variables. And what I mean with that is that if I think about my data set and I have their treatment, and I have 1 for treatment A and I have 0 for treatment B, I have a second variable which has the data source, and I have 1 for clinical trial single-arm study, and I have 0 for real-world data. So in a way, the data source and the treatment are the same variable, the same, though, and the treatment is the one that I'm interested in. It is that one that I'm trying to estimate is the treatment effect. And that is directly identical by definition with the data source. And that, of course, raises the big question then in my mind that how can I distinguish from estimating a treatment effect from estimating a data source effect? Okay. Data generation mechanisms are fundamentally different, right? So when I assess RCTs, benefit risk based on RCTs, I spent most of my time looking at patient disposition and intercurrent events. I look at all those post-randomization events that happen and may cause imbalances and may bias or impact the treatment effect estimate in one way or another. But because in real-world data versus a clinical trial, those things are fundamentally different by definition, it's difficult to assess those, right? And usually in this kind of external controlled study, they are usually based on one endpoint, on the one most important primary endpoint for which real-world data is available. But like I discussed, we usually are interested in much more, right? So then I'm lacking the secondary endpoints, I'm lacking the safety data, lacking tolerability data, discontinuations because of tolerability issues, all kind of things, PD markers, right? And these are important to understand the totality of evidence, but they are also important for assessing consistency of treatment effect, right? You usually want to see a benefit in multiple efficacy endpoints. So basing kind of a benefit risk assessment on one variable only, though it may be the most important one, still leaves us short-handed. And I also, of course, do not in any way want to undermine the big field of epidemiology, which has for decades developed methods like we just discussed with target trials to take these things into account, right? To kind of consider these uncertainties in a systematic way. But they are mainly focused still on situation where we look at observational data from the same source, right? Which is different than looking at this comparison where one treatment arm comes from a clinical trial and another treatment arm comes from real-world data, right? And in this specific situation, I think these tools leave us with more like this absence of evidence type of situation where I'm more interested in evidence of absence of bias, right? Whereas that I don't have hard evidence for bias, right? For me, in regulatory decision making, these are fundamentally different situations to be in. So something then that may be considered, I wanted to raise here something a bit controversial in a way, that we all know that there has been approvals in recent years based on single-arm trials. So then one may wonder, why are the regulators so reluctant against external controls when they are willing to give approval based on single-arm studies? So wouldn't the external control at least still increase the evidence package compared to a single-arm study? And I think here the key point is to understand that the single-arm study-based approvals fulfill certain core elements which are well reflected in the reflection paper of single-arm studies that has been published. And there the most important one is an endpoint that can isolate a drug effect, right? So it's the example here is a response in an oncology study, right? That a response cannot happen in the absence of treatment. So we are looking there at absolute treatment effects. Or if you want to see it as a comparison, it's a comparison to non-zero response to placebo, but it's an absolute treatment effect. So the approval is based on descriptive data rather than hypothesis testing and rather than a comparison. Whereas if you look at an external comparison, you look at a difference between arms, right? So you're looking at the difference between two arms. So you need to make sure that your comparison is comparing apples against apples, right? And their exchangeability is the most important element because it is a comparison. And that is a multifaceted concept which includes everything from population to clinical practice. Clinical practice could, for example, be this, time bias. How is your data contemporaneous or is it historical? And from endpoint collection to intercurrent events, right? So that is, I think, something why we are so have so much difficulties, right, with these external control comparisons, because we can't be sure that we are comparing like with like. And most clinical trial endpoints that we are interested in, right, usually the most relevant clinical trial endpoints do not single out a drug effect, right? So for example, if we then used for single arm studies as an example response rates, but then when we look at a comparison, an external control in PFS or overall survival, those endpoints do not single out a drug effect because people die without the drug working, right? So it's fundamentally looking at a comparison where we need to worry about exchangeability. And single arm studies are suitable only for the situations where the endpoint can isolate a drug effect. So then the take-home messages here from me are that the bar for evidentiary standards in efficacy for benefit risk assessment is high for the right reasons. And by definition, an external control from real-world data adds uncertainty in decision-making as compared to an RCT. And this uncertainty is twofold. It is embedded in the treatment effect estimate, so in the comparison of the endpoint that we are looking at, but it is also in the totality of evidence, right? So usually this external control study can always give us less. There is always less endpoints that can be derived from that real-world data source in a reliable manner, right? So we are missing endpoints, and very importantly also missing endpoints for safety. And this additional uncertainty is rarely scientifically justified because alternative designs with less uncertainty are usually possible. So if you think about scientific advice, and maybe one could say that it's maybe quite conservative scientific advice that we give to many external control comparisons, is because we evaluate them also against what we think is possible, right? And usually something else with less uncertainty is possible. But then to end on a positive note and kind of set the scene for the discussions to follow, I guess I would be then interested in discussing in which settings does external control rather complement the evidence base than add uncertainty. Thank you very much.
Thank you very much, Elina, for setting the scenes about the challenges, I would say. So we will move for the second part of this session. That will be the use cases. The first use case will be the industry use case that will be presented by Moril Tepi. Moril Tepi is a statistical epidemiologist with over 20 years of experience in pharma industry. He has a PhD in medical statistics, and he's currently the global head of real-world evidence and digital science function in UCB. Many thanks, Moril, for being with us. The floor is yours. Thank you.
Thank you, Elina. Thank you for the opportunity. And again, it's really our pleasure to have the chance of sharing this ongoing example, practical case where we are using external control arm for regulatory interaction. And I hope from, again, the previous speaker, this external control will not be adding uncertainty. It will be kind of reducing the amount of certainty around the data we have in our single arm. So I will be talking to you a bit about the condition. We are applying it here, the TK2D condition. Talked a little bit about the treatment option, actually the lack of treatment option here. I will also share the challenge we were facing or anticipating to face if we were to do a traditional phase 3 confirmatory trial. And then hopefully outline why we think an external control arm will be the most ethical and viable alternative in the particular situation. And then I will share a little bit our external, how we went about this external control or how we are going, the source of the data, analysis and methodological conclusion, analysis and methodological consideration, and then I will conclude with one or few questions. So before moving on and share about the background, I thought we need to bring these 5 key points we think are some of the key drivers of considering external control arm. Again, hopefully to reduce the uncertainty rather than to add the uncertainty. The first point is where, in principle, you see randomized control trial being not feasible for practical reason, for ethical reason, and it seems to be in rare condition or rare disease. This seems to be more or less the case. Secondly, if you have situation where you have a serious considerable unmet medical need, and then you think we need to consider all the options into detail, but practicality should not be the key hurdles. Thirdly, where we have a well-defined, well-understood natural history. Why is this important? We want to make sure that we are putting our finger on the key prognostic factors so that we can really contextualize what we are seeing and as we design our external control arm. The fourth point, to have an objective endpoint, something for many reasons, including the fact that it's something that has value in a routine clinical practice. And then secondly, because we will be using data from routine clinical practice, it's readily available there. If you use biomarkers, which are not necessarily collected regularly, you will not find it there. You will find it make it difficult. Last but not the least, the effect size. Your anticipated effect size should be big enough. Again, why is it important? Well, you will be using external, the real world data to try to contextualize or to compare what you observe in a trial, in a investigation drug to what you are observing in other population. There is always, and rightly so, the question are the two populations comparable? Don't we have other confounding factor, even if you adjust some remaining factor? The key point here being, if the effect size is big enough, you could argue that even by adjusting this element, I may not necessarily lose my effect size. But if the effect size is marginal, then you have a bit of problem or justification. So we thought we need to put this on the table and could fuel some conversation on that. So back now to the condition. Yes, this is TK2D. It's a mitochondrial deletion and depletion syndrome with substantial morbidity here, and underlying substantial progressive muscle weakness and considerable mortality, you will see later on. It was first described in 2001 in 4 children and with 4 different mutations, so a lot of variability. And actually, with this condition, you have more than 50 TK2 mutations that are linked to the condition. The similarity in presentation with other neuromuscular conditions make it really a lot of misdiagnosis and considerably underdiagnosed. Most of the patients with this most of the patients have childhood onset, more than 84%. So, as I said, it's a ultra rare condition. Less than 2 out of 1 million people. So it does provide a lot of challenge. And actually, there is no approved medicine out there in EU to treat the condition. These conditions are managed the management is limited to supportive care actually, and the supportive care being provided have not changed since the initial characterization of this condition. The drug we are working on, the investigational drug we are working on, will be labeled here DCT, and has been granted orphan drug designation in 2017. Subsequently, it was deemed eligible for the PRIME program. And then our preliminary data suggests that we have a substantial reduction in mortality with this compound. So there is a lot of promise behind it, particularly in children under 12. And then we plan to submit it in the near future. And again, as I said, the ultra rare condition, and you could anticipate how difficult it will be to run a proper phase 3 confirmatory trial with acceptable sample size. You can see on the right there how the Kaplan-Meier, in particular the survival for the patient under 12. Our new therapy is very promising. And back to the point I was flagging for Elena is to say, we do have a situation where we look at our single arm. The drug is promising, and we believe some context is better than no context at all. Hence the idea of considering an external control arm to further contextualize our data. So again, I just mentioned it, strong belief that the most ethical and feasible approach is to consider the external control arm. And I'm summarizing again the 5 key bullet points we have here. So our objective here in this was to characterize the clinical costs, including survival, I mentioned it, hard outcome, loss, gain or regain of motor milestone, including a respiratory and feeding support for this patient, both treated in our single arm and treated in the external group. Describe as well how are the safety and tolerability occurrence of this investigational product. The challenge, as I said, is in ultra-rare disease, but at the same time, we could not expect to use large conventional multipurpose data as a source of the data. And also, there is no existing registry for the disease from which we could extract our external control arm. So there is a call for external control arm, but we also have the challenge of the data source. So what we did, we built a new repository data using retrospective data, but with no prospective follow-up. So systematical literature review, go to investigator and trying to ask them to collaborate with us to give individual patient data, collect additional data. Again, as I say, it's an extremely rare condition, so we'll do with anything we can find. Our treated arm are made of our patient from our investigation from our trial, plus patient receiving our compound as part of sponsor compassionate use program. So again, what are some of the critical analytical considerations we have here? First, we make sure we have a hard outcome that we can find in the clinical practice survival. We also try our best to make sure that we will have comparability here, and then at the same time, our appropriate control for potential bias. Selection bias, potentially less an issue here because we are taking any patient we can find, given that the universe is so limited. Immortal time bias is a consideration. Xavier mentioned this morning the time zero, we are hoping with the matching treated patient from age of onset to the treatment to untreated one, assigning the index of the treated to the untreated, somehow take care of this. Confounding by, you have a reasonable understanding of the disease natural cause, and the age and onset is one of the key drivers. We are making sure that patients are being, matched on those elements. Just mention the current knowledge of the natural history, mention the similarity, and then we have a kind of a battery of analysis, cost proportional hazard analysis, the marginal to complement the cost into the sense that our cost stratify is stratifying on the matching pair, and we all know for the matching pair, the more value will come from concordant concordant pair. And we lose some of the patient because of discordancy. The marginal try to capture those elements, but we also can make sure that we optimize with appropriate variance covariance structure. We also use exact conditional logistic regression to try and sample size being small, exact approach will be potentially more amenable, but with the drawback that we lose the time to event and then just consider death, not death situation, and other battery of sensitivity analysis as well. So with that, that's key point we mentioned, and then just to stay in time, these were some of the key questions we had in our mind, and then we said we can throw it here for us to think about. First, the data. As you can see, we are scratching our head as best as we can to find some data to make sure that we provide some concept to our single arm. And then there is this challenge, how good look like for this. And then the second point here is to your point early on, how much of the clarification could we have had upfront as we prepare for this filing using external control arm? Thank you.
Thank you very much, Muriel. We will have the discussion in the end, but thank you for flagging these two questions. We will continue on the landscape of the use cases. The next speaker is Donna Rivera. I don't know if she's online. Donna, yes, she is. Many thanks, Donna, for joining us from the other side of the Atlantic. Donna is a pharmacist and pharmacoepidemiologist, and she's the Associate Director for Pharmacoepidemiology in the Oncology Center of Excellence at the FDA. She leads the Oncology Real-World Evidence Program that is focused on the use of real-world data and real-world evidence for regulatory purpose. Many thanks again, Donna, for joining us. And the word is yours.
Hi, good morning.
Thank
or
you
I
very
should say
much.
good
Thank you.
afternoon. Good afternoon, everyone. I'm so pleased to join. I think it's been a really great discussion so far. And so I'm Donna Rivera, and I'm excited to present to you about externally controlled trials in oncology. So I've pretty much put this talk into 3 key components. I'll talk a little bit about our externally controlled trial guidance. Then I'll talk a little bit about the challenges we have had associated with the use of externally controlled trials from a methodological perspective. And then I'm going to provide an example of our most recent approval using an externally controlled trial. So beginning with guidance, the FDA has been quite active in the area of RWE guidances led by the Office of Medical Policy in collaboration with CDER, CBER, and OCE. And listed here are several recently released guidances that cover 3 key domains, really around real-world data sources, around submissions of real-world data, and around design. And today we're going to focus specifically on design and the design of externally controlled trials. So in this specific guidance around considerations for design and conduct of externally controlled trials for drug and biological products, put together by CDER, CBER, and OCE, for the purposes of this talk, I'm going to use this definition, which is that an externally controlled trial measures outcomes in participants receiving the investigational treatment according to a protocol compared to outcomes in a group of people external to the trial who did not receive the same treatment. And really, the appropriateness and suitability of an externally controlled design depends so much on the clinical setting. So it's important to consult the relevant review division early in drug development to determine if an externally controlled trial is reasonable. So thinking about reasonability and rationale, before selecting an ECT design, it's important to consider the likelihood that such a non-randomized design would be able to distinguish the effect of a drug. In many situations, the likelihood of credibly demonstrating the effectiveness of a drug with an external control is low. And there are factors that affect the suitability of an externally controlled trial. And if you just heard in the prior presentation, some of these, but around the heterogeneity of the disease, the preliminary evidence that's already available for the drug or medical product, the approach to ascertaining the outcome, and the specific goal of the trial around superiority or non-inferiority. And so it's important to think about this potential context for use. So I cannot emphasize enough that when you can randomize, you should randomize. But today we're talking about situations where an ECT may be appropriate. So typically the context for use in the use of an ECT design is when there are feasibility challenges, ethical concerns, or questionable equipoise. And some potential applications where you may see this are in the pediatric drug development, rare diseases, areas of significant unmet medical need, molecular subgroups, and underrepresented populations. Within the guidance, there are really 4 key components around ECT considerations for design, data, analysis, and review. I'm not going to go through the entire guidance, and so I'm just going to highlight a few key points. One of those points is, what is the regulatory objective? So as we're thinking through this, what is the level of evidence needed to demonstrate effectiveness? So an external control can serve as a benchmark, a baseline natural history, more of an epidemiologic benchmark, or as an individual patient level match study for formal comparison. And in the context of that, and really the concept we're talking about today, it's really important that the data are fit for use, they're relevant and reliable, that the program uses an adequate study design and that there's appropriate study conduct. And so there are a lot of factors that go into this and really an intense amount of upfront planning. But just to mention that assessing the comparability of various key factors upfront can help elucidate potential sources of bias and also think about identifying where sensitivity analysis or quantitative bias analysis can be implemented to assess the robustness of treatment effects where differences may occur. And the following are some important factors. And again, I think you've heard some of these, but thinking about time periods, standard of care, geography, treatment characteristics, index date. I think time zero is a significant and specifically important issue in the conduct and design of externally controlled trials, as well as measurement of the outcome and, of course, missing data. So I think this comes to the conclusion from a regulatory perspective that it's important to consult with a relevant review division early and often. So now I'll move to some challenges when it comes to methodological approaches in the use of externally controlled designs. And so one way to conceptualize externally controlled designs is to think about, again, defining the study objective and key design elements, then thinking about determination of whether the external controlled data that is proposed is fit for use. And only if it is fit for use, then it would be logical to think about putting together a detailed study protocol and robust statistical analysis plan a priori. And here listed are some important factors around data source, population, comparator that we think about. And when we think about these, we think about them really in two categories around completeness of capture. Do you have all of the data captured that you really need in the context of this study to conduct a well-designed external control, as well as the comparability of the populations? So making sure you have all the data available and then assessing the comparability to understand whether an externally controlled design is really going to be adequate to demonstrate the evidence necessary. So based on externally controlled designs used in previous regulatory submissions, these designs were typically used in a scenario when there was limited or no randomized studies available, and often limited to providing important clinical context. And then often in these applications considered supportive only. And that was because there were several limitations. Those limitations included the lack of a pre-specified protocol to ensure that comparability of patient population, and of course, led to concerns for selection bias, the lack of formal statistical comparisons or statistical methodology that was not established a priori, data source appropriateness. So was there available covariate ascertainment, available demographic information necessary, or was there information on the outcome that was well ascertained, or were there concerns about outcome misclassification? Of course, confounding and concerns in that context of measured, unmeasured, and residual confounding. And then, as I mentioned before, a really important aspect of index date selection and the concerns, therefore, with lack of understanding of that index date selection or lack of ability to adequately create an index date that was comparable, and then leading to immortal time bias. And so it is logical to note that the vast majority of approvals were based on clinical trials using a single arm, potentially with response as evidence of clinical benefit in these contexts because of the challenges associated with externally controlled trials. However, there is a recent example, and so I'd like to talk a little bit more about that and about our approach to regulatory review of externally controlled trials. So this is a bit of logic in the stepwise approach to evaluation of external controls. So first, it's whether the external control is appropriate for use, right? Is the rationale and justification appropriate in the clinical context? Are the data fit for use? And then moving to the second of these are really specific to our U.S. regulatory requirements, but whether there is an adequate and well-controlled study, whether or not it's able or sufficient to establish substantial evidence, and then overall the risk-benefit assessment. So I will briefly go over a portion of these steps as is relevant, and then talk about the example. So really, thinking about the context in evaluation of an external control, it's important in accordance with our guidance that this be a situation where the natural history of disease is well defined. The external control is very similar to the treatment group. Concomitant treatments that affect the primary endpoint are not substantially different, and there is evidence of change in the progression of disease in some context. For example, in a tumor shrinkage context situation. And of course, the data must be fit for purpose, and including the relevance and availability of key data elements and the reliability of those data elements. And so there are a lot of scientific considerations that go into this, and therefore it's a very careful and robust stepwise process. And I would just note that I'm not going to go through every single step, really just covering step 1, because steps 2 and 3 are very specific to our regulatory requirements, and I think a little less relevant to this meeting. So I'm going to focus on how bringing all of these components together led to a positive benefit risk assessment for use of an ECT. So eflornithine, or DFMO, received regular approval on December 13th, 2023, to reduce the risk of relapse in adult and pediatric patients with high-risk neuroblastoma who demonstrated at least a partial response to prior multi-agent, multimodality therapy, including anti-GD2 immunotherapy. And the context for this was a high unmet medical need in a rare, life-threatening disease. The clinical evidence submitted was evidence of efficacy based on an externally controlled trial comparison of study 3B and ALNB 0032. The investigational arm was a single-arm trial of 90 treated patients, and the external control arm was an NCI-COG-sponsored clinical trial of 270 patients, propensity matched to DFMO treated patients on 11 demographic and clinical characteristics, along with additional strong mechanistic non-clinical data. And listed here are the outcomes for event-free survival and overall survival that reflect the protocol-specified primary analysis. Given the potential treatment estimation additional uncertainties with an externally controlled design, supplementary analysis were also conducted, as well as use of external statistical methods to ensure the robustness of the results. So let's talk just briefly a little bit more about the methods and talk a little bit more about the data source. So the source for this external control was the experimental arm of study ALNB 0032, which was, again, a multi-center open-label randomized trial. The propensity score matching was used to match on clinically important baseline covariates to build a model with a propensity score and exact matching to ensure balance across 11 key characteristics. And so patients who met criteria for comparison had complete data for those specified covariates were matched 1 to 3. And the 11 clinical covariates used in the model, including an exact match for MICA status, which is considered the most important predictor of relapse and survival outcomes. These clinical characteristics reflect the expected demographic characteristics for high-risk neuroblastoma. And it is important to note that this was a very robust process, and the data set had all of this data available in order to do the matching process. So this application specifically, given the primary evidence of efficacy, was from an externally controlled trial, and available confirmatory data was from non-clinical studies, went to ODAC, or Oncologic Drugs Advisory Committee, in 2023. And during this discussion, there were uncertainties raised about the potential biases due to non-randomized design, as well as the limitations of animal models. However, there were a lot of strengths presented as well, including the large effect size, robust statistical analysis. As I mentioned, there were a myriad of analysis conducted, and the strong preclinical model. So the discussion highlighted challenges of the application, and including the feasibility of an RCT, and the committee voted 16 yes, 14 no, that there was sufficient evidence to establish efficacy. So as regulatory context, this is the first oncology drug approval which relies on an externally controlled trial as the primary clinical data to support substantial evidence of effectiveness. In the clinical context of a rare life-threatening cancer, for which there were feasibility concerns regarding successful conduct of a new RCT, this evidentiary package was unique. Each component was essential in supporting its approvability, including the high-quality external control arm in a comparable population, the consistent estimation of the treatment effect through numerous sensitivity and supportive analysis, the manageable safety profile, and the strong non-clinical mechanistic data. So even in the context of a severe disease with a high unmet medical need, without each of these evidentiary elements, an application relying on an ECT as the primary basis to support efficacy would have been unlikely to have been successful. Randomized controlled trials remain the strongly preferred development approach to demonstrate an effect on a time to event endpoint, even in rare cancers. So to provide just a few concluding regulatory insights, I want to finish up by summarizing thoughts around the use of ECTs. With rare exception, RCTs are required to assess effect of a drug on time to event endpoints because randomization balances known and unknown prognostic factors. And it is often challenging to use, very complex. And I think all of these presentations highlight the complexities associated with the design of ECTs. Of course, fit for use data can be a rate limiting factor in establishing data for an external control as a formal comparison. The appropriateness of that data is a first step that's often not reached in a drug development program. That's step one. And so where randomization is not feasible, ECTs may be useful to inform drug selection. And it's important to think about how careful design and selection might be just key factors in even thinking about moving through this going forward. When reliable objective responses or low magnitude effect are not expected, development plans may move quickly to a randomized trial. So in conclusion, I think it's really thinking about trials that include external controls and their significant complexity. And if sponsors are thinking about these designs in their drug development programs, it's important to evaluate this early and discuss the potential methodological complexities. So with that, I'd like to thank our wonderful team at the FDA and all the individuals that contributed to this. And thank you so much for the opportunity.
Thank you very much, Donna, for sharing your experience and your knowledge about the external control used in what concerns to regulatory decision making. Now we will move to EMA use case. It will be a joint presentation by Andrea and Theodore. Andrea works as a product lead in the Office of Advanced Therapies and Hematological Diseases and has been acting as a product lead for Abecma, which will be the case that will be presented. And Theodore has joined EMA in 2020 and works as a biostatistician in a data analytics and methods task force. Thank you very much, and the word is yours. Thank you.
Thank you. Good afternoon to everyone. So indeed, as already introduced, I will be providing some introduction and introductory details on Abecma, and then I'll leave like the flow to my colleagues to provide the details on the external control and historical control study. So Abecma. Yes. So the content of the presentation provides introduction on Abecma, indeed, on regulatory context, main results, and details of methodology of external controls. Here is the usual disclaimer. And just for you to know, the results shown in this presentation are mainly based on the EPAR for the initial approval and the SMPC, so all published data. Abecma, also known as idecabtagene vicleucel, ide-cel, is by Bristol Myers Squibb, is a CAR T-cell product, and it has been the first CAR T-cell approved for treatment in relapsed and refractory multiple myeloma in fourth line at the beginning when it was initially approved. It was approved under conditional marketing authorization, and the European Commission decision was received on the 18th of August 2021. He was a beneficiary of a priority medicine scheme at the beginning, prior to submission, and it is an orphan product. So in general, the indication at time of approval was for treatment of adult patients with relapsed and refractory multiple myeloma who have received at least 3 prior therapies, which includes an immunomodulatory agent, a proteasome inhibitor, and an anti-CD38 antibody, and have demonstrated disease progression on the last therapy. The approval was mainly based on a pivotal study, the MM001 KarMMa-1, also known, and was a phase 2 open-label single-arm multi-center study that evaluated the efficacy and safety of Abecma in the same population as described in the indication. Of course, there were also like some supportive studies. The CRB401, which was a dose escalation study, provided also like very preliminary efficacy data. And as mentioned, there was the use of historical control data through the study NDS-MM003, which is a global non-interventional retrospective multi-center study to generate real-world evidence of subjects with relapsed and refractory multiple myeloma with prior exposure to an anti-CD38 antibody. As mentioned, the pivotal study, KarMMa-1, was the basis for the approval, and the efficacy data that have been used to approve the product were the following. So the efficacy was indeed assessed on the basis of overall response rate, which was the primary endpoint, complete response rate, which was the key secondary endpoint, followed by the duration of response, secondary endpoint, as determined by an independent review committee. Other key efficacy endpoint included minimal residual disease using next generation sequencing. And as you can see highlighted in red, these actually are the numbers that you can also find in the published SMPC. Based on this, I will give the floor to my colleague Theodor Framke to provide insight on the study design, external control, and methodology of the historical control study that has been conducted for the analysis of Abecma.
Okay, thank you very much, Andrea. I will now give you a more detailed overview of the study design, choice of external controls, and methodology used for NDS-MM003. This is restricted to the time of the conditional marketing authorization and mainly based, as already mentioned, on the EPA. Note also that the CMA was based on a single-arm trial with ORR as the primary endpoint. So this was the KarMMa-1 study, and I will refer to this study during my presentation multiple times. In addition, a comparison with this single-arm trial with real-world data was submitted. This external control can also be seen as a single-arm study, external and composed of real-world data. It was not pivotal for the CMA. There were some limitations and uncertainties when it comes to, for example, time to event endpoints, and it can be seen as some contextualization. The submitted evidence based on the real-world data was assessed, and it was decided that it will not go into the SMPC. And to give it a place, it was discussed and presented in the EPA. So I'll now go into the data sources here. So this external study was a global non-interventional retrospective study set up to generate the external comparison for KarMMa-1. Data from sources including clinical sites, registries, and research databases were collated in a single data model and then further analyzed. There was an application to the PRIME scheme. So this is an acronym for priority medicines, and eligibility was granted in 2017. PRIME products benefit from enhanced support from the EMA. And scientific advice was recommended here. Also, clinical efficacy was discussed during the scientific advice procedures. And now coming to the cohorts here. So subject level data was collected on 1,949 real-world subjects who had relapsed refractory multiple myeloma and who had received at least 3 prior regimens. So that's the broad cohort here on top of this table. From this cohort, subjects were subsequently selected based on having met eligibility criteria for KarMMa-1 as closely as possible, including initiation of new therapy after becoming refractory to the last regimen and lack of comorbidities. Subject selection based on these criteria identified 190 subjects, and this is here in the second line, the eligible cohort, with similar characteristics to the KAMA1 population. Matching would have led to an even further reduction, so this was not pursued. The selected cohort of 190 real-world subjects from the eligible cohort provided an adequate match for about only 80 subjects of the ABECMA cohort. So this were 140 enrolled and 128 treated with ABECMA. As a result, the primary analysis had to be changed. Trimmed stabilized inverse probability of treatment rating was used for the primary analysis method for the effectiveness endpoints. And a number of covariates were considered. Missing data is also an issue in collecting information on real-world data sources. Matched pair and untrimmed stabilized IPTW analyses were also utilized as supporting analyses. Compared to the ABECMA cohort, patients in the eligible cohort were slightly older. So, for example, the median age was 64 versus 60.5 years, somewhat less heavily pretreated. So the number of prior anti-myeloma therapies was 5 versus 6. And the proportion of patients with at least one prior autologous stem cell transplantation also differed, and they had a lower degree of refractoriness to prior anti-myeloma therapies. I'm now coming to some of the results here. So this slide shows the results for the time to event endpoints. Note that so for PFS and for OS, please note that the colors are not consistent across the plots here. So the lower Kaplan-Meier line represents the external comparator. The upper line is from the ABECMA treated patients. The results as such were considered clinically relevant and significant. However, there were a few limitations, and I would like to highlight some of them that were noted by the CHMP here. So despite extensive efforts to match these patient populations, the comparisons were limited by several factors, so including the rather long time period allowed for the collection of baseline data. So that was up to 60 days from the index date. Then we had the overlapping recruitment periods for the real-world data and the KAMA1 at the same study centers, the large proportion of missing data, which were up to 30% for some of the included covariables. Several covariates were excluded from the propensity score model due to more than 20% of missing data. The eligible relapsed refractory multiple myeloma cohort were treated with currently available therapies as the next line study treatment. There were more than 90 different regimens, and approximately 70% were able to receive 3 or more drug combinations at their index therapy. And then on my last slide, I have a couple of general considerations here. So we're talking here about a single-arm trial. There are several issues with this approach here. So early submissions, they are frequently based on single-arm trials, especially in oncology. They may have high overall response rates. The question is whether this is then reflected in PFS or OS, and whether this translates into a benefit of patients. The study is uncontrolled here, so the reference is missing. The question would then be, what would be the causal interpretation? Well, one way of dealing with design shortcomings would be to provide a reference by contextualizing contextualizing the single-arm trial with external data. This may come with sometimes quite strong and sometimes untestable assumptions, and may add additional uncertainty. Potential unknown confounders and other differences cannot be fully addressed by propensity scoring methods. So the impact cannot always be precisely quantified. There's also the question of representativeness. So is the real-world data, is it reflecting the trial, and also vice versa? P-values are generally for this approach discouraged, as it would suggest a higher level of inferential independence. There are also challenges with time to event endpoints in non-randomized comparison settings. So this may be prone to bias, might be challenging to find a adequate common baseline here. I think this has been mentioned a couple of times also today, like the index date or day 0, I think. Then the isolation of the treatment effect is not always clear. Also, the overall response rate for the external data set was 32% here, which differs from the threshold that was used as for the null hypothesis in the KAMA-1 study, and this was 50%. So that's also a difference. Then I would like to briefly mention the draft guide on the SMPC 5.1 here. It also has a subsection 6.4.10 on single-arm trials, where it's explicitly mentioned that the ORR may be able to isolate the treatment effect in a single-arm trial as opposed to PFS and OS. However, there is another explicit sentence on single-arm trials in the section, and I'll quote from it. So the presentation of comparisons of single-arm trial data to an external or historical control group is generally not appropriate due to efficient control of potential bias. So what we can take out of this is that here, I think it can be seen as there is a need to be very confident with what is presented in such a document. To conclude, it was highlighted that the few submitted studies at the time of the conditional marketing authorization were not considered comprehensive enough to fulfill the requirements for the full marketing authorization, but an RCT was already ongoing at the time to provide more comprehensive data package in the future. This external study was considered as a supportive. It was not the main driver for the CMA decision. I think this was an insightful exercise, and the efforts of the sponsor were acknowledged. Thank you.
Thank you very much, Theo and Andrea, also for sharing reflections from an EMA submission that used the real-world data-derived external control, and in particular, reflecting a bit upon the role it had from the assessment perspective, the uncertainties that were perceived, and also importantly, how this was reflected then in the communication from a regulatory perspective, namely the EPA or the SMPC. And now, in the interest of time, we're going to move to the next phase, which is we have the pleasure to have some panelists also for this session who are going to share some reflections on the presentations that were just given, but also on the topic overall. And we'll stay in the hybrid world. So the first panelist that I have the pleasure of introducing is Dr. Mehmet Burku, who's a senior director in epidemiology at MSD, and he's leading a group of epidemiologists there for real-world evidence studies. But on top of that, he's also involved in several cross-sector methodology development initiatives on advancing methodology in the context of real-world data. And so, Mehmet, the floor is yours.
Thank you so much, Marcia and Carla. But quick sound check. Can you hear me okay?
Yes, we can hear you.
Perfect. Well, good morning, good afternoon, or good evening, depending where we are. As you mentioned, we are in the hybrid world. And thanks for the kind introduction. And many thanks for the presenters earlier for very informative presentations. And thank you to the agency for organizing this workshop. So building on and reflecting on prior presentations, happy to provide some perspectives on external controls within the context of real-world data and also some challenges associated with them. But first, as I alluded earlier by many speakers, external controls are not for every situation. As we all know, there needs to be a clear, appropriate clinical and regulatory context for their use, and they are usually reserved for special clinical and regulatory circumstances, mainly to support single-arm trials in high unmet need situations. Why? Because in these cases, randomization is not feasible or unethical due to lack of clinical equipoise for most part. So in such unique situations, then the question becomes, can external controls using real-world data be helpful as opposed to having no control, no comparison of any kind? Well, it depends on the clinical context, obviously, and it also depends on regulatory context for sure. But if they're constructed appropriately using appropriate fit-for-purpose data set, if available, they can be helpful to help interpret results from single-arm trials depending on the clinical and regulatory context. That being said, however, they're not simple, they're not easy, and they're not straightforward, as we all heard throughout the discussion earlier today. There needs to be a lot of thinking and a lot of consideration that needs to go in the evaluation. First, the feasibility of an external control arm, whether or not if it is at all feasible with the available data sources, and then if feasible, a lot of thinking that needs to go in the construction of an external control arm. And because of the underlying data that is routinely collected healthcare data, and I would like to underline routinely collected as opposed to protocol-driven clinical trial data, there will be some degree of uncertainty in many situations. Therefore, it is critical to mitigate the uncertainties to the extent as feasible. And reflecting on excellent presentations provided earlier at a high level, I would like to also quickly provide perspectives on 5 high-level themes and challenges, and at least from design and planning perspective to mitigate uncertainty. First, it's related to the timing of external controls, meaning whether patients in the external control data are from contemporaneous data, historical data, or combination of both. Usually, as we know, contemporaneous data is preferred, but we also know that that may not be always feasible, especially in ultra-rare indications. So depending on the clinical and regulatory context, it will be very important to understand in a transparent fashion the degree of uncertainty the timing of external controls can bring. The other aspect is related to the regions of data. We heard that in the prior presentations as well. As we know, many single-arm trials, if not all, are international trials. But at the same time, we also know that having access to real-world data sources in each country represented in this multinational clinical trial is likely not going to be feasible as well. So therefore, depending on the clinical context, the standard of care, access to care of that clinical context, it will be important to assess, prioritize, and select appropriate geographic representativeness and sites of care for a particular external control arm, and also understand in a transparent way what's the level of uncertainty that the choices will bring. The next is related to index date, which is this time zero, which was highlighted earlier as well. This is very important, and this is related to how to establish the start of follow-up in real-world data setting in relation to the single-arm trial, where follow-up usually begins at the time of randomization. It is very critical to mimic clinical trial situation in the real-world cohort so that we can mitigate multiple biases, including the so-called immortal time bias when it comes to index date issues. The other aspect I would like to mention about is the variables, meaning the covariates, confounding variables, or your inclusion-exclusion criteria. In all likelihood, realistically speaking, most real-world data sources will not have all the variables in the universe. So we know that. Therefore, at a minimum, it will be important to rank, understand, and evaluate the availability and accuracy of must-have data elements and must-have inclusion-exclusion criteria in the real-world data sources. And from the same token, given that we know not everything will be potentially available, it will be also important to understand the degree of uncertainty of unmeasured or missing data will bring, and it will be important accordingly to potentially conduct as feasible sensitivity analyses among many other approaches to mitigate uncertainty. Finally, another issue I would like to also mention is related to follow-up and endpoints. For example, endpoints in clinical trials may not be always available or assessed in similar fashion in real-world routine healthcare settings. So therefore, to mitigate detection bias, evaluating, developing, and as applicable, validating endpoints that can be used in real-world settings are quite critical. And we also look forward to cross-sector validation efforts for many endpoints on that one. So that's also important in terms of methodology development efforts. So in sum, there are many important design and feasibility assessment factors that need to be considered in establishing external controls, both at feasibility as well as in the construction phase. But in the absence of randomization and due to the underlying routine healthcare data, it is critical to mitigate uncertainty to the extent as feasible. To achieve that, external controls do require careful, detailed, and transparent planning. And also, they do require adherence to best methodological pharmacopy and real-world evidence principles so that we can minimize bias and confounding, decrease uncertainty, and hopefully produce credible, actionable, and reproducible evidence with external controls in unique, high unmet need clinical situations so that we can contribute to totality of evidence as an evidentiary element. So with that, thank you for listening, and back to you, Carla and Marcia.
Thank you very much, Mehmet, for sharing these insights in terms of your view on the role that external control comparisons may have, in particular pointing out for contextualization of single-arm trial results, but also talking about the uncertainties and about a few mitigating measures that you see in that field in relation, for example, the data comparability as an important element when we talk especially about real-world data and clinical trial data. And with that, we will go to our next panelist. And actually, we are switching to the physical room for him. It's Denis Lacombe, who introduced himself quickly earlier, but I'm going to do him the honors once again. So he's Chief Executive Officer at EORTC. But in addition to that, he has expertise, quite some strong expertise in European policy affairs. So in that part, he is both the co-chair of the ACT-EU Advisory Group, but also the co-chair of the EMA Cancer Medicines Forum. And on top of that, he's a member of the EMA Management Board. So Denis, the floor is yours.
So thank you for the introduction and congratulations to the EMA for organizing this meeting. I think it's quite informative. First of all, I would like to say that I echo very much what has been said, especially by the regulators in this session. And maybe what was mentioned this morning is that I think we should take the best of the two worlds. And indeed, everything we have to learn from all the methodology. Something that we do, at least we say in our environment when people come for discussing design is, what is your question? And basically the question will inform, of course, what is the methodology that you have to use. So maybe you will need a clinical trial, maybe you will need an explanatory clinical trial, maybe you will need a more pragmatic clinical trial, maybe in some situation, indeed, you cannot randomize, you go for a single arm, and then you will go into the external, to using external control. What I would like also to say is that something that was mentioned this morning by Patrice, and that I would like to emphasize here, is that we have said also that clinical trials are difficult, but something, again, that was mentioned is pragmatic clinical trials. I think if we would make our trial or some elements of our trial more pragmatic, maybe we would solve some of the problem. Pragmatic clinical trials are not in the European definition of real-world data. It is in the US definition, so maybe there is something here to be addressed. What I would like also to say is that in our environment, we are often approached for indeed single arm and external control for rarer disease, for rarer cancers. And I like to say that the rarer the disease, the more you should consider randomization, because these patients are so rare that you want, of course, to bring them to the most informative methodology. Of course, I admit that there are situations, of course, where we cannot randomize. Something else also where I believe we should be more ambitious. Often also people say, I don't have access to the patient in my institution or in my city, or, okay, so it's not a good reason not to have a a proper design. So go internationally, try to, of course, develop the means of the ambition and bring the best to patients. So I think these are messages which I think are important, that in the situation that we are discussing in this panel this afternoon should be as much as possible a fallback position, and this has been quite emphasized by the regulator. Now, coming back to the external control, I think what is the most of concern, I guess, of course, is the quality of data, because indeed, what are we comparing to some extent? And I think it's important to dichotomize between data that have been collected retrospectively versus data that have been collected prospectively. And also something that maybe we should approach or discuss is how we can re-engineer to some extent the use of prospective registries and plug, of course, as much as possible, or use these registries for, I would say, high quality external control. So I think this is something that is important. I was quite impressed by the situation of the TK2 disease when it was said that this patient, there is no registry and they are underdiagnosed. So maybe if there would be a prospective registry, we would improve maybe the access to these patients, and we could do more, and companies could use this registry to do maybe proper research. So maybe here there is something that we could address. The last point also that I would like to highlight, that I should have mentioned earlier, is that when we talk about a randomized clinical trial, that was also the case for the TK2 disease with phase 3, and that maybe somebody will correct me. I guess that any form of benchmarking is better. It doesn't have necessarily to be a randomized powered phase 3. Sometimes indeed it's not possible, but any form of benchmarking maybe can be considered before again we go to maybe methodologies that are less informative. And of course, I think the DFMO example is a very positive example, although I would have a question for Donna, because I've seen that there has been, I guess, a cooperative group database of 270 patients that has been used. So I guess these people have access to patients. So the question is, why not a clinical trial has been done if there are so many patients out there? Thank you.
Thank you, Denis. So I think the question for Donna we're going to park for the moment, also to make sure that all other panelists can speak up. And but thank you for flagging the importance of the research question guiding really the evidence needs, and accordingly what from the toolkit in terms of study designs, data sources and methods is appropriate to answer a specific question, which I think we've heard as a key message throughout the day a few times now. So then we're going to move to our next panelist, and this is Jan Cornelissen. He's a professor of hematology at Erasmus Medical Center Rotterdam, but who's also working currently at EMA as a seconded national expert in the Office of Advanced Therapies and Hematology Diseases. And I think, Jan, you're connecting from Madrid. So,
yes, thank you very much. At the EHA in Madrid, and tomorrow we have a interesting EMA EHA real-world evidence session, which nicely follows what has been presented today. I would like to add shortly to what has already been said that, well, first some examples which we explored last year in a collaborative effort between the study group HOVON, EMA and Erasmus University. And the two studies we performed, and I think they were informative, and it's nice to not to show them here, but just to quote them. We tried to validate external comparators arm within a prospective randomized study. And these were two different prospective randomized studies which were performed by HOVON, International Prospective Randomized Studies in Acute Myeloid Leukemia, And we looked for real-world data, of course, outside these trials, and identifying them in the National Cancer Registry in Holland, and also in the Harmony database, which is a large integrated European database supported by EHA, and has data from tens of thousands of patients with hematological malignancies. So in the first study, we tried with a cohort from Harmony and the National Cancer Registry to identify an external comparator arm and compare it with the control arm in a randomized study in elderly AML patients. And what turned out, in short, is that the external controls from the Harmony database, a database which contains predominantly study patients, compared reasonably well after matching with the control arm of the randomized study. But the data from the National Cancer Registry, even after matching for age, disease risk score, performance, these patients fared significantly worse, 10% survival disadvantage. It's the second study, also performed a randomized study by Hovon in elderly AML. We identified a different comparator arm that was a comparator arm consisting of data which were registered prospectively in 5 different hospitals in the Netherlands via the digital hospital systems, via a newly developed program that uses a so-called sieve, in which overnight all the digital hospital data are screened for control patients, and these patients are then prospectively monitored until either death or end of study. And it turned out that in this study, even after matching, the external comparator arm fared, with respect to overall survival, 10 to 20% better survival, to our surprise. And we still cannot explain this. So, and the point I would like to make is that when we're going to use external comparator arms, we need to validate the external comparator arm. So not only identify one source of external control patients, but try to find somewhere around the world another external comparator arm in order to validate it and to see whether we have a more or less reliable comparator. And in this collaboration between Hovon and EMA, we also did a retrospective study looking at phase 2 studies in hematological patients, which were published the last 25 years in literature, and used an external comparator arm, and just looked at the methodology. And it turned out that there were serious flaws with respect to statistical analysis plan, with respect to follow-up, with respect to the application of in- and exclusion criteria, with respect to the use of missing data, and so on and so on. And not one study fulfilled the criteria to which a comparator arm in a randomized control arm should fulfill. And all in all, this turned out, at least in my view, to be worrisome. And therefore, I would like to make a plea that the randomized control arm is still the golden standard and should be the golden standard, and the regulator should be a bit strict. And if there is an external comparator arm, ask for validation and be as critical as possible. I say it a little bit bluntly, but that's maybe for the discussion. Thank you.
Thank you very much, Jan. And also for bringing up that same terminology that Mehmet has been using earlier in terms of validation, really, of the external control arm to kind of be a bit provocative in terms of the really, or rather pinpointing down the importance that the data quality there has. And now, before I hand over to our last panelist, Bruno Delafont, I would like to reassure you, Bruno, that apologies to everyone in the room, but we will spill over a little into the coffee break. Otherwise, it would give you only 1 minute to talk. So Bruno is a statistician who works at ANSIM, the French Medicines Agency, but he's also co-opted member at the CHMP with expertise in biostatistics and clinical trial methodology. Thank you, Bruno.
Thank you, Marissa. Can you hear me okay?
Your volume is a bit low, or
Is it better or?
it's still a bit faded, I would say.
And now?
Better, thanks.
Okay, thank you. Sorry about that. Yeah, so thank you, and thank you very much for all the clear and then stimulating presentations. I think this perfectly illustrated the value, but also the uncertainties that we're facing with external control arms in general, and more specifically with external control arms based on real-world data. I liked the reminder from Elina's introduction that the bar is high for the demonstration of efficacy in a benefit-risk assessment, which explains why the conduct of randomized controlled trials remains, of course, the default standard way to provide confirmatory evidence for a new treatment whenever possible. And yeah, and this also explains why, from a regulatory perspective, the many uncertainties and so the multiple sources of bias associated with an external control arm, that explains why they frequently prevent the relevant use of external control arms, or at least that why they create a number of challenges. And there were several questions raised at the end of the industry case presentation. So Morial's case with the treatment of TK2D. The questions were regarding how to demonstrate the acceptability of such approach and how to demonstrate the robustness of an external control arm based on real-world data. And I think it is, of course, very difficult to answer for this specific example without further knowledge of the specific clinical context or without further knowledge of the trial setting and real-world data source. But what I would say is that what is definitely encouraged, and I think this was the scope of the second question as well, is to engage early with regulators whenever an external control arm is considered. And this would be typically through requests for scientific advice. And I guess first, in general, this would be to discuss whether the use of real-world data as external control is actually adequate to address the research question, because we know, and I think this has been described in the various presentations too, that this will depend on a number of factors. This will depend on the clinical context, whether the natural course of the disease and its heterogeneity are well understood, whether we have an endpoint that can isolate the treatment effect, and then, of course, whether the real-world data are of sufficient quality, whether they can be matched reasonably with the trial data and based on the main confounding factors. And then, when we are in the situation where an external control arm might be suitable, then from a regulatory perspective, I would say that what is likely to matter a lot, and that's been mentioned by others before as well, is the transparency of the approach taken, so that the whole process can be understood and retrieved, and of course the need for pre-specification too, because prospective planning is important in general, but probably even more in the context of an external control with real-world data, because it's likely to be even more difficult to exclude data-driven decisions otherwise. And I think all of this was also highlighted in the second presentation from the FDA, which I think again clearly highlighted the specific situations where external control arms could be suitable, as well as all the steps taken and considerations to assess data comparability, there was also the presentation of the individual case where the external control arm was used as primary data for FDA approval. But again, we could see how the uncertainties were raised and taken into account and triggered some challenges. And then the EMA case about Abecma was also interesting to follow because we could see here that although the comparison to the external control arm was not the main piece of evidence, it still made a contribution as a supportive element to the assessment. But again, the uncertainties were recognized. And this leads me to the point that the use of external control is a challenge on the regulatory side, also from a pragmatic point of view. Because as we said, there are many potential sources of bias at various steps in the process. And we say that of course, we wish for as much transparency as possible, but the consequence of this, of course, is that this often represents for the assessment team some extensive review of the relevant documentation. And when some pieces of information are lacking, this might also trigger more questions at the time of assessment. And this is another reason why I agree that further regulatory guidance on the topic of external controls will definitely be helpful for all parties involved. But I think this will also be the topic of the next session. And I should probably stop here to leave time for further discussion, if any.
Thank you very much, Bruno. And you're actually already partly moderating into the next session. Thanks for that. Unfortunately, so first of all, thank you for flagging two elements. I'll keep it really short. The clinical context as something that has been mentioned a lot in various presentations, and the importance of that for the acceptability to determine whether there is actually a place for an external control comparison, but also highlighting again something we also saw in a few slide decks, the pre-specification as an important element. And with that, I have to unfortunately let you know that there won't be any further Q&A because it's Friday afternoon and we've been instructed to remain on time. But I would like to thank all speakers and panelists again for the great contributions to this session. And then everyone can enjoy a short coffee. Peter?
So thanks very much to our co-chairs, to the speakers, to the panelists. We're going to have a brief coffee break. I'm going to be a brutal tyrant, and you're going to be back in here at 20 past, because we're going to start bang on time for our last session, where Jeppe and Kit will help us navigate the road to guidance for the future. So grab a coffee. Let's be really naughty. Nobody's listening online. If you want, you can bring your coffee back into the room, but don't tell anyone, okay?
Good. While the last couple of people are finding their seat, I will start up this final session of today. It's 20 minutes past 3 here Friday afternoon, and I know there's also a lot of people who have either a train or a bus or a plane home. So also with no further ado, let's make sure we keep the time also to make sure that people can get home. We don't all of us have to eat dinner here in Amsterdam. But here the final and third session, I think, to copy Harold a little bit. This is also the most important session of today. We have been looking very much on what is actually the current practices. We've been looking at what have a good paper, which has also been the reflection paper on real-world evidence, which we have had some good perspectives on as well, which we need to also take account into moving forward. And now we have come to what I'll call the crystal ball, and we're going to look into the future a little bit and see what is it that's coming down the road. I think we have had some good sessions also for today, where one of the things, key takeaways I think that's been is also regarding building bridges, both between the regulatory areas, between industry, regulators, and academia and healthcare sector, to gain a better understanding and also to have a better dialogue between each other. And then also, I think it's also leaves us also a little bit time for reflection. Also moving forward is what is it actually we need to bring in based on all these discussions and presentations we have had today. In the break, I was approached and asked if we could have a little bit more time for discussion. And I don't know if it will be possible, because I think we should be done at 4:30 with Peter having his final say. So, I will also kindly ask both the presenters, but also the panelists to be brief in their presentations and also keep the time slots in that sense. So, what we're going to look into now is a short introduction to the methodology working party by Kit and also Olaf Klunge, who will look into the roadmap for the development of the real-world evidence guidance. Both Kit and Olaf have already been presented previous today, so I hope I won't go through the entire mentioning again. So with no further ado, I think, Kit, what's in the crystal ball for us?
Yeah, thanks for the kickoff and the opportunity. And indeed, in light of the time, but also the time I think we should also spend at the content of the roadmap ahead of us, I will aim to at least stick within time or be briefer. It's an introduction to the methodology working party. Yeah, great if it works. The methodology working party is one of many working parties supporting the CHMP in decision making, and it serves the entire European medicines regulatory network more broadly. So it's not only CHMP, but it's more broad. Established not too long ago, we're there since 2022, following also a recommendation of the Big Data Steering Group. And I think the most important point I would like to make here is there were working parties previously with disciplines organized. We had a biostatistics working party, we had a modeling and simulation working party, and we realized at that time that if you look at the developments which we are facing in terms of decision making, evidence needs, and the opportunities that the data are offering us in the broad sense, we need to pull those together. That's a more multidisciplinary problems that we need to attack. So it's good to actually have that capacity through together in one setting. That's one, and there is a need to build new expertise in the system. So that's briefly the background. Here, Patrice already highlighted what does a working party do, and that ranges from product-related support, guidance and concept papers, and that will be partly the topic that we will dive into more deeply, engaging with stakeholders, and also that intensity has increased over the past year, which I think is a very good thing given the shared challenges we have and building indeed the competence in the network. The working party itself has 23 members across the different disciplines which you can find on the slide. I would want to make one additional point regarding product-related support. We've seen the example of Abecma, and as you can imagine, this is not an isolated case in its importance. These are typically challenges which are not only discussed by the rapporteur and the CHMP. For those cases that emerge and that we need broader thinking, also the methodology working party or the broader network will be asked for advice to make sure that we actually get the expertise together, but also learn together so that we have consistency in the network. So that's the product-related support here and the learning that's happening in the network. Yeah, we're there. It's not only the working party. Associated with it and also active now is a European specialized expert community, and that's actually core to the success. And if you look at the bottom right, we now have 202 experts associated with the methodology working party, which consists of the assessors, the methodological assessors within the system throughout the European Union, as well as academic experts which have a close relation to interest or expertise that's relevant for the regulatory network. And we did foster the original disciplines. You can see that we have special interest areas, and if you quickly count, the numbers add up to more than 200. We do allow experts to have interest in more than one area. And for today's discussion, I think at the very least it's interesting that the largest interest is with the real-world evidence topic. But it also means that we are pooling resources here in terms of the expertise that we mobilize within the system. And we pull from this broader expert community in guideline development, in product support, and we reach out to them to share learnings and build the knowledge throughout the network. A few outcomes of our first 2 years, and I'm not complete here, but I've limited myself for those things that could be interesting in the context of the topic of today. there are draft reflections papers out there. We've discussed one this morning. There are others out there as well. The single-arm trial reflection paper, on a different note, but I think also relevant for this area, the use of artificial intelligence throughout the medicine product lifecycle. And we've also contributed from within this data, from this working party, to the data quality framework, which is out there and in my view quite instrumental across pretty much everything we will be doing in the context of real world data. Then, where are we in our planning? We're in the last year of the first 3-year work plan. I must admit that was very much built up from the basics that were there before. So still thinking in the original disciplines. We are now in the process of looking more strategically, and we've started working on our work plan, the rolling work plan for 3 years ahead and beyond. I'm not presenting a draft work plan because this is not the stage we're in. We are in the stage of actually interacting with all of you to make sure that we get the work plan right. So just over a week ago, we had an extensive discussion with, let's say, an overlap from who was all either here or also online to actually get feedback on where you think we need to prioritize in terms of drug development and guidance. And I also see today, and I already made quite a few notes for my own attention where we could benefit from. Also today, for me, is input into what we actually will prioritize on our agenda for the work plan. A couple of things which I'm pretty sure are there. Partly they are completing work that's already ongoing, but partly it's because it's quite clear it's needed and the message has gotten through. And external controls is one of them. And I do want to, and Bayesian statistics, platform trials are there as well. On a different note, but also in a development we see is quite important, predictive biomarker co-development. And as we also noticed with the AI reflection paper, there we are bridging to the medical device field. So I think, yes, we are on our road, and I think actually this is an important point to reflect on our work plan as well. And we will not stop with what we are aiming for in artificial intelligence with clinical development and pharmacovigilance as the first priorities identified there. A brief remark on external controls. We have the single-arm trial paper out there. There was a very deliberate choice not to include anything there on external controls. And for me, there are two important reasons. First of all, we are looking at single-arm trials, which somehow ended up in a protocol, were submitted to ethical committees and were executed. If that's the case, they should be able to stand on their own and answer the relevant research question of interest. If after the fact, external control is needed for answering research questions that apparently the single-arm trial was not able to address, that's a different matter. And we want to actually make that very, very clear. So that's a research question. And I think we should move towards having the research question clear and identify the settings where apparently external control or any control data is needed to answer that question. And then actually you're not in the realm of a single-arm trial anymore. So that's the thinking behind the single-arm trial paper. So we are taking that perspective for external controls as well. So maybe even external controls is not the best possible connotation. Is if you have a research question that left or right needs a proper control there, that should be identified at the design stage. And then we might end up with the preferred design of an RCT. If for one reason or other that's not possible, we trickle down. How far and how good can we actually get in terms of having controlled data there to answer the question of interest? So this is our initial line of thinking of having guidance on what I quote here as external controls. I think it's hugely important, but also really important to start at the beginning and at the design stage, because after the fact or after the trials conducted and adding external controls adds uncertainties that are almost impossible to quantify. So that's it, the thinking where we are now. One of the other, I will move quickly, a few notes on how, this is partly personal, how I think of guidance development and at the same time supporting innovation. What we've noticed already with the guidance that we have developed, that any guidance of substantial importance is essentially multidisciplinary. It's not restricted to one methodological area, but most of them by now also engage the clinical and other perspectives while we are developing. So it's kind of confirming that we indeed are on the right track to have a methodology working party. Scientific guidance, I also today, the logical question, what's acceptable? If I'm totally honest, I would love to have that disappear from the vocabulary. Because I think, and the thinking there is, if we are going to define very precisely what's acceptable, we are forced to be very conservative. That's inevitable. And then still we might be wrong. And you come with a particular case, and then that might be we might have actually still have a false positive. So I would rather have that we really focus on the key scientific considerations which are essential to consider for the decision making and the evidence we need at hand. So that's what we try to aim for in the guidance. I do take the note made earlier today, the purpose of the guidance and which problems do we aim to solve, and to be more precise there. I think that's I think a very important input we received this morning. So this is my note. Although I know that question is asked very, very often, and it reduces uncertainty for some, but I'm afraid not for all. High standards of evidence, I think it's in the interest of parties and of patients and society, and I see it as a joint effort, so not something that's only on the side of the regulators. Finally, crossing borders. We are aware that from the perspective of market authorization, we are somewhere there between a novel drug and the patient, and there are other decision makers. So although the remit is different, it's important for us to consider that the same evidence and the same trials and the same studies that we perform will also be used for other decisions. So I think that's going to be an important part of our thinking and our way ahead. As part of that, we've actually established coordination between the methodology working party, the clinical trial coordination group, and the newly formed methodology group under the new HCA coordination group to make sure that we align, and because we are quite aware we are actually providing guidance on the same trials and the same research being conducted. Second point is we are, I think the right call is, are there places where we can interact and have a discussion across stakeholders? Research projects under Horizon Europe offer that opportunity. So quite a number of MWP members are engaged in Horizon Europe projects. One of few were mentioned this morning in the area of real world data. They provide an opportunity to interact, also between regulators, academia and industry. And I see the big advantage there is that we can actually think about frameworks and concrete ways of how we assess novel methods while they are developed. So we are much closer to our thinking on how to judge novel methods than waiting until they're there and look at them after the fact. And I mentioned the interplay with medical devices is entering our agenda, and I think that's relevant. And I'll stop here, and thanks.
Thank you very much, Kit. I will wait with the questions, and I will pass the stage on to you, Olaf Klumel. If you ask you kindly to take the stage and give us a presentation on the methodology working plan. Thank you.
Thanks. Thank you. Yes, it's my pleasure to present our current roadmap from the Methodology Working Party for Development of Real-World Evidence Guidance, and this is on behalf of the Temporary Drafting Group. Also Carla, Stine, Xavier and Juan Jose, who are working together in this group. Yes. So before going into our plan, I think it's good to give a bit of an overview of the current real-world evidence guidance available across the globe, with mainly regulatory, but also some HTA agencies that are listed here. As you can see, and also presented already, I think by Donna Rivera, there's quite some guidance from the FDA available, some guidance from EMA, where I will go into more detail just later on. At the level of ICH, there is guidance mainly on safety assessments of medicines. NSEP, which is a joint effort by centers of expertise on pharmacoepi and pharmacovigilance in Europe, together with the European Medicines Agency, has developed a guide. And many of these principles have also found their way into the reflection paper that has been presented this morning. So I'll not go into detail in each of these agencies and the guidance that they have, but we did sort of a brief analysis of what topics were covered in these guidances, and that's maybe more relevant to look at right now. So on the left, you see all the different key topics and specific topics that have been addressed by guidances by the different agencies. So again, you see the FDA with many guidances. Good to point out here that we have from the European Medicines Agency a guidance on data quality, so the draft or the data quality framework, which has been published, and the let's say specific chapter on real world data that is ongoing and under development. The reflection paper that was presented covering design aspects of non-interventional studies using real world data, and there's also registry-based non-interventional studies that for which guidance is available by the European Medicines Agency. So you can see that for most of the agencies there is some guidance available on non-interventional studies using real-world data, so that's covered by most. But on specific topics, you can find some gaps, and that's also why we did this analysis, to identify gaps and where guidance might be needed. You can also see there's one specific topic on external control data to supplement control arms in clinical trials, for which there's no guidance available as far as we have been able to identify. There is some guidance on external controlled clinical trials, so from the FDA and also from NICE, more from the HTA perspective. And one specific guidance on pragmatic trials, so clinical trials using real-world data, from the MHRA. All right. So to summarize, the areas covered at the European level by the European Medicines Agency, we have the data quality framework available, and the follow-up real-world data deep dive chapter. So this will be very important for the assessment of data quality in studies using real-world data. So that will also soon be finalized. And then we have, of course, the reflection paper that was presented and the guideline on registry-based studies. So what are the areas of potential interest that we think are important to address? And also covered, I think, quite a bit this afternoon by several presenters. So using real-world data to support clinical trials, and mainly externally controlled clinical trials. So we can distinguish externally controlled clinical trials using patient-level data, which is preferably, I think, and at some point you could also consider group-level summaries, but this is more a crude comparison. And then external data to supplement control arms, and I will get into a bit more details later. Then thirdly, the clinical trials using real-world data we have identified as a topic, so basically pragmatic clinical trials. And maybe there are other areas of interest that we need to cover. So the roadmap is trying to address these topics, to try to prioritize topics for further guidance development. But also, I think this is something of a dynamic process. So innovations and future developments should also be considered and maybe needing also further guidance, or when certain methodologies have matured and are mature enough to be considered established, it's also something to consider for guidance. So I will not go into details here because this was already presented by Donna. So the FDA guidance on externally controlled clinical trials. It's good to realize here that is, if you see under B data considerations, data from control arms can come from clinical trials and can come from real-world data sources. So this is, I think, important that we do not completely separate these two, because in terms of the methodology and the research question that we want to address, these are similar methodologies and should be covered in an integrated way, I think. So this is what is covered in the FDA guidance. Then here, this is an area where there's no specific guidance available. It may also be a bit of a smaller area, and we need to consider this, I think, whether we want to have this under one umbrella of externally controlled data, or that it is a separate piece of guidance. So it might be applicable to situations where there's comparable control data from another clinical trial or real world data source, and then a clinical trial design that recruits less subjects in the control arm might be attractive because participants have a smaller chance to be allocated to the control arm, so you'd be allocated to the intervention arm. Trials could be shorter, and feasibility might be larger. And the clinical trial analysis combines in this way historical and current control data. So as I said, no current guidance available, but important, I think the last point just to highlight is that many aspects related to the design, data comparability analysis will be shared with externally controlled arms. So this is something that could fall under the umbrella of externally controlled data. And then the last topic that for which MHRA has guidance available, so clinical trials using real world data, basically pragmatic clinical trials. It's important to realize, I think, that these will be trials that are conducted on treatments that are usually already approved. So this is post-authorization. So this also drives the types of questions that will be answered by pragmatic clinical trials. And so my last slide is basically about, let's say, summarizing what we think are currently the topics of interest to further work on. So to prioritize, let's say, the use of external controls to support clinical trials. Also to define the scope of this guidance, which has not been finalized yet, I think, and consider both controls from real world data and clinical trials. Then secondly, also consider clinical trials using real world data, so pragmatic trials as a topic for further guidance development. And both of these topics have been mentioned today, and also these topics came also from our stakeholder interaction meetings between the methodology working party and stakeholders as areas where guidance would be needed. And we're open, of course, for future real-world evidence guidance development, but that's depending on what the future brings. So that's really crystal ball, I think. All right, thanks.
Thank you very much, Olaf, and thank you also for a very interesting presentation. I think it's linking it to previous presentations also on how to operationalize actually all the knowledge and all the experience we have within the academic world, and actually also to bring it into industry and also the healthcare sector. I think it's very linked also and to that. So thank you very much. As previous, we have also 4 panelists where I will shortly just introduce them also. And also thank you both to Kit and Olaf to be on time, so we have good time also hopefully for a little short discussion. I think we'll start up with Michael Schoenen, who has 15 years as a senior director, and 15 years within as I'm working with as an epidemiologist. Sorry for the word. And right now she's currently a senior director of observational research at Amgen's Center for Observational Research, where she leads the European team of epidemiologists, data science, and also programmers. So, Maike, can I ask you to take the floor and come with your reflections on from what you've heard so far?
Yes, thank you. And thank you very much for the opportunity to speak on behalf of the UCOPE Trade Organization. And this day has been very interesting, and so I'd like to take the opportunity to not just reflect on this particular session, but as I overheard the day, what made me realize is that everyone in this meeting and in this room, what brings us all together is our passion for real-world evidence, and in particular then also the passion for having real-world evidence to the highest standard so that we can be confident that the decisions are being made
off the back of this evidence that they are the right decisions. So then from the industry perspective, speaking about these guidelines, they are certainly extremely helpful in ensuring that the evidence is being generated to the highest standard. And then when we think of areas of focus or perhaps where there might be some gaps, they are broadly highlighted in 4 buckets, which you can see here on the slide as well. And maybe then starting around data and bearing in mind the time, keeping it fairly short as well. I think we've heard a lot about fit for purpose assessments already, and what one thing to highlight there is, in the crystal ball, I think we know with the European health data space becoming a reality, there's going to be more and more data and potentially also more parties trying to generate real world evidence. And so at the basis of all of that lies, of course, having trust in the data. So the data quality framework is extremely helpful, but what may be even then additionally helpful also is to be more specific and having an actual framework around what does that fit for purpose assessment mean? How do we do the feasibility assessment? And maybe some ideas around totality of evidence, when a feasibility assessment is made and it is found that a data source is not fit for purpose, or it is not feasible to do a study, where does that get recorded? Because that's essentially also evidence which is not currently necessarily available for everyone in the community. I think then maybe pivoting to methods, one area which is very fast evolving, and it was mentioned, the AI reflection paper. One thing to highlight, of course, also is AI in real-world evidence generation. What about using large language models to go deeper into the data, to get unstructured data and have that form the basis of a new data set? Where does that fit into the data quality framework? Do we have, how can we assess reliability of that? It may be helpful to have further guidance around that. And then maybe lastly to say around study execution, I think particularly studies where causal inference is taking place, it's very important. We've heard about target trial emulation, but what about perhaps also taking an approach in terms of the execution of the study that is very similar to how trials are being executed, such as, can we have a staged approach where we assess first whether the comparisons are as affected potentially by unmeasured confounding, and not advance to the next step of making the actual comparison unless you are satisfied that you're comparing apples with apples. And perhaps even going as far as a clean room approach, whereby the analysts who are ultimately involved with interpreting the information are not being exposed at all to some of the iterative decision making as the study is being executed. So very similar to a blinded execution of a trial. So I think, last point then around global harmonization. It was very impressive to see all the guidelines outlined on the slide that Olaf just showed. But of course, as marketing authorization holders, it is very helpful to have harmonization across the globe. So that really brings me then to the conclusion to state, these guidelines are extremely helpful. And I think it was mentioned before by others as well. These are helpful, particularly also in the context of creating transparency and trust in real-world evidence. But the guidelines alone are not going to accomplish all of that. I think the additional elements there are to make sure that we consider the totality of evidence that's being generated by stakeholders across the healthcare ecosystem. And lastly, very importantly also, I think the interaction between the many stakeholders, such as a meeting today, such as the public-private partnerships, etc. So thank you.
Thank you very much. And thank you also for leaving us with the notion of having passion about a common subject here in the room. That's always very nice. And I'm hoping that Viviana Giannuzzi, you are also up for taking the stage afterwards and also talk a little bit, hopefully, about the same passion. Viviana Giannuzzi. She is currently she is at the Fondazione per la Ricerca Farmacologica Gianni Binzi Unlus, where she coordinates the research department and participates in the EU and also national research projects. And in addition to this, also previously, she has also held the position where she was the member, where she represented patients' organizations and of the pediatric committee. So please take the stage, Viviana.
Thank you very much. Yeah, I will try to take the point of view of the academia looking at the future. So we heard that academia is playing a role in using real-world data for research purposes, but also in regulatory procedures like and not only in the research and development, but also in the marketing authorization application and price and reimbursement and HCA and so on. So academia is demanded to keep the quality of this data. Is demanded to collaborate, as we heard. There are new and upcoming research projects on, for example, implementing innovative methodologies to real-world data like the pharmacometrics approaches, and you are demanded to partner, to participate in public-private partnership. But I would like to underline that this happens much more in the areas where randomized clinical trials are often difficult or unfeasible, like pediatrics and rare diseases, to cover unmet medical needs, or where we have no an appropriate control in the trials. This is a reality. There is a quite recent publication from COMP members demonstrating that about 30% of orphan medicinal products have been authorized on the basis of real-world data to demonstrate efficacy, so not only safety. Another important point for academia is that real-world data is the main source of evidence of off-label uses. And it, of course, affects research in the repurposing field. Of course, it's needed to have a regulatory perspective here, because otherwise off-label will remain real-world data without any approval status. This demands continuous updates of real-world data sources. So if we want to collect data off-label, we need to update the approval status, the new indications, the new populations covered by indication. And there is something that affects the control arm, especially in the pediatric field. Sometimes it's unethical to use placebo if we have a well-established off-label use. So it's unethical to use placebo, but in theory, we must use placebo because the off-label is not authorized. So this means that we need maybe specific requirement to collect real-world data according to specific ages and subset, because we can have rare diseases that are subset of a more common disease. Today we heard about the advantages, but also the issues to be solved. And we need to have multi-database sources that are still scattered, and we need specific permission to access and use data for research purposes, because we can have very good quality real-world data sources, but without a valid consent, without the right permissions, without the ethics committee approval or supervisory authority, for example, we cannot use them. Another point is that real-world data sources and tools can be qualified by EMA through the qualification procedures or by FDA in other regions. And recent data demonstrate that data on the basis of the qualification of these tools does not contain lots of data on pediatrics, for example, and on rare diseases. So what we need maybe for the future, we need to collect in real world data in adequate duration. For example, if we would like to look at adverse event, we need to consider the long-term effects of medicines in children. We need to collect in the relevant population and divide the source according to different age groups. Otherwise, we cannot extrapolate data if we want to take the pediatric one. And this should be considered in the context of use of all the research and regulatory procedure. Maybe we need to evaluate the need for specific measure to collect real-world data for on-off-label uses. Ethics guidance, so academia need to engage not only with the regulators, but also with ethics expert, patients, industry, and health management systems, because real-world data strictly rely on the national system. And I would like to underline that within the European Joint Programme for Rare Diseases exist already tools to enhance the quality of data, for example, verifying registries, or the template to collect informed consent within ERNs. To conclude, I think that academia has now a great responsibility if we look at the upcoming rule from the new pharmaceutical legislation that will possibly apply shortly. Don't know how shortly, but there will be a kind of revolutionary rule there, because academia will be demanded to submit data to regulatory authorities to make a repurposing use and in-label use. So I think that this would require academia to have really good data and to be presented to regulatory authorities. Thank you.
Thank you very much. And also thank you for putting the perspective of both the academic world, and also actually the importance of having the data also in a right level, so we can also use it actually in a couple of years of going down the line that way around. For our third panelist, we also have George Palioras, hope I pronounced it correctly, who is both a member of the Big Data Steering Group, and in addition to this, he is also the research director and head of artificial intelligence at the lab SKEL at the National Center for Scientific Research and Demokritos in Greece. So, George, can I ask you to take the floor?
Yes, thank you.
Thank you very much.
Introduction. Although I am doing all the things that you said, primarily I'm here as a patient representative, and I am chairing the Duchenne Data Foundation. So this is the perspective that I'm trying to give today. And I have to agree with the previous speakers that today was a very interesting day for me as well. I enjoyed very much the enthusiasm that people shared in bringing forward the use of real-world data. And with this statement that I would like to make now is, I would just like to encourage to do more and to take bolder steps in the same direction as you are already doing. In this respect, in this context, there are clearly issues that may worry us in using and going further in the use of real-world data. And so what I wanted to say is that there are a few issues that we may want to deal with and move forward. One of these being, I will start with some that have to do specifically with the participation of patients. Increasingly, patients do participate in the design of many of those initiatives and activities that are happening. So what I was saying is that increasingly patients do participate as representatives like myself in the big data steering group, but also in other groups. But when it comes to studies, to clinical studies, patients can have a more important, a more active role. And not only patients, but even citizens that are not patients. So I think we need to move from the stage where we have patients participating at the design stages to be patients being more actively involved in providing additional data like patient experience data or interpreting data. In some cases, the perspective of patients can be very important in interpreting the data that we get, or even in assuring the quality of the data that is being generated. So that's one of the things that we can do further. Another aspect that we're discussing also in the context of the EHDS is how much can citizens in general, how much would we like citizens to control the use of their data? And in many cases, the technical aspects of that seem too hard to overcome. At the same time, I think we need to rethink and consider that, giving the citizens the control of their data, because it may actually provide an opportunity to have citizens actively providing their data in the studies that we want to run. So I think we need to find ways to make it more, to make the involvement of patients and citizens more active in this context. And so these are the 2 points that I wanted to make regarding the participation of citizens and patients in studies and in the new real-world data environment. I have a couple of more general remarks. Clearly, as we move from the controlled environment of clinical studies to real-world data studies, there is some fear that we may be losing control of some of the parameters of the study. And this is understandable, and we want to avoid it to some extent. But at the same time, we have to realize that the real world cannot be controlled. And in some ways, it should not be controlled. Some of the useful properties of the real world data come exactly from the fact that it cannot be controlled. So I really enjoyed today to see interesting, innovative approaches on how this can be reconciled. And it's a direction that I would like to see more in the future. Another point has to do with using data that already exists. And I know there are efforts to do that. There are of course many reservations in going for retrospective analysis of data. But if we take into account the potential volume of existing data for some of the studies that need to run, it is clear that looking back into the near past might actually give us a head start in some of the studies that we want to run. So looking at existing real-world data that has been collected in the near past might be a useful thing to do. And finally, I want to mention one aspect that is always a concern about me, and especially, and I think it's the same in all patients that deal with degenerative diseases. That's the aspect of timing. We often have a reservation that we may be moving too quickly into the unknown, into what is not fully known. And this, of course, creates worries. You know, are we doing the right thing? Are we moving too fast? When thinking about that, we should always think that of the opposite. issue, which is that any delays, anything that we do more slowly comes with an opportunity cost. And this opportunity cost in some rare diseases has an effect on people's lives. So moving quickly is important. And especially when it comes to the real world data, the real world at this point is accelerating, especially in terms of technology. We see AI and many other things that are moving so fast that if we try to slow things down, our plans, our strategy may be obsolete in a very short amount of time. So we need to take that into account in whatever plans we make. In conclusion, what I wanted to say is that we can do more, and I would like us to try to do more. But at the same time, I have to repeat that I was very pleased with today's meeting and the enthusiasm and the innovative ideas that I've heard. And thanks very much to everybody who is making those efforts.
Thank you very much, George. And also thank you for putting in the perspective of the opportunity cost and actually also focusing on say we need to also do something, continue our work with a certain speed to make sure it doesn't come too great. Finally, we have Professor Jona Agash, who is a professor of allergy and clinical immunology at the Transylvanian University at Brasov in Romania, and who is also currently the chair of the EACI Research and Outreach Committee, who will also give her perspective from the healthcare professionals. So can I please ask you also to take the stage, give your perspectives?
Good afternoon, everyone. And I realize that I'm the last speaker between all of us spending the Friday afternoon. So I will try to be brief and keep, some space for discussion and questions as well. So first I would like to congratulate you in putting together such an exquisite panel of experts discussing today health research methodology, evidence synthesis, decision making, which is important, and also big data analysis. As I'm also a member of the Big Data Steering Group, and in these years that I've been there, I learned a lot because I think it's a very good place for dialogue, and I would like to thank you for this opportunity as well. So what I would like to bring you today is the perspective of a clinical guidelines developer and what the hurdles that we have with the real world data and how we are trying to overcome this in the way that we increase the quality of the recommendations that we formulate. So as you know, for developing clinical guidelines, we use this multifaceted approach to inform decision in a transparent but also in a structured way. We call this evidence to decision criteria. And here is where traditional study design intersects with the so-called real world domain. So in this process, we included several non-randomized studies, either of intervention and of observation, using them as complement, sequential replacement. We apply a methodology looking at congruence between RCTs and non-randomized studies based on thresholds. That's fine. So the methodology is there, but when we starting analyzing the non-randomized studies, we coming from the so-called real world, we had the issue of transparency and of understandability of the data that we are working with. And we realized that the simple use of this term, just real world evidence, without clearly specifying the source of the real world data, entails several practical limitations due to a lot of ambiguities that are there. So for us, it became very clear that it's imperative to adopt a transparent and flexible approach to categorize and interpret evidence, besides the usual, dichotomia that we have randomized and non-randomized and real world. So trying to move away from the use of real world evidence in isolation and as I was mentioning, reporting on data sources is important. Also, if the intervention assignment was predetermined by the protocol or it was routinary. and whether the data were collected for research, so primary research data, or derived from routine practice, because this is really different when you analyze them. And I see, and I am very happy that we have the data quality framework that is currently being there for the regulatory use. We hope that we can expand it a little bit more to all end users, if possible, in the future. And also for us, it will be very, very helpful if we have some pragmatic approach to all this, because we are not, all experts in methodology. So it will be very, very useful if we have the guidance from the regulatory in a brief format, which is easily understandable for all end users. So with that, I would like to close my intervention today and looking forward to discussions and questions.
Thank you very much, Iona. And also thank you for some really good perspectives. And I would say thank you for all of both panelists and also presenters for being right on time. I think we have time for 1 or 2 questions from the room. I can see Jin Wang. Silvanto, you have a question?
Hi. Okay, I'm on. Hi, this is Jin Wang Silvanto from Astellas. I want to congratulate really very enjoyable discussions and presentations, and also such forward-looking reflection paper on RWE. So my question, I actually have 2 questions, also quite future-focused. One is about what are the next steps of reflection paper once it's published, and whether we have updates and also developing to guidelines in the future. The first question. And the second question is about actually how to implement those guidelines. Because at the end of the day, we want to utilize this opportunity to accelerate the high quality RWE across the stakeholders. Do we have more opportunity to have dialogues and to develop implementation tools and frameworks to make sure we can use it properly? Thank you.
Thank you very much. And I'm looking at you, Olaf. Do you have a perspective, perhaps, to give an answer?
Or maybe the first question. So first, let's finalize the reflection paper and get it out. It's for public consultation until the end of August, and then I think we should people should read it, use it, and as guidance. And obviously, when the time's right and there's sufficient new information, there could be updates, but that's a bit difficult to say now when and how we're going to do that. So first, I think we need to work with what we have right now. And the second question, I don't know if that's for me.
Maybe I can help with that. Yes. I think it's very, as regulators, we often think that writing a guideline is the end of the story. Actually, it should be the beginning of the story. So I just want to acknowledge your point about outreach, implementation, engagement, training, etc. Now, I think there was quite a lot of debate within the network as to whether this was a reflection paper or a guideline. And it was called a reflection paper partly because there are difference of opinion about what weight should be put on real-world evidence. And it was made it a bit of a more safe space to call it a reflection paper, if you like. People could then be a little bit more innovative in the drafting. And I think that's probably good. I think my personal view is it gives some guidance. And we should then engage with stakeholders once it's adopted as final to leverage and increase its impact, I would say. Now, how we do that, I will leave to Kit and Olaf and so on. By the end of the year, I'm sure they'll have a master plan.
Yeah, a very brief comment on that. If the reflection paper is out there after having received the comments, it's there and should be used. And it holds for applicants, for academia, but it also holds for regulators. It should also guide us in the way we approach the discussion we have on any proposal. So in that sense, I'm in line with what Peter mentioned. We call it a reflection paper, but it will guide our discussions. And I think that's the essential use we will all have to make of it.
Thank you very much, Kit. I will leave the last question for you, Jesper Kjær from Novo Nordisk.
Thank you, Jeppe. This is to Kit, both a reflection upon how nicely laid out you had the work around real-world evidence. I think it's kind of an elegancy from the European perspective of how these guidelines and reflection papers come about, going across some of the thematics.
I think seen from a global perspective of how we as an industry submit globally, right? Real world evidence also plays an increasing role in other regulatory frames. And I think as we build our European health data space with some of the work around that, we will have a network to access the data, yet will that data ever be possible to send to the FDA for approval as they actually expect to get on a patient level? And in Europe, as we expand on receiving data for the assessment for market authorization applications, how does that look in the future? Is there a role in there? And I bring this forward, Kit, because I know you're involved in a number of projects also looking into synthetic data and what that role could play. Maybe there's a room for the methodology working part to think about the divide we have globally of submitting data on a patient level versus having federated data access to real-world data where there's a lot of sensitivity around people's control and knowledge about how this data is being used. Just curious about the methodology working party thinking into synthetic data, how that reflects the statistical assessment, etc., what else not. Maybe a topic for the future.
Thanks for putting the challenge out there. Maybe one you are aware that actually we are building very quickly, I hope, experience in the European network, actually receiving data and how we do that, and we will probably do it differently than the FDA because we're in a different place. I think your broader question on a global level, we already face that problem within Europe. So I would kind of turn it around. If I see what's happening across a number of the innovative research projects, I think we will be shaping tools that will allow analyzing data without having to move them. And once that's there, that's globally. And I think actually we have, in Europe, we probably have a position to do that more quickly and more fed by the data as it's structured in Europe.
And I hugely welcome that. It's just there's a legislation divide here between the US and Europe to be aware about, right?
Thank you very much. I'll close the floor now for questions and just say thank you both to presenters and again to panelists. And speaking of passion, I have promised the last word to Peter, who will do the closing remarks now.
Thanks very much indeed. Well, what a great day we've had. I mean, I genuinely think that was a great day. We've moved on from talking about the generalities of real-world evidence to talking about the specifics. And I think that's really, really good evolution. So real-world evidence can produce impactful evidence. It can produce impactful evidence on disease epidemiology, on treatments, on safety. And we've also heard about the fact that the primary demonstration of efficacy, and I think we all knew that coming into this workshop, normally will be demonstrated by a randomized controlled trial, except in exceptional circumstances when real-world evidence will contribute and can contribute and perhaps should contribute. I think we've heard that the research question should drive the choice of evidence. And my co-chair is Danish, so I don't think today is about Danish by design. I think it's impactful by design, because we heard it throughout the day that if we, at the design, the protocol stage, think through very carefully about all of the questions and perhaps use the target trial emulation, think about the estimand, we will have more trustworthy evidence at the end. So impactful by design, plan, plan, plan. We believe at in the European Regulatory Network that we need to embrace the spectrum of evidence, and that includes randomized controlled trials and real-world evidence, and we will support future work in this area. By embracing the spectrum of evidence, we will have faster drug development, we will have optimal and optimized regulatory assessment, and we will help to fulfill unmet medical needs of patients. I'd quickly like to thank the EMA team, particularly calling out Chantal Quinton, Juliana Fodge, Gosha Durka-Grabowska, and Valerie Muldoon from the Control Centre in Haarlem. I'd like to thank the Programme Committee, the chairs, speakers, panellists, and everyone for engaging in this really important topic. At the EMA and in the European Regulatory Network, we're enabling use and establishing the value of real-world evidence, and we're doing it step by step together with stakeholders. So thank you for a great day. Have safe travel. Thank you.